Hello and welcome. I'm Noa Rappaport. I'm a principal scientist and a computational biologist at ISB, the Institute for Systems Biology. ISB is a nonprofit biomedical research institute located in Seattle, and we are an affiliate of the Providence Health System. We focus on some of the most pressing issues in human health, including aging, the microbiome, COVID-19, brain health, cancer, and many chronic and infectious diseases. ISB also focuses on improving K-12 STEM education and beyond. In particular, we're addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn much more about ISB by visiting our website at isbscience.org. As a nonprofit research organization, we rely on the generous support of our donors. If you would like to support the scientific and educational work we do, you can find a donate link at the bottom of the event website. We acknowledge that we are on the traditional land of the coastal Salish people, including the Duwamish people, past and present. We honor and gratitude the land itself and the Duwamish tribe. Before we begin, I'd like to express our thanks to the sponsors who helped make this day possible: Phenome Health, OptiSpan, Olink Proteomics, and the UW Nathan Schock Center of Excellence in Basic Biology of Aging. Because of their support, we're able to provide this virtual work course and symposium free of charge to over 600 people from all over the world. We're grateful for your time today, and we're excited for this opportunity to help train the next generation of computational aging researchers from all around the world. If you're on social media, please remember to share your experience throughout the workshop using the hashtag ISB Systems of Longevity. And if you tag ISB, we are ISB SCI. This year, we are focusing on frailty, which is a measure of age-related decline. We will explore different ways to develop a multi-omic signature of frailty as a way to get mechanistic understanding. We will start with a keynote presentation by Professor David Furman from the Buck Institute Research of Aging. And then the first session will focus on multiomic analysis of frailty through an exploratory data analysis and clustering techniques. The second session will be focusing on statistical strategies for classification and longitudinal trajectory analysis. So we will learn about discriminant analysis, which is a supervised approach, and on regression via generalized linear mixed models, which is in an unsupervised manner. The third session will introduce concepts in machine learning and how it can be used to infer mechanistic understanding through jointly analyzing multiomic data. And after the hands-on session, we will conclude the workshop with a short seminar featuring talks from Dr. Jesse Poganik and Dr. Cara Green, followed by a discussion led by myself and by Professor John Newman from the Buck Institute. If you are having trouble keeping up, please feel free to deposit your questions in Discord. Our dedicated TAs will monitor it throughout the day and answer any questions. Remember that the course is recorded and the materials will continue to be available following the workshop. Our hope is that by the end of this day, you will have the tools to analyze your own multiomic data. I'd like to introduce our amazing instructors, Drs. Lance Pflieger, Max Robinson, and Alex Ralevski. Our TAs will be behind the scene answering your questions: Dr. Kengo Watanabe, Dr. Dantong Zhu, Dr. Gwenlyn Glusman, and myself. We also thank Dr. Andrew Mages, the Director of Data Science at ISB, for his technical assistance, and our events and communication team: Dominic Lewis, Alison Kudla, Joe Mixter, Audrey Hubbard, Connor Kelly, and Ray Rosario. A few technical things. It's our first year running a workshop using Amazon SageMaker Studio Lab. This is a cloud-based data analytics platform that runs Jupyter Lab. We'd like to thank SageMaker team for working hand in hand with us in the past few days to make it possible in order to allow you to access securely a real-world data set. But we're sure there will be hiccups. The instructions on creating your login and accessing your Studio Lab space were sent last night. Your data access will only be active until our computational funding is exhausted, but the code and videos will continue to be available. If you run into any problem, use the tutorial that was sent to you. And if nothing works, our TAs will help you on Discord. If worst comes to worst, we place dumps of the Jupyter notebooks on GitHub under each session folder. So you'll be able to follow the instructors using those. And now I'd like to hand it over to today's keynote speaker, Dr. David Furman. Dr. David Furman is an academic entrepreneur with expertise in aging biology, systems medicine, and translational immunology, focusing on aging and precision longevity. He serves as the director of Stanford 1000 Immunomes project at the Stanford School of Medicine, and also serves as an assistant professor and director of the bioinformatics and data science at the Buck Institute for Research on Aging. His work leverages multi-omic technologies and advances AI and machine learning to track health and identify biomarkers to reverse and prevent age-related diseases and conditions. He founded several spin-offs, authored over 46 articles, and holds more than 25 patents. David, take it away.
Okay. Thank you so much, Noah, for the invitation. And thanks the ISB for organizing this terrific event. I'm going to start sharing my screen. I hope this is okay. I'm assuming it is. If it's not, IT people can help me here in the chat. So I've been focused on immunology for over 15 years. At Stanford, we founded the 1000 Immunomes project to study how immunology drives aging and aging-related diseases and see if we can intervene. And been with the Buck Institute for about now almost 5 years. It's a terrific place to do science, really strong community, and we're very collaborative. And so the idea here is we're doing systems immunology for a variety of reasons, but I love to cite this joke where kidnappers take an immunologist and a cardiologist, and the kidnappers say, we are going to shoot one of you, but they promise to spare whoever has made the greatest contribution to humanity. The cardiologist says, well, I've identified drugs that have saved lives of millions of people. And impressed, the kidnappers turn to the immunologist and what have you done? And the immunologist says, well, the thing is, the immune system's complicated. And the cardiologist says, well, please shoot me now. So I think there is this idea the immune system is highly complex. Cardiologists don't want to hear anything about immune system. No one wants to know all the jargon. It's complicated. It's a complex system. And so there's a challenge for us to be medically relevant because it's so complex. There's no single target. There's no single pathway. And so we're using systems immunology to understand the complexity of the immune system. And this is a nice visualization of how the immune system could work. Different cells and cytokines and genes interacting with one another. And you could be really good at studying a single component, your favorite CD4 T cell or your favorite protein, but you're going to miss these clusters. These clusters are important. When you study more things, now we're doing a lot of different multiomics to try to understand that network that may be, driven by aging or a particular infection. And that gives rise to emerging properties. And that's the concept of systems biology or systems immunology, that we're studying this from different perspectives using multidisciplinary teams and a network analysis to see if we can capture those emerging properties. And when you look at the actual data, and this is data from 1000 Immunomes in a Bayesian network type of approach, you can see that the age effect is pretty large. These are different gene modules that contain over hundreds of genes, different cell types, compositions, and so on. And you see a lot of correlations between the aging and different parts of the immune system. in different ways, up and down, and cytomegalovirus is also important, BMI and gender. The idea of systems biology is actually not new. It started probably with Bertalanffy in the 40s, 50s, and 60s. And he came up with this concept that these are open systems. So anything that happens in one side of the equation or the system will affect other things in the same system because these are open environments. And so that evolved with time. And I love this chart that talks about the complexity sciences and how they evolved from the early 40s with Bertalanffy here and the cybernetics. So information technology has evolved in parallel. And there's a lot of overlap here. And systems biology was only proposed as a term later in the early 2000s, and we're here, right, in 2020 or 2024. We're talking about data science, which is very different from bioinformatics, for example, or informatics that you can get a whole bunch of p-values. In this case, what we want to see is understanding those p-values and really gathering biology and knowledge. So it's the application of knowledge and the tools that we have to really understand what's going on in the immune system as it relates to aging. I don't have to convince you that chronic diseases are #1 killer and economic burden, over $4 trillion annually in healthcare expenditures, up to 90% of the total expenditures in the US and worldwide. And I've been focusing a lot on inflammation. You probably all have heard that inflammation is important for aging and related diseases, but we didn't know much, not until the past 10 years. It has been proposed that inflammaging, this concept of inflammaging that was coined by Franceschi in the 2000s, participates in each one of these different diseases of aging. And so if we could study inflammaging and understand what are the causes and consequences of this, we could possibly resolve many of the different diseases of aging. I just have to mention that acute inflammation, which is probably what you have seen when you cut your finger, it gets red, swollen, warm. All those signs are acute inflammation, signs of cardinal acute inflammation. So largely driven by infections, right? And we have established biomarkers for this type of age-related sterile inflammation. We don't have those markers, and I have a lot of data showing that IL-6 is not a good one. CRP is the crap protein. We actually, cardiologists now are calling it crap because it doesn't really predict anything well. And so we set out to study chronic inflammation in a more comprehensive and unbiased fashion using systems approach at Stanford, and we put a really strong clinical core, a technology center that had all the omics possible, and then with bioinformatics and the right immunological expertise, we have been able to produce new models, and we're testing new therapeutics as I go. And so that created the 1000 Immunomes project that was founded in 2007. We've got around 70 million from NIH, and we've been studying proteomics, all the gene expression and cell frequencies and cell stimulations and a number of different things, everything from blood, not really too much different solid tissues. And one of the things that became pretty important in the field of inflammaging is the inflammasomes. And these are molecular scaffolds that enable the maturation of two major proteins that are pyrogens. You don't want those to have high levels in circulation, IL-1 beta and IL-18. And we've seen that many of the danger signals that come from the environment, DAMPs, danger-associated molecular patterns, are actually activating the inflammasome in different ways. And we published a paper in 2017. This is already an old paper, but people forget about these things. And I think it's quite important because it talks about the exposome, how the exposome drives inflammation, and how that really can stratify different extreme clinical and immunological states. And I'm showing here some of the damps that can activate the inflammasomes. So here we have nanoparticles, silica, cholesterol, crystals, asbestos, things like that that are recognized and by different receptors. And then there's expression of inflammasomes that are these scaffolds that I'm showing you here. And that enables maturation of IL-1, pro-IL-1 to IL-1 beta by caspase-1 as activation. And you have production and secretion of mature IL-18 and IL-1 beta. And so we've heard asked the question, we have longitudinal data in the 1,000 immunomes that we follow for now 15 years. So at that point in time, we only have 5 or 6 years of data. And so we asked, are the gene expression changes in the inflammasomes increasing with age? And that's what we saw for different types of study or different years in the study. Overall, you see an increase in gene expression of those inflammasome gene modules. And when we stratify individuals into inflammasome module high and inflammasome module low, by looking at how chronic these inflammasome activation are in time. So if they were increased in more than the population in the top quartile, in 3 out of the 5 years, the individual was classified as inflammasome module high, and the same if he was in the protected, low expression of inflammasome genes. And it turns out that people in the high expression of these, most of them were hypertensive, and only 10% in the inflammasome module low. And when we looked at the mortality, because we looked at these individuals over time, they died over the study. And those having low expression of inflammasome module 62 and 78 actually are still alive in 2016. Those with high expression died. They passed away, okay, by 2016. So it's associated with mortality. We looked at inflammasomes in the context of metabolism. We looked at around 700 metabolites in people with inflammasome module high and low, and we identified 2 very key metabolites, adenine and N4-acetylcytidine, that are present in high concentration in individuals with high levels of inflammation and inflammasome module high. Now, when we put these compounds in animals, we're able to increase the infiltration of different T cells in the kidneys, and they develop hypertension, and they have this massive activation of different immune cell types in the blood. And so this is really important. We're looking for different countermeasures for this type of inflammation that is driven by the inflammasomes. And we found that the caffeine is actually an adenine antagonist. And there's a number of studies demonstrating that caffeine consumption can reduce all-cause mortality. And I'm sure you guys are enjoying this part. Or at least I am. I actually bought my espresso machine I had in my at the Buck Institute because I believe it's actually pretty good at preventing this type of inflammation. And so I'm going to show you some of those data. So when you look at expression of inflammasome genes, caffeine is negatively correlated with the expression of these both independent inflammasome gene modules. Now, when you ask the question, in individuals with inflammasome module high and low, how are the caffeine and caffeine metabolites distributed? Well, caffeine and its metabolites are high, very high in people with inflammasome module low, which means that this may have an implication to decrease the expression of these inflammasome genes. And we actually measure that. So we took monocytes and we've done a lot of different analysis showing that this NLRC4 inflammasome is elevated in the presence of N4-acetylcytidine, which was the metabolite that we identified. And then when we treat that with caffeine, signal 1, which is expression of this inflammasome, goes back down to baseline levels. And so interestingly, caffeine was picked up by many media and reporters, and I was called for like a couple months. It was bombarded by interviews and stuff like that. Nobody really cared about the story about IL-1 beta and mortality, but they were basically showing that coffee can be the fountain of youth, which is not really the case if you consider many other parameters. That's probably only one among a large list of different things. So how much do we know about inflammaging? Well, not really, not much. People talk about IL-1, but maybe not TNF alpha. IL-6 is definitely not a good one. People have mentioned C-reactive protein, but then HSCRP, so high sensitivity C-reactive protein, it only gives you about 0.6 in the prediction of in the area under the curve to predict cardiovascular disease. So we're really puzzled. And it's really interesting. There's a lot of studies demonstrating that this is really complex. And when I hear the word complex, I think it's an ideal case for what we're trying to do here, which is trying to understand the network type of structure in the data and how that leads to emergent properties. So I'm not going to touch upon this because I have a lot of other stuff I want to show today, but we set out to define inflammaging to construct this inflammatory score and to build reference values for systemic chronic inflammation. We use an autoencoder, which is a type of deep neural network based on the 1000 Immunomes project to build an inflammatory clock for aging. That inflammatory clock for aging predicted multimorbidity, frailty 7 years before it kicks off. So we measured this in 2010. The frailty, this is the predicted frailty score based on the inflammatory age, and this is the observed frailty score in 2017. We're doing quite well at predicting frailty. So Noa, 4-year studies moving on, we should discuss. I think there's a lot that can be done to integrate inflammatory age into your analysis. And then we also looked at immune function. Overall, people with a high IH have low immune function, which is somewhat counterintuitive, which means, so in aging, you have this inflammaging, but also immunosenescence, right? So the immune activation that you see in older adults basically saturates the system. And then when you get a virus or a vaccine, you don't respond as well. We also see ventricular remodeling in these individuals, and we can actually correlate that with mortality as well. So I'm just gonna go a little bit broader and say that the immune system has major changes with aging. And so I'm showing you only one subset, that's the terminal effector memory cells. You have a huge amount of these type of cells in older adults. And if you take many other cells, you can build an immune clock, right? And so this is your immune clock, and many of these cells have very nasty consequences for humans. So they accumulate in hearts. This is showing in the carotid arteries plaques that you can observe a number of different T cells that are accumulating in those plaques and predict cardiovascular events. And so we decided to look to see if there are other types of alterations in cell identity during aging with age. So here, what I'm showing you on the left-hand side is the analysis of 1K1K study. I think, okay, for some reason I lost, sorry guys. Okay, hope you see this well. If not, just let me know in the chat box. Let's see. Okay. So what you see on the left-hand side is the analysis of one case study where we had data for 1,000 individuals that PBMC single cell sequences was done on these 1,000 people. So you know that there's reference values and reference data set for identifying the different clusters of T cells, B cells, and all these different flavors of cells. And you can estimate how far or how close different cells are with respect to that reference data set. Or you can also do that with respect to the centroid of that cluster. So there's a couple ways of doing that. And so what you see on the left is the correlation of the proportion of cells exhibiting low annotation score compared to the reference data set. And the number one cell type that has the largest lost or losing identities and the naive CD8 T cells, you can see here, and the memory B cells as well. So that's kind of the 2 or 3 different cell types here that lose identity with aging. So what happens is we believe these cells are becoming stem cell-like. And so we're losing that identity. This quite crazy, pretty interesting. And what you see in the lower part of the plot is that these cells are actually becoming more similar to the reference data set. So the correlation is negative. And we see that in both type of analysis. So in the right-hand side, you see the naive CD8 T cells. There's an increase in the distance from the centroid, and the B memory cells, the same story. And then for the CD4 terminal effector memories and CTLs that are shown here, that is the opposite. So that means that there's more cells that resemble that cluster, the centroid of that cluster on a UMAP. Now, something that we've been looking for some time is also GTEx, the GTEx database. And the GTEx database, I should explain this a little bit, is a database of cell sequencing from different tissues of postmortem individuals of biospecimens. So we have for 54 different tissues, we have gene expression measured in all of them. So I asked Fei Wu and my team to look for enrichment of immune genes and aging genes in these aging tissues. So individuals are taken in different times of their lives, and they have different ages. So you can actually have this aging enrichment. And what you can see is that for 51 of the 54 different tissues, there is a significant immune gene enrichment associated with aging. So again, I think there's 2 things that happen here. One is that there's infiltration of immune cells in different tissues, and you're picking up that signal in the transcriptomics level, or there's this loss of identity where cells become immune-like. And I think it's both. And we've seen that in a different way, as shown here. This is overlapping different immune genes and aging genes, and you have a significant overlap of both. And when you look at different hallmarks of aging on the right-hand side, for the hallmarks of aging that are listed here, there's a significant immune enrichment annotation here for things that go from innate immune response to inflammatory response to things like immune system and so on and so forth. So for each one of the hallmarks of aging, you see different immune gene enrichments that are important for us to study, because then we can map which hallmarks of aging are driving or are consequence of this different derangement in the immune system. So I'm just gonna quickly walk you guys through some pretty interesting results we've had over the past couple of years. This started with a collaboration with NASA. They contacted me to see if we can help them develop countermeasures for space flight exploration. And it turns out that astronauts suffer from high degree of risk for cancer and cardiovascular disease, and they have also viral reactivation at large, and they basically have some sort of immune dysfunction as well. So we can simulate microgravity on Earth, and this is a rotating wall vessel that keeps the cells in free fall. So you're rotating cells at the same time that they're falling. So this is a simulation of weightlessness or microgravity. And we've done a lot of different things using cardiac organoids, immune organoids, PBMCs, brain organoids, and so on. I'm going to focus on the immune system today. So we looked at data from data with Inspiration4, JAXA-6 twin study, and we also did the simulated microgravity on the ground. We compared those. We repurposed some compounds that were treating these cells in the rotating wall vessel and see if the transcriptomic changes that are induced by microgravity and that are age-related can be reversed. And so the first thing that we observed is that monocytes are largely affected by simulated microgravity. Also the conventional dendritic cells with low antiviral response and lower interferon signaling. And we also observed that there increased coronavirus pathogenesis enrichment for those type of gene sets. And so we measured interferon alpha of all these subtypes in the cells that are in microgravity unstimulated. And you can see that there's a significant decrease in alpha interferon across the board. So it's quite interesting to see that latent viruses can be reactivated in microgravity, and not only in our system, but also in space. And so we observe that infection by RNA virus goes up, viral infection goes up again. This is all based on network analysis. But when you look to see if using the meta-transcriptome detection by Fei Wu, my team, we can identify that there is actually increase in gamma retroviruses and total viruses in simulated microgravity. And we believe it's due to changes at the level of the cytoskeleton. And so the cytoskeleton would recognize different viruses and will induce a type 1 interferon response. The cytoskeleton can also respond to different mechanical forces in the environment and produce a number of inflammatory mediators. And so we measured cytoskeletal changes. And what you can see, this is microgravity, this is 1G. There are these different spikes that are quite interesting. And the cytoskeleton, this is the actin cytoskeleton, is changing dramatically in microgravity. And what you can see here is the confirmation in different ways by looking at actin mean intensity and also the spike length, which is increase in microgravity. We also see increase in IH. I mentioned the inflammatory age goes up in microgravity, and also cellular senescence, mostly in monocytes, as you can see from this plot down here. So we could take an approach that is the GCNA approach, the gene compound enrichment or enrichment analysis, and we selected one of the compounds, that quercetin. We treated the cells in the simulated microgravity. We were able to reverse up to 70% of the differential expressed genes. We drove down inflammatory age and Sen-Mayo cellular senescence type of signatures, and reactive oxygen species also go down in the presence of quercetin when you expose the cells to simulated microgravity. We've been also working, and this is the last part. I think I'm running a little bit late. Maybe I'm okay here. This is the last part. I have a slide here to share with you guys, is about using tonsil organoids to simulate in vivo responses to different types of antigens. So what you see in this figure here is the workflow. We take tonsils from young, middle-aged, and older adults. We dissociate those tonsils to form single cells and then freeze them down. When we have enough that we want to compare between younger and older, we take those cells, plate them, and they aggregate. And those are the immune organoids that we're using. And what you observe here is when you toss a flu vaccine, the live attenuated flu vaccine, you see an increase in plasmablasts, in flu-specific IgG, in the treated versus the unstimulated one. You also see antigen secreting cells. So the system's working. It's a beautiful system. And then we see a decrease of that response in older tonsil-derived organoids, which means that we are recapitulating what happens in vivo. This is absolutely powerful. Imagine we put this in microgravity to screen for anti-immune aging compounds. That's exactly what we're doing. So we took an adapted organoid that we put in a hydrogel to be able to maintain those organoid structures healthy during the rotating system in microgravity. And so we have that for 1G and the stimulated microgravity. And what you can observe in this volcano plot is a huge upregulation of many anti-viral immune signatures, including the MX genes, the OAS, the IFID genes. All the interferon response is upregulated. But is that really resembling what happens in vivo? Yes. So we took data from a study that was done in individuals, in healthy individuals that were vaccinated with the flu vaccine. Day 1 after vaccination, you see a very similar upregulation that we observed in our system. So what I'm showing you with my cursor here is the stimulated control 1G. So it's basically the same that you see here. And we observe that the selected genes that are upregulated day 1 after vaccination in vivo can actually go also increase in our system. And this is the microgravity. So microgravity actually drives that down. So in other words, when you look at overall the overall picture, which is down here, this is a rank hypergeometric type of analysis where you can compare different studies, right? So here you have the stimulated and unstimulated based on the vaccine that we added to the organoids in 1G. And the upregulated genes will be ranked in this way, and the downregulated genes here. And in here, you observe the microgravity versus 1G, no stimulation. Okay, so just the pure effect of microgravity. And what you observe is that the genes that are upregulated in the stimulation are downregulated in microgravity, and those that are downregulated with stimulation are actually upregulated. So we're causing this accelerated immune aging phenotype in tonsil organoids that we can now use to understand several things. So for example, if we do a time series, and this is only 25 hours simulated microgravity, but imagine you do 3, 6, 9, 12 hours. And so you can start looking at the causal relationships of those genes that are predictive of the poor response to the vaccine. But then you can also put the cells back in 1G and see how those cells recover after 1G. So you can have an idea of the intrinsic mechanisms to rejuvenate that immune aging that was caused by microgravity. So with that, I'd like to thank my team and many other members that I didn't have time to put in the slide, but and this is an old picture. We should definitely update this one. But a very solid team. They like to go surfing and they have parties. And thank you everybody for your attention.
Thank you so much, David, for this great presentation. We'll break now and we'll be back at 9:45 for session number 1. Thank you.
Thank you all.
Right, great. So everyone, welcome to session 1. Hopefully you can see my screen. The session 1 is meant to be a hands-on session, and it is a multiomic analysis of frailty. My name is Pflieger. I am a data scientist at Phenome Health and a senior data science at Phenome Health and a visiting scientist at ISB in the Hood Price Lab. So before we get started and actually dive in, I want to just go over some resources that are available to everybody in the workshop. So the first is our Discord server. This will be used for questions and answering. It'll also be used for technical support. This is a live demo and hopefully nothing goes wrong. But if you need help logging in to AWS or anything like that, then this is where to go for support. I also encourage everyone to interact in the server. There's a bunch of us that all have the same interests, and it's a great place to network. As Noah mentioned earlier, we're going to be using Amazon SageMaker. This is a cloud-based analytical platform. It has access to all the raw data in a secure environment. It'll have access to the Jupyter notebooks that you can run along with me, and it will provide all the visualizations. We do have a GitHub for this course. This will be where you can download the slides, PDFs of the slides, and also PDFs or HTMLs of the notebook. So if you have problem running Amazon SageMaker, then you'll still be able to see what we're doing. The GitHub is linked at the bottom, and the link to Amazon SageMaker is here on the slide. If you're gonna go along with me, go ahead and click it now so we can make sure everything is up and running. It should bring you to an AW Access portal if you're not signed in. If you need to sign in, go ahead and do that now, and then click the Amazon SageMaker button. This is going to bring up a bunch of apps, and the main one we're going to be using is Jupyter, and this is where the notebooks are. Hopefully nothing else should work, but go ahead and click on Jupyter, and it should bring up a couple spaces for you to use. I actually don't have a space running, so I'm going to fix that by creating a space, naming it, changing my image, and running the space. So for everybody else, you should see in this screen a space running, and well, now my other space is running. So go ahead and click open. I have 2 going now. And you should see a Jupyter notebook. If you're not familiar with Jupyter Notebook, it's just a coding platform. Over here on the right is the directory or data tree. Click on Aging Workshop, click on Session 1, and we're going to start with the single omics analysis. Hopefully what you see is a Jupyter notebook, and it should be running an R kernel, which you can see over here. I'm going to change my settings so that hopefully everybody can see it a little bit better. All right, so to make sure everything is working, click on this first cell that says setup. All this is doing is loading a bunch of R libraries, and you can run the cell by clicking play or clicking shift enter. What you should see is it goes from empty brackets to brackets with a star in it. So everybody is kind of testing and seeing if everything is up and running. I'm going to just go through some introductory slides, and then we'll get back to the notebook. So the goal for session 1 is to really do a basic exploratory analysis. We're going to start pretty low level and move high level by the end and do some WGCNA, which is what Dr. Furman mentioned in his analysis. But first, since this is an aging workshop, we're going to kind of do a little deep dive into our phenotype of interest, which is the frailty index. I'll give an overview of the Aravel dataset, and I want to say that this is just a really unique and cool opportunity to be able to work with a raw data set. We're going to do a systems approach, or WGCNA, to a single omic, which will be proteins. If we have time, we'll compare that to a standard differential expression analysis. And then finally, at the end, we'll explore a multi-omic or an integrated multi-omic analysis with WGCNA. I encourage you to stick around for the second sessions that will explore the outcome in a little bit more depth, and the third session, which will be based on machine learning. So what is multiomics and systems biology? Well, biology is a complex field. I don't think that that's really hard to say. I think Dr. Furman explained that pretty well. But many processes that occur in our cells or across tissues are not driven by single molecules, but by complex hierarchical networks. And a breakdown of any of these molecules in the network can lead to disease. Systems biology is all about unraveling how biological networks, or biological models, interact at a systems level. And thanks to technological advances, we now have the capability to simultaneously measure comprehensive data that represents various molecular levels within a cell, and these levels are often referred to as omics, and the combination of which is multiomics. So to start very basic, there's a bunch of omics that everybody's probably heard of. Genomics, which is the measure of DNA sequencing, proteomics, or the measure of proteins, which play crucial roles in both cell structure and function, and metabolomics, which examines small molecules often involved in metabolism. And remarkably, we can obtain all of these data from just a small amount of blood, which allows us to track measurements over time. So multiomics can also be enhanced by some other information, which I've kind of listed out on this slide, such as wearable information from Fitbits, electronic health records, self-assessments, gut microbiome, and more. The integration can offer a broader perspective on physiology as we age. And in reality, the reality of being able to generate all these measurements at an acceptable cost is that the knowledge generated from multiomic analysis will soon be implemented into a clinical setting. So I think it's really important to understand the underlying methods of multiomic analysis. But what we're here to talk about is kind of aging. So I'm going to define what our outcome of interest is today. And we're going to be looking at frailty or frailty index. It's worth noting that there are multiple ways to define frailty, but for our purposes, frailty is a state of impaired health that arises from an accumulation of health deficits. The frailty index is simply a measure of an individual's proportion of health deficits. Well, what do I mean by health deficits? These can be things like disease status. Do you have diabetes? Do you have cancer? Do you have metabolic disease? Or questions around levels of activity. Are you able to walk? Are you able to shower? Questions around mental health. Do you feel happy? Are you depressed? Are you lonely? Or it can come from a range of outer range values from well-known clinical labs, such as HbA1c. So why are we looking at frailty index? Well, it's an inherently holistic approach that captures a heterogeneous phenotype. It's well validated with multiple measures and multiple publications, and it is a measure that can span across data sets because of how health deficits are measured. The actual deficits can vary as long as they capture multiple systems. I'll talk about that specifically with Arabelle here in a minute, but the frailty index is also the best predictor of mortality in humans and obviously associated with multimorbidity. But the final reason we're looking at frailty index is because it is a nice proxy for aging. So how do we calculate a frailty index? And as I mentioned on the previous slide, it's pretty simple. It's just taking the sum of health deficits for an individual and dividing that by the total number of health deficits measured. So if you have a frailty index of 0, that means you're not frail. You probably are not going to the doctor very often. Whereas if you have a frailty index of 1, then you're incredibly sick. In reality, if you have a frailty index of 60 or 70%, then it likely leads to death. So there's kind of a theoretical maximum around 0.7. So there are a couple things to consider when calculating a quality frailty index. And that's first, it should be associated with health status. And that kind of goes back into making sure that the deficits that you're measuring are associated with health. It should increase with age generally. It doesn't saturate too early, so making sure we have a nice diversity in our deficits. And as a group, it should cover a range of systems. You need at least 30 items to be included in the frailty index, and they should be very simply coded as presence or absence. So now I want to talk a little bit about the data set that we're going to be using today, and this comes from the Aravel program. The Aravel program, I ran for about 5 years and collected multiomic longitudinal data for over 5,000 people. The multiomic data collection was comprehensive and included whole genome sequencing, clinical labs, metabolites, and proteins, all from blood, gut microbiome, and a bunch of digital self-measurements. I'm not going to go into a ton of detail about the program because there are multiple publications. A nice place to start is the publication here at the bottom. This is actually for the P100 paper, which is kind of a precursor to Aravel, but please feel free to reach out on Discord if you want some more papers. But the important thing about Aravel is that it had a wellness coach that provided personalized interventions based on the multi-omic profile of individuals. This included things like nutritional advice. So maybe a participant had high levels of mercury and they needed to lay off the sushi, or maybe they had a mutation in their cytochrome gene that helped with vitamin D metabolism along with low vitamin D, meaning their supplementation needed to be much greater than standard of care. But the overall point is that participants in Aravel study generally got healthier while they were in the program, and we need to take this into account while doing these analyses. So for the first analyses, because we're going to look at the association with frailty index, we're just going to filter down to baseline. So hopefully everybody's notebook is up and running. I'm going to jump back over the notebooks, and we can actually look at what the frailty index looks like for Aravel. So go ahead and click this first block after the getting session started. And what I'm showing here is just a data table or a data frame. I'll use those terms interchangeably, looking at the frailty measures. So on the left here, you see a public client ID. This is an individual in Aravel. The next column is the self-frailty index. There's some other information in here that we're going to use later, such as sex, age, and race. The next few columns, I'll deal with the actual health deficits that we're measuring. And then finally, over on the far right, there's a merged frailty index. So clicking on this next cell, actually, I'm going to jump back over to my slides real quick, just to point out that for Aravel, we are going to look at 3 different frailty indexes. The first is self-reported. So these comes from questionnaires at baseline. There are a couple different categories, which we'll go to back in a minute. The next frailty index is for labs. These come from clinical labs. And then the final one is a combined or merged frailty index. So for session 1, we're going to focus on this combined frailty index. The other session 2, is going to look closer at labs. So jumping back to the notebook, these are the actual deficits that we're measuring. So the first are about questions about disease, asking participants, do you have an autoimmune disease? Do you have fatigue? The next are all about activity of the participants. Do you have ankle swelling? Do you have pain or discomfort? The next set comes from satisfaction. These are kind of mental health. Are you happy? Are you not healthy? There are some deficits around medications. Are you on cholesterol medication? Are you on blood pressure? Some questions around digestion. So do you have abdominal pain? And then for the lab frailty index, these are just kind of your normal clinical chemistries. So measuring glucose or HbA1c. Also in the lab deficit, we're looking at blood pressure here at the bottom. So clicking on the next block, we're just going to look at a histogram to see when the measures were collected. As you can see, most of the measures were collected around baseline, although there are some that were almost a year out, and we'll work on filtering those here in a minute. The next block, go ahead and click through that, and it's just a comparison of the different frailty indices. So we're looking at a histogram with the index on the x-axis. They're colored, lab being blue, green being self, and the red being merged. The interesting thing I want to point out here is the self index is shifted slightly to the left, meaning when people are assessing themselves, they tend to consider themselves healthier than their labs would actually show. And this has been backed up in the literature. So just for a sanity check to see if our frailty index follows some of the quality measures that I mentioned, go ahead and click this next cell. And what you should see is a couple scatter plots. This is split by male and female with age in the x-axis, the merged frailty index on the y-axis. And as we can see, we have a really nice association between age and our frailty index. So go ahead and click this next cell, and it's all about data cleaning. And I think as a data scientist, this is a lot of what we do. But what we're doing with the cell is loading a bunch of different data frames. We're going to start with proteins. These come from Olink and associated metadata. We're going to load metabolites. These come from Metabolon with the associated metadata and the clinical labs. And then the bottom functions here are just some annotation functions to make it easier to read. But I'm going to jump back over to the slides for just a minute and talk about some of the things that we have to consider when we're doing multi-omic analysis. So data cleaning or pre-processing pipelines are part of any data science project. But when looking at multi-omic analysis, some of the pre-processing steps can be omic dependent. So the first one I've listed here is missingness. So what this is an NA value in your data frame. So I might call the actual values, or the actual features in the data frame, I'll call them features. I might call them analytes, but these are the proteins, the metabolites, whatever we're measuring here. But if we consider clinical labs, a missing value might be concerning, or we can assume that it's actually a technical error. So what I mean by that is if you have a HbA1c measure and you have an NA, then it's likely not 0. There's actually a real value there. But if we compare this to metabolites, we have a bunch of NA values. The reason being, with Metabolon here, it's an untargeted panel where we're measuring a bunch of different things in the blood. So these could be things like medication. So say some of the participants are taking Tylenol and some aren't. Well, the ones that aren't are going to have an NA value. Now, to deal with missingness, it really depends on what method you're using for an analysis. I think the most common methods are probably regression-based, which don't handle missingness well, but other methods such as tree-based methods that we'll look at in session 3 can handle missingness better. But the way we're going to deal with it is through imputation. So this is coming up with a way to actually fill in the missing values. One of the easiest things to do is to fill in the value with a 0. But going back to kind of my comparison of clinical labs and metabolites, if we fill in a clinical lab with 0, say blood glucose, then that's not great and going to lead to a false signal. We know that there's a real value there. There's probably just some kind of technical artifact. So instead, we can use more sophisticated methods. A lot of times with clinical values, you might just use the mean, but it's something that we should really consider, because if you use the mean in metabolomics, you might be giving people a bunch of drugs that only a handful are values for a drug that only a handful of people are taking. So the final thing I want to talk about here is just hard filtering, removing participants or features. We do this pretty commonly using a technique called PCA, which is used to look for outliers. These could be just low data quality samples or participants. But to give a little bit of an idea of what PCA is, since we're starting kind of low level, is that it's a dimensionality reduction technique, and it's been around for a very long time. So dimensionality reduction is just when you transform a set of, or a feature set with a very large amount of variables into a smaller feature set that still contains as much as the information as possible. And this is helpful for a variety of reasons. I've listed some here out on the slide. We're going to use it for exploratory analysis. We're going to look at outliers and we're going to do some visualization, but it can also be used to extract features or reduce noise. So for those of you who are new to data science, you can consider PCA as just looking for the highest axis of variation in your data set. So over here on the right, I have a graph with 2 different features. You consider this something like weight and height. They're obviously correlated, and there's often an axis of variation that kind of goes diagonally across these dots, and that is what we'd call PCA, or principal component 1. orthogonal to that, or the axis of the next most variation, is PC2. And that's all PCA is doing, is finding this axis of highest variation. When we get to a large feature set, obviously this becomes harder to think about, but if you think about it in 2D, then it should be pretty simple. So I'm going to jump back over to the notebook now. Hopefully everything is running okay for everybody. And the first thing we're going to do is look at the metabolomics and Aravail. Clicking this next cell should bring up 3 different data frames. The first is the metadata for the metabolites. Over on the left, you'll see the clinical ID that is given by metabolon. See some pathway information and also some annotation with KEGG and HTMB PubChem IDs. The interesting thing in this table is really probably the biochemical name, because that's the most thing that's easiest to identify. The next table we're looking at is actually our features. So here we have a public client ID again, some data about when the measurement was taken, and then the columns on the right are showing the names of the metabolites. So you'll notice that there are some metabolites that have a biochemical name X dash, and here I'm just looking at the bottom of the metadata data frame, and you'll see there's a bunch of NAs also associated with it. These are unknown or unannotated metabolites. Now, if you're doing an exploratory analysis, you might want to kick these out because it's really hard to actually generate knowledge at the end. But if you're doing something with discovery, you might leave these in. So for our analysis, we're going to go ahead and drop them, which is what this next cell does. And we're going from 1,300 metabolites down to closer to 1,000. So we're dropping roughly 300. So the next thing we're going to do, as I mentioned, is get just baseline. But first, I want to show how many participants had multiple occurrences. So for the most part, the majority, most people, the people in Aravail had 1 time point for metabolites, although there are a handful that had 2, 3, 4, even 5 time points. If you click the next cell, this shows roughly how often these occurred. So people tried to come in every 180 days or 6 months. And that's why you can see a peak at 0 or roughly 200 and then 400. The next cell is just going to do the filtering to bring us down to baseline. So if their day since first draw is less than 75, then we keep them in the analysis. And then we're going to move on to missingness. So as I said, metabolites tend to have higher missingness because we're measuring things like medications. So this is a histogram showing the missingness, and you can see a nice peak between 90 and 100%. If we take a peek to see if I was right, and these are actually medications, you can click the next cell and you see things like tramadol or sertraline, which are both drugs. So again, if we again, I think this proves the point that metabolites are measuring a bunch of stuff, including drugs. So the next cell is going to drop 50%, anything that has greater than 50% missingness, and then do a K nearest neighbor imputation. So this is trying to get us just to a little bit higher quality data set. K nearest neighbor is something that clusters participants by feature and then takes the mean of those clusters. So this is a little bit better than taking the mean of the entire data frame, but and it's fast. But there are more sophisticated imputation methods that can be quite timely and costly. So for this workshop, we're just using K nearest neighbor. So now you can see our data frame is much cleaner and there are no missing values. So now to go back and look at kind of outliers for metabolites, you can click through and start the PCA. So the first plots of PCA are colored by male and female, red and blue. On the left here, we have PC1 versus PC2. On the right, PC3 versus PC4. Now, in general, everything is kind of clustered together nicely. There are some outlier participants, but not very many. These might be interesting to look at for another analysis. But what I really want to show here is that there aren't separate clusters, which would be indicative of something like a batch effect. Now, this data has already been corrected for batch effect, so that's not really a surprise, but I just wanted to say what a batch would look like. The other thing I wanted to point out with this analysis is people often just look at PC1 and PC2, but you can see there's no real separation between male and female. But if we dig in just a little bit farther and look at PC3, PC4, we can actually see a nice separation between male and female. So when you're doing your analyses, I really encourage you to look beyond PCA or PCA1 and PC2. Clicking the next cell, we're actually going to look at our frailty index measure. The reason for doing this is if you see something in this plot, then you're more likely to see something in a downstream analysis. So what I mean by this is there's a nice separation of frailty index with low on top, high on the bottom. So that means that as we continue on with this analysis, we should have some decent results. So this is a good place to start just exploring the data. So the next couple of blocks are doing roughly the same thing or the same filtering with proteins in clinical labs. If you want to go back and do the PCA analysis or different imputation methods, I encourage you to. For my notebooks, I've put little exercises in here. So if you want to come back after the workshop, then you're welcome to. And there's kind of some questions or prompts that you can go through. If you click over on the left, there's a table of contents that actually shows everything all that we're going through. And you can see the exercises have a star by them. So go ahead and click through. We're going to look at the clinical labs next. You can see there are quite a few records for all these. If you want to look and see what labs we're measuring, they're listed out here. There's a bunch of stuff that you should be familiar with, such as LDL, and there's some more rare measurements, such as lead or mercury. Next, we're going to click this final cell here. What this is going to do is just save these baseline measurements so that we can load them later and we don't have to completely go back over this notebook. So I hope everything is working okay for everybody. Actually, now since we're through kind of the intro to what the data look like, we're going to start our first analysis, and that is WGCNA. This method came out of Steve Horvath's lab nearly 2 decades ago, but it remains very popular because it's well validated and relatively easy to understand. And why are we looking at this method? Well, as I mentioned earlier, biological networks are complex and hierarchical, so it's kind of a natural fit to use a network analysis approach. WGCNA is an unsupervised analysis approach that was originally developed for gene expression data, hence the G in WGCNA, that stands for gene, but has since been applied to other omics and even imaging data. So that's why the G was dropped out when we're just looking at weighted correlation network analysis. We're going to start by going over WGCNA in quite a bit of detail, and the reason for that is there's some choices that we're going to have to make when doing multi-omic analysis that we need to understand. So the WGC framework is outlined on the slide. If you're familiar with using this method, you're probably familiar with this slide, but it starts by constructing a network and breaking that network into modules. So modules is a bit of an overused word. But for this case, it just means a cluster in a network. After the clusters are generated, modules can be summarized and related to external info. So in our case, it's going to be the frailty index, and we can learn about unknown molecules in the pathway to understand their function. This is kind of a guilt by association analysis. We're not going to spend too much time going over preservation across different data sets, although I will mention it briefly for the multiomic analysis. But the last step here is looking at key drivers in the modules. These are often called network hubs and can be targets for therapeutics or biomarkers. So one thing I do want to mention is this is an R package, and it was developed in 2008. This is probably the main reason this session is using R. There is a Python wrapper for this package if you're more comfortable using Python, and I encourage you to go out and then use it. So why not just use correlation? Why is there an entire framework around creating a network? And we're going to start just by explaining what a co-expression network is. And this is just a correlation network. So you consider a bunch of samples and a bunch of features, in our case, proteins. And if they go up in one sample, down in another sample, up in one sample, down in another sample, then they're considered correlated, and you can look at measures of correlation that you're familiar with, such as Pearson or Spearman. Now, there are two reasons that we're gonna that there's a framework around network analysis. The first reason is to create a scale-free network, and the second reason is for topological overlap. And I'll go over these kind of individually. But a scale-free network is a network with few hubs compared to a random network, which would be a bunch of connections between all of the nodes. So there's an example of the scale-free network here on the bottom right of the slide. You can see there are hubs in orange that have a bunch of connections, and the blue nodes have very few connections. So it's useful to model biological or our networks as scale-free because many biological networks are scale-free as well. So we're kind of fitting our model to biology. Topological overlap kind of looks at indirect associations, and I'll cover that just a little bit more in a minute. But first, I want to talk about how to build a scale-free model or scale-free network. There are two ways to do this. The first is to look at an unsigned network, and the equations are here on the slide. I'm sorry if you don't like math, but it's pretty simple. It's just looking at the correlation about the absolute value of the correlation and then raising it to a power function or a beta. So with an unsigned network, since we're looking at the absolute value, we're ignoring the sign, clustering positive and negative correlations together. This is opposed to this is a sign network that preserves the sign, and it's really looking more at positive correlations. And biologically, positive correlations tend to be more informative. So to generate the actual scale-free part of it, you use this beta or this power function. So as you increase, or if you take a correlation coefficient and increase it, the higher correlation coefficients are going to stay higher. The lower correlation coefficients are going to move quickly to 0 or 1. So the histogram over here on the right shows a scale-free network. You can see connectivity on the bottom and frequency of connectivity on the y-axis. The scale-free network has a bunch, or a small number of hubs. So there's a couple with a couple nodes with 60 connections, but most of them have 1 or 2 connections. To get the beta to reach an approximate scale-free, what we're going to look at is the correlation of connectivity, so the x-axis in this chart, compared to the log transformation of connectivity, or frequency, so the y-axis in this chart. And if you plot those two together, what you see for this network, which is approximately scale-free, is an R-squared, or just a Pearson correlation value of 0.92. So this is what's called our scale-free index. If this was not scale-free, this would look more like a curve and have a much lower scale-free index. So WGCNA authors say that approximate scale-free network, you want your index close to 0.9. So the last point I want to make on this slide is just why do we use this approach? And it's a soft thresholding approach that preserves a bunch of information in the network. We're not doing any hard cutting or hard thresholding that would remove a bunch of information. So finally, to topological overlap, and again, there's an equation on the slide, but I tried to explain it as simply as possible. What topological overlap is meant to do is to deal with spurious or missing correlations. So I think an easy way to understand this is looking at social networks. If you have a group of friends and you're going out with this group of friends frequently, then you all probably know each other and are well connected. But if you go to the coffee shop and meet somebody, you might hit it off nicely with them and have a good connection, but it's kind of a one-off thing. And unless that person is integrated into your group of friends, then the connection is probably low or minimal. So that's exactly what topological overlap is trying to do. It's taking each of the nodes and looking one node away and then counting up the shared connections with another node. And then it's dividing by the total number of nodes that are also one step away. So I hope that makes some sense and helps understand topological overlap. So the next step after we have our topological overlap matrix is to actually identify clusters. This is just done by simple hierarchical clustering, or taking a dissimilarity matrix and then calculating the pairwise distance between all nodes in the matrix. Those nodes are combined, and then the calculation is done over again until all nodes are combined, and we see kind of a tree-like structure over here on the right. This is called a dendrogram. It kind of looks like a tree with branches. You can consider the leaves to be the features or protein in our analysis, and the branches refer to the clusters or modules, and they're usually color-coded just so you know what they are pretty easily. With a dendrogram, it's possible, or hierarchical clustering, to just do a strict cut height. So say this is pretty close to something like 0.98, and each of the branches falls out into its own cluster. But with WGCNA, there is something called dynamic tree cut. We don't have too much time to go into it today, but it's more flexible than a strict cut height, and it allows for automation. So finally, now since we have our modules, we need a way to summarize them, and that's this term called an eigengene. Maybe you've seen it before. It's kind of a scary term that's it deals with linear algebra, but I want to make it as simple as possible for this workshop. And the best way to think about it is to look at a module and look at expression of a feature in that module. So this graph here, we have a bunch of samples. We can have, say, our protein, and it goes up and down across the samples. Everything's nicely correlated. Now the module eigengene is just the representation of the expression profile in that module. So for this case, it would be this dark black line. If you think back to PCA that we talked about earlier, all this is for WGCNA is the first principal component of the module, or of the features in the module. So why do we break it down into something so simple? And it's because we can then relate modules to each other. So we can just do correlation analysis of all the module eigengenes and see how similar they are, maybe merge them if they're super similar. We can also correlate this with our phenotypes of interest, so in our case, frailty index, and we can define module membership. So we can correlate each of the individual features in the module with the eigengene to see if they are hub genes or not. Cool, so that was a lot. And now we're gonna go back to the notebook and walk through how to do this in with a single omic. We're going to start with proteins. The reason for starting with proteins is it's a little bit smaller of a feature set, and it can run in the time it takes to do the workshop. Although I encourage people to come back and look at metabolites in clinical labs. So go ahead and run this first cell. There's not much that's going on here. It is just preparing a bunch of data tables. You can see the tables here. The first one is the feature table looking at the proteins. There's a bunch of NA values that we need to deal with. The second is our phenotype table with our frailty indices, and the third is a metadata table that has information on the individual proteins. So as I mentioned above, there's a bunch of missingness. WGCNA has its own filter to deal with missingness. It basically removes anything greater than 50% and then does K nearest neighbors. So go ahead and run this next one, and it's going to spit out quite a bit of text. So after filtering, you can see that we only have 274 proteins in this sample. If you scroll back up to the top, you can see we started with 1,335. So the reason for this is that the majority of Aravel participants had 3 O-link panels, and a small subset was run with a larger number of panels. You can see the panels annotated here with the alphanumeric. So this is the oncology 3 panel, and these are all being removed just down to the smaller subset. So to start, we're going to pick a soft threshold for WGCNA. This is going to take maybe 20 to 30 seconds to run, but a couple things I want to point out. Our network is going to be using the Spearman correlation. It's often more recommended to use a Pearson, but just for sake of time and because it fits protein data a little better, this is what we're using. And we're building a signed network. Now, to assign the power, we're just looking at a beta from 1 to 15 and going through and seeing which of those fit the best scale-free index. So this should finish here pretty shortly. There we go. And we have plots similar to what I showed on the slide. So here we see our R-squared fit and the soft threshold power, or the power on the x-axis. There's a red line at 0.9, and usually you pick something close to the red line or the thing that is right above the red line. So the next cell, if you click it, it's going to pick our soft power for us, and it's going to pick a 10, which is the next power right above the red line. The next cell is going to perform the topological overlap. Maybe it will. Give it a minute, see if it goes. And WGCNA provides a nice functions for all these, but with, as I kind of briefly mentioned with hierarchical clustering, it's better to do it on a distance matrix or a dissimilarity matrix. So we're taking 1 minus the tom to get to dissimilarity. Next, we're going to do clustering. So we're going to create our dendrograms. This is the bit dendrogram of all the proteins. Again, the leaves or the end are the proteins, and how close they are together is how similar they are. The next cell is going to do the dynamic tree cutting, and it generates 4 different modules for us. These are numbered, so the next cell is going to change them into colors. So with WGCNA, it will always give turquoise to the largest module and gray to the module of unassigned features. So printed out a heat map here just to show, if you're more comfortable looking at heat maps, that the clusters in the heat map are those correlate or associated with the clusters in the dendrogram or the colors. So the next couple of cells, they're not going to do much for our analysis here, but I've left them in there if you want to go back and play with some of the different parameters. So this is just merging based on my eigengene distance. I put a cut height in here that we're using, and you can see there's nothing below the cut height, so nothing is going to get merged. You go ahead and click through the next couple cells. This is showing that nothing is merged, but again, if you want to go back in and change the parameters, feel free to. And then finally, we're just going to output some data tables. So now, since we finally made it through that, we're going to look at our phenotypes of interest. The first thing we have to do is actually do some hot encoding. And this is changing categorical variables into numeric. So for our analysis, we have female and male for sex. There's nearly twice as many females, which is just what happened with the Arabelle study. But for race, we have a little bit of a problem. There's quite a few categories, and Arabelle was not horribly diverse. So there was 1,000 people that's identified as white, and the rest identified as a different race. So for this analysis, and it's not ideal, we are just going to encode each of these sex as 0 and 1, and race as white 0, and the rest of the races are 1. Again, not ideal for analysis. I don't recommend doing it for something you're going to publish. And I also recommend coming back in and doing different encodings for race. But just for simplicity of the workshop, this is how we're going to handle it. So go ahead and run this next cell. And you can see our male-female is hot encoded to 0 and 1, and our race is coded binary as 0 and 1. So now that we have our hot encodings, we're going to go through and actually do the correlation with our trait. There's a lot of code here, but all we're really doing is in this line where we're looking at the correlation of the module eigengenes and all our phenotypes. We're then doing p-value, or generating our p-value based on those correlations, and then we are doing a Bonferroni-Hochberg post-hoc correction. So after all of that, we finally have some results. We have our 3 modules. We've cut out gray because it's uninteresting features. And we can see the associations, or the R-squared on top, the p-values on the bottom. There are 3 modules, the turquoise, blue, and brown. And kind of looking across this heat map, there's one that sticks out pretty nicely to me, and that is in this brown module. We have an R squared with our merged frailty index of 0.5, which is actually a pretty phenomenal correlation in biology, and the p-value is very, very significant. It is also moderately correlated with age, but not nearly to the same degree. So here's a little exercise to remind you to go back and change the encoding for race if you want. But one of the problems with just this simple correlation analysis is we aren't adjusting for any covariates. So the next couple blocks here, we're just going to do a basic regression analysis and account for those covariates. You can click the next block. It's going to generate a data table that's combined with the eigen genes and our outcomes of interest. And in this next block, we're going to do some regression. So here we have our covariates listed out: age, binary sex, binary race. We're only going to look at the brown module just for time. And the variable outcome we're interested in is the merged frailty index. So go ahead and click this block. It's going to output something, or a summary of the regression, which maybe you're familiar with looking at. But what I want to point out is the graph here at the bottom. So this is the adjusted module eigengene values, adjusted after our covariates, and our merged frailty index score. So as you can see, after adjusting for age, sex, and race, we still have an incredibly nice association for our brown module. So we're going to kind of continue looking at it. The next thing to do is to look at intramodular connectivity. I mentioned this briefly in the slides. There's a bunch of code here, but basically all we're doing is taking the row sum of each of the modules' connections, and the ones with the highest intramodular connectivity are considering the hub genes. There are other ways to do this, looking at module membership, but we will just be looking at intramodular connectivity today. So what we have again is our modules, intermodular connectivity, and topological overlap sum. But now we have some data that we can actually dig through and look at the biology. So what we're going to do first is an enrichment analysis over the brown module. So this first block of code is just pulling out each of the modules, and then the next block of code is performing the enrichment analysis. So if you're not familiar with enrichment analysis, all we're doing here is overrepresentation analysis that's done using a hypergeometric test. This is like picking a bunch of proteins at random and having them be part of the same annotated pathway. How likely this is, or the p-value, depends on how large the pathways you're looking at are and how many proteins are in the experiment. For this analysis, we're only looking at one set of pathways, which comes from the gene ontology biological processes. You can change the pathway type here and look at different ontologies, or look at different sub-ontologies in Go, such as the cellular component or molecular function. So finally here, we get to some analyses, some actual results. If you click here, you should get some bar plots. And as you can see, there are nominal p-values that are significant for this module, but nothing after post hoc correction. But the overall general signal we see here is pretty strong, and that deals with immunity or lymphocyte differentiation. So one other thing to do is to look at just enrichment in the hub genes. So these modules can be quite large, and some of the proteins in them are not well associated with the module. So looking at the drivers is another way to get biological significance. So I'm going to go through and click these next couple cells. All we're doing here is splitting it by intramodular connectivity, looking at the top 10%. You can see on the histogram here that this gets most of the hub genes, although we could lower this threshold just a bit. The same enrichment analysis, and then finally looking at the bar plot and the p-values. So now we have significantly adjusted or significance after post hoc adjustment. And the top tier thing is response to interleukin-1. And this wasn't planned, but Dr. Furman talked about interleukin-1 in his talk. And so I think this is a pretty cool result that we could dig in a little bit further. So interleukin-1 is part of pro-inflammatory cytokines that regulate innate inflammatory response. So I put another exercise here at the bottom. Let's see how I'm doing on time. Okay, I got some time. To look at metabolites or clinical labs if you want. But what I want to do next is actually compare the systems analysis that we've broken down into clusters and then looked at the hubs of those clusters to come up with interesting results to just what would happen with differential expression or quantification analysis. So if you click this first slide, all we're doing here is splitting our frailty index into quintiles, or looking, or basically by 20%. And we're going to compare the first quintile, the first 20%, to the last quintile, or 80 to 100%. This is just to be able to do differential expression and get some decent results. The next thing we're running is a program called Lima. You might be familiar with it if you run on any RNA-seq experiments. And then finally, plotting the results. Lima is basically linear regression under the hood. So we see a nice volcano plot here with a bunch of different significant results. The bottom slide here is actually going to list out the number of differentially expressed peptides. So we see we have 213 differential expressed peptides when comparing the first quantile to the fifth quantile. So if you remember, there were only 270 peptides total. So nearly all of our peptides are differentially expressed when we look at it in this way. These peptides are split across a bunch of the modules. So 74 of them would fall in the blue module, 68 in the brown, and 71 in the turquoise. And if we run differential expression, which is the next couple blocks here, what we'll see is, so what we're doing is looking at anything that has a p-adjust below 0.05, running differential expression. Again, as I mentioned, this is almost everything in the panel, and it's kind of unsurprising that we don't get good results just because there are so many things. So I hope this kind of shows that systems approach is a little bit better way to do it than just going off a hard adjusted p-value with your differential expression experiment. Okay, so we're going to jump back to the slides now and talk about multi-omic integration. So this is kind of the crux of what we're doing here. And we went over everything at the beginning to kind of bring people up to speed and went deep into WGCNA so we can talk about some of the choices we have to make when doing multi-omic integration. So I've split the integration techniques into kind of two extremes here. These are early integration and late integration, and these are things you have to think about when you're doing combined analysis. So with early integration, you are just concatenating each of the omics data types together and then doing the downstream analysis. So what's nice about this is you're going to preserve correlation between omics. So this way you're still going to have the correlation of proteins to metabolites or metabolites to clinical values. But there's a con here, and that's each of these omics has a different underlying data distribution. And when doing the modeling, you're kind of combining everything together, and you might have spurious or incorrect signal because you're not accounting for these different distributions. This is opposed to late integration, and this is a little bit more straightforward with the modeling because you model each omic individually, get your results, and then do some analysis at the end to combine all the results. So the problem with this is we're not directly capturing the inter-omic relationships. And if you're doing an analysis where you're interested in those, this might not be the ideal way to go. Now, these are two extremes. Obviously, there's a bunch of methods that have kind of middle integration, if you will. But for WGCNA, there are a bunch of publications out there on late integration. And this is mostly done by looking at the correlation of the eigengenes for each omic and then comparing them there at the end. So you build your eigengenes for proteins, metabolites, clinical values, and then do a correlation at the end. If you have highly correlated modules, then you can kind of consider this as a multi-omics signature. However, what we're going to try to do or attempt to do today is an early integration method. So, as I mentioned before, you have to account for the difference in distributions when early integration. What does this look like for us? Well, on the left, you can see the distribution of protein-protein correlations. This distribution is kind of shifted slightly to the right, meaning there are more positive correlations than negative correlations. And we see roughly the same thing when we're looking at transcriptomic data, by the way. But for picking our beta for our adjacency network, it's pretty easy with proteins, and we showed that in the analysis. You can get a nice scale-free network with an R squared or a beta of 10. But looking at the clinical values, this distribution is much more centered at 0, and picking a scale-free or picking a beta is much more difficult. The graph on the right shows that 15 might be the most reasonable number, although that's getting to be pretty high and unreasonable to use. So if we just concatenated all of our correlation values together and then tried to use WGCNA, we would get a beta that kind of poorly fits all of the omics. One other consideration with early integration is that if there are differences in number of features, so if there are, say, 10 times as many metabolites as there are proteins or clinical labs, then the metabolites or the metabolic signature is probably going to dominate your analysis, and it's probably what's going to be used to pick the beta. So our strategy to deal with this for early integration is to start with the correlations separately by category by omic, and to make a smooth model of the distribution of correlations of each type. We're then going to transform the correlation values into a single smooth or shared smooth model. While the resulting values are no longer interpretable as correlations, this will standardize the significance of correlations from different sources onto a single shared significance scale. When WGCNA fits the values on the scale, it will be applying the same significance standards to all the correlations regardless of their source. Since the correlations are bound by -1 to 1, a Gaussian distribution is not really appropriate for this, and instead we're going to rely on a beta distribution for both fitting models to our observed correlations and the shared target distribution. We're going to look at agreement of the bulk distribution for our model, paying particular attention to the peak of the module, and try to capture as much of the variance in the distribution as possible. The centering process on the standard shared distribution, we're going to use kind of a z-score approach as opposed to a ranked based approach that you might consider for like a quantile normalization method. So what this is going to do is it's going to be the value under the source distribution, the target distribution is going to have the same significance or p-value. So let's jump in back to our notebooks to take a look at this. The first thing I'm going to do is stop my notebook 1 kernel. The reason for this is if we have a bunch of different notebooks running with a bunch of different data, the instances do have a limited amount of memory, and sometimes it can cause it to crash. So go ahead and start notebook 2. Make sure my appearance looks good, and run the setup. Again, this is just loading a bunch of different packages from R. Good, and now since there's a 1 here, we know that it ran successfully. I'm going to go through and load all the data that I created earlier during session 1. So all of the baseline data, right, and the frailty measures. If you're following along, this next cell here is just going to merge everything together and spit out some information about the number of participants that had in the merged data set. So that's what all these numbers are here. And then finally, we're going to jump into WGCNA directly as we did with the single omic analysis, starting with our data cleaning. So as you can imagine, we're going to be dropping a bunch of different features, a bunch of different clinical labs here in the beginning, metabolites next, and then all of our Olink panels that were only run on a subset of patients. So what we're left with is 868 participants with about 1,200 features. The next block, we're just going to look at, we're going to pull out all of the names of the features we're interested in and spit out more information about how many features come from each category. So we have our 276 proteins again, 872 metabolites, and about 50 clinical labs. The next block of code is split into two. The top is loading something from the useful files. So this is something I've pre-generated. And the bottom here is what was run, or the code that was run to create this pre-generated file. It's just running pairwise correlation between all the different omics. So this is both intra-omic, so protein-protein, and inter-omic, so looking at protein metabolite. We'll have 6 data frames here in total. The reason I commented this out is the correlation can take quite a bit of time, I think 10, 15 minutes. So I just preloaded it in order to save us some time. So finally here, we're going to start looking at our protein-protein correlation. And as I mentioned, we're going to model with a beta distribution. In order to model with a beta distribution, we're going to use 2 variables that are shape variables. These are V and W. In order to see what the shape variables are, here's a formula. This is how it is associated with the mean, which is MZ, or the variance, which is S2Z. So all we're doing here is looking at our proteins and calculating what the distribution would be based on the mean and variance. The next cell here shows in blue what that model would look like. And under it is the actual distribution of the protein-protein correlations. Now, what we see here is that the model based on the mean and variance doesn't do a great job fitting our distribution. The reason, as I kind of mentioned earlier, is the protein-protein correlations are skewed slightly to the right. So there are kind of some outlier positive correlations. And this distribution, the mean is slightly shifted. So what we want to do is make sure that our distribution captures the peak and kind of preserves these positive correlations. So this next block here, go ahead and run it, and you can see that the model that is run for this block fits the distribution much better. Now these are all pre-generated, but all we did is created our 2 shape variables, so V and W, and then played around with them and kind of fit the distribution by eye. So you can change these and say, I wanted to look at what if the ratio of these 2 shape variables was the same. You can see that the distribution then shifts to the center to 0, and slightly lowers. So I'm going to go ahead and change this back to where it fits the distribution pretty nicely. Again, I want to point out that the purpose of this is to fit the peak, to fit the bulk of the distribution, and to preserve a bunch of these positive correlations. So the next few steps are just repeating that same thing over again. We have some values in here where we've already kind of gone through and fit them by eye, but I encourage you to go through and play with the 2 shape variables, change the distribution, and see if you can get a little bit better of a fit. So this is looking at metabolite. You can see again, we're doing the same thing where we capture the peak of the distribution and try to preserve these positive correlation outliers. Clinical labs is next. Go ahead and click through it. It's much more zero-centered distribution. And then finally looking at the interomic correlations, running the same analysis again. So protein metabolite, protein clinical test, and metabolite clinical test. So finally, after we've done all this, we're going to create a standard beta distribution that's centered at 0, and then use kind of that z-score property to merge all of our other distributions into that standard distribution. So that's what this next cell is doing, a centering. And then finally, we're going to construct a complete centered correlation matrix. So what we're doing is, after we have our centered distribution, we're going to create a correlation matrix that we can put into WGCNA. The structure of it is here. So protein-protein correlations up in the top left, clinical correlations in the bottom right. We do have to transpose the interomic correlations just so that our correlation matrix is mirrored across itself. So finally here, just checking the actual, checking our distribution. So the orange is our fit distribution, and the blue line would be the distribution of the fit distribution if we were based on the mean and variance of this centered distribution. So again, I want to point out that our distribution fits the peak. We're preserving these positive correlation values, whereas if we just looked at a beta distribution based on the mean and variance, it would be slightly shifted and kind of ignore some of these other correlations. So now we can just go back to WGCNA. We have a standardized distribution and run it similar to we did in the single omic analysis. So the first step here, I've kind of clumped a bunch of the code together, so it's a little bit easier to run. But again, we're running a signed network. And as you can see, we do reach approximately scale-free with R squared of 9, or our power of 9. There's something a little odd with this scale-free, as it kind of flatlines after that, but that's okay. Since we're kind of merging omics, it's not surprising that there's something odd in our data. The next block is going to create our dendrogram. We can see a nice dendrogram with a bunch of different branches. So we know we're going to get nice clusters. Go ahead and click through this, assigning colors to the clusters. We can see that there are 15 modules or clusters in this network. And the 0 here is the gray. So there are 20 features that just kind of don't fit in with anything else. Next, we're going to calculate our eigengenes and look and see if we should merge anything based on their similarity. I've created a cut here where we're not merging anything. Again, go back and feel free to play with the cut height and merge modules that you think should be merged. Finally here, we're going to clean all our data frames so we have a nice output for our phenotype analysis. This next cell is creating looking at the correlations between the eigengenes and our phenotypes of interest. So the same thing we did in session 1, looking at the significance and correlation. And we get this nice heat map again. This time we have a bunch more modules, so we have more interesting things to look at. So kind of going from left to right, you can see that there are a couple modules that are highly correlated with our binary sex variable. So this purple and red module. Interestingly, the purple module is also negatively correlated with age. So this might be interesting to look at later on. But for our purposes, we're looking at the frailty index. So I've listed them out here, the self-lab and merge. We've been looking at merge kind of this whole time. And there are 2 modules with nice p-values and nice R-squared or correlation values. So the turquoise module has the highest correlation and lowest p-value. And the green module up here has a moderate correlation and I mean, it's half the p-value, but still interesting. The green module, you'll notice, is also correlated with age, so it might not be as interesting. But the turquoise module is not as well correlated with age. and highly correlated with frailty index. So the turquoise module is probably the most interesting for us to look at for this analysis. So that's what we're going to do. We're going to continue on just creating some nice data frames here. This is more cleaning code. And then generating our intermodular connectivity, or again, this is looking at the row sum of connectivity per module. Finally, as I mentioned, we're going to stick to the turquoise module and kind of dig into it just a little bit. So what I'm doing here is I'm looking at the top 20% hub genes by intermodular connectivity for the turquoise model or module. So the first thing you'll notice is a chart here where the analyte ID is over on the left, the module, which is always going to be turquoise, the data set of where it comes from next, and then the chemical name over here, which is easier to understand for metabolites. The top modules you'll see are dominated by these glycerols. These are diacylglycerols, or short-chain fatty acids that are commonly precursors to triglycerides. And I think this is pretty interesting because it could lead to a microbiome analysis that's also associated with fatty acid metabolism. The first protein we see in our analysis is LDLR, or LDL receptor, which is a protein that controls blood cholesterol by clearing LDL particles. And scrolling down just a little bit farther, the first chemistry we see is LPIR score, or a weighted combination of 6 lipoproteins that are used to measure insulin resistance. So we have a pretty consistent signal here around lipid dysregulation for our frailty. The other thing I want to point out that's kind of, I would say the biggest roadblock in multiomic analysis deals with annotation. So we have a bunch of metabolites. They're all pretty similar, short-chain fatty acids. But looking over here on the right, we see KEG. So this is another pathway database, and HMDB. And this is another annotation. But there are a bunch of NAs, so a bunch of unannotated short-chain fatty acids. So if we go through and try to do enrichment as we would normally, since most of these metabolites are unannotated, we wouldn't get a very good answer. So that's one reason why I'm just kind of looking at the top of this table manually. But that's not gonna stop us from trying. And the next block here is looking at multi-omic enrichment. So again, this is just overrepresentation analysis. I've preloaded some pathways. So we're looking at KEG and reactome. And then there's a bunch of code here, but really it's just collapsing these pathways and making sure that they actually have an annotated metabolite or protein from our sample. So what the pathways look like is for the proteome, we see the glycolysis pathway. These are uniprot IDs over here. For metabolome, we have the glycosylis pathway again, and it's annotated by HMDB IDs. But remember that a bunch of the short chain fatty acids up here have NA, so we're going to be missing out some information. Looking at the turquoise module, this is just running enrichment again. I've kind of combined everything into one giant cell block, so it's easier to run. But if you want to change some things, you can change the module here. So turquoise, you can look at the purple or green, and you can change the number of hubs you want to focus on for the analysis. Down here, the results are shown, and you can see that as far as a p-adjusted value, we do reach some things of significance, but there's only one protein that is actually in this or labeled in this analysis, and that is LDLR. So really, we've boiled down to all this. This is just showing that enrichment for these modules can be difficult because there were not enough annotated metabolites, but we still see that lipid signaling as significantly enriched. I also mentioned that the green module was interesting, and I just want to run it really quickly because It was associated with the frailty index, a little bit associated with age. But even though there are two things annotated for this enrichment analysis, they deal with the TNF signaling pathway, which Dr. Furman mentioned in his analysis. So I thought that was also a nice tee-up for me. So that brings us to the end of session 1. We have about 10 minutes to go over questions, and session 2 will start shortly after that. And thank you.
We're going to talk about two things, one of which is longitudinal trajectory analysis. But before that, we're going to talk about a method called Diablo. So first, I want to make sure we're all on the same page. You should have had instructions to open a browser and go to this website to log in. You should have received an email about how to do this, and you need to click on this. And there's a whole, in the email, there should be a whole set of these instructions. So I'm going to move on. I believe that all of you should be there. I just want to quickly review what we talked about in session 1. This is the data set, the Airville data set that we'll be using, we'll be analyzing. Airville was a company that did wellness coaching, and it collected longitudinal data about all of its participants. They were of a variety of types, and we're showing how to, the main purpose of this is to show how to analyze that longitudinal data. But before that, we're going to talk about one more analysis related to what we did in the first session. So the outcome we're looking at as our measure of health is frailty index, which is a fraction of health, defined health defects. The research has shown that you can define this in a number of ways and get similar results about correlations with health and such. We actually, in this, for this webinar, we did 2 different ways and then combined them. The first way was a questionnaire at the beginning. I'm sorry, Airville collected a questionnaire at the beginning from each participant, and then when they collected longitudinal blood samples, they were able to use clinical laboratory tests to define a second version of frailty index. And then the combined, which is what was analyzed in the first session, was the sum total of that, but only, I believe, only using the laboratory frailty index from the baseline time point. Oh, and I wanted to mention that just to show that this actually does represent health. One thing you would expect with health is that from age 30 to age 90, you would expect your health to get worse. So in both males and females, these plots show that that is indeed true for the combined one. Number of health defects increases with age. So last time in last session, we looked at PCA, and we went through a process of combining correlations that were on different scales so that we could identify clusters that transcended the different types of measurements that we collected. And then we represented those clusters with eigenvalues, which are essentially characteristic patterns that represent an individual's values on each of the types of measurements for the data within each cluster, for the measurements within each cluster. And on the right, we see a plot of some imaginary data that has been colored with 2 levels of an outcome variable. So the blue might be people with poor health, and the red might be people with good health, or vice versa. And the point here is that the shape of this data, the pattern of the data, is long and thin along the anti-diagonal axis there. And that is what PCA looks for. It looks for the longest stretch of the data in any of the dimensions in which, well, in the combination of dimensions in which the data was measured. And it doesn't take into account the outcome. Diablo is a very similar method to PCA. However, it is a type of discrimination analysis. And discrimination analysis is specifically intended to pick the direction that is most correlated with the outcome that you're interested in, rather than, as in PCA, with the stretch of the data in the data space. So that is why PCA will pick the major dimension in this data, PC1, going along that long axis, whereas PLSDA, which is a type of discrimination analysis very similar to PCA, that's implemented in the Diablo package, Mixomics, will pick a very different direction, one that is more correlated with the outcome. Okay, so we're going to be talking about Diablo first, and that is what I have called session 2.1. We're going to look for trends in the outcome and how they are related to the data. We're also going to make a multiomic model, and we're going to evaluate this with cross-validation so that we're always testing in an outgroup rather than from the data from which the model was made. That's another difference from what was done with PCA. PCA typically is used to look at all of the data at once rather than holding some out and seeing it as a prediction issue. All right, so what are we going to cover in Diablo? And we'll be going to the notebook in a minute. I just want to give you a brief outline. First, we're going to have to talk about a little bit of data preparation, which was another large topic earlier in the seminar. Then we're going to look at the PCA-like evaluation of discrimination analysis for each of the omics measurement types separately. So proteomics, metabolomics, and lab tests separately. And then we're going to look at combining those with an advanced version of SPLSDA called block SPLSDA, where the blocks refer to the 3 different omics. And we're also going to be there talking about sparsity, which is an effort to try to help make the model that this method creates more understandable or explainable. And we're going to talk about some issues with the model fitting process. Right, so last time, as I said, we looked at the combined frailty index. This time, we're actually going to change that. In session 2.1, we're going to look at only the self-reported items, the items from the questionnaire. And that is because we're going to be using the laboratory data as one of the 3 omics that we combine in Diablo. So we didn't want to use the data that we're trying to predict. So we excluded the lab stuff, and that left the combined out and left us only the self-reported to look at. If you look at the bottom, we look at the Spearman's rank correlation between these 3 kinds of frailty index. And you can see while the lab and combined are very fairly closely correlated, the self and the lab are completely independent and are to some extent different measures of health. However, they are still both clearly, they're correlated to each other, and they're both clearly correlated to health, which you can see based on the set of questions that were used. And prior research on frailty index has shown that even questionnaire-based frailty index is relevant to health. So let's switch over to the notebook. This should be notebook 1, Diablo, under session 2, over in here. So Aging Workshop 24, session 2, notebook 1. I have 2 forms of it. One is the live notebook, and the other HTML version that has all the cells run and output for you to look at. So if you get stuck, or if you'd just rather follow along and try the notebook out later, you're welcome to follow along in the HTML version. But if you want to do each of these steps yourself, you'll have to do this in the live notebook. Now, there are a couple of these steps that take a good 10 to 20 minutes. So if you race ahead, you may get stuck on one of those cells. I'm sorry about that, but the method takes what time it takes. So my suggestion is to go through it with me. I personally am going to be switching over to the HTML notebook when I run into one of those long ones, and from that point on, it will be from the pre-canned data. All right, so sorry about that, but that's the best I can do, I think. Okay, so let's get started. First, we're going to load up the code packages, ARC packages we need. And I meant to mention, although this sets a random number generator seed at the very beginning, and I believe it's set a couple other times afterwards, we were not able to get this notebook to produce the same results each time. So if your code runs, but you don't get the same results I do, I'm not surprised. And again, we have the HTML version so that you can look at the same results for at least one run of this notebook as I got. So they weren't the best results I've seen either. So hopefully you'll get better results. Yeah, so we're just reading in data. This data was generated during session 1, but I'm reading it from the alternative storage path, which means you don't have to have run session 1's notebooks in this run in order to be able to read this data. Then we're going to filter out anything with more than 15% missingness of any of the data measures. And this is going to filter out a large number of proteins, especially because of the way we sampled those. That was described in session 1. If you want more information about that, please ask the TAs on Discord. We're now combining that data together. So there's our combined data set. We have 891 participants and 1,182 analytes at this point. This is just setting the data up into blocks at the end, we're going to be using the block form of Diablo. And here's where I show you the correlations between them. So for this purpose, we also needed to cut the frailty index into categories, because Diablo is intended for discriminating among categories. So here I show the self-quantile, which is what we're going to be using as the categorization that is our target, versus the laboratory quantile, 5 quantiles of the laboratory. And you see the correlation coefficient is low, whereas for the ones that actually, for the merge, it is correlated fairly highly with both the self and the lab. Here's the breakdown by sex. We have 572 females and 319 males. And here's the age distribution. So this is going to play a role a couple times when we talk about frailty, because as we know, frailty is associated with age. As you age, you get more frail. And we have a lot of data for fairly young to middle-aged people, but as we get closer to 90 years old, the data drops off. And there are a couple different reasons for that. And there are a couple of different effects of that. I should say that at this point, I am not an epidemiologist, and epidemiologists understand how to deal with the effect of, for example, people dying and therefore no longer being able to be in your study. So we are, as data analysts, we are stepping into the medical realm when we look at human patients, and epidemiologists are specifically trained to deal with issues like that. So when you have a question about something like this and you're doing data analysis, please consult a bona fide epidemiologist. There's a lot going on in the health arena that I personally was not aware of when I started doing this, and it is something that an epidemiologist will know all about. And then I'm just checking to make sure that all the clients are listed in the same order and with the same IDs across all the different types of data. All right, so let's look into the single omics PCA analysis. This is what we had as the output from session 1, and I just want to recapitulate that here so that we can compare with it. I believe that in session 1 it was also colored. Actually, I'm not sure whether it was colored by quintile of, well, it certainly wasn't colored by quintile of self-reported frailty index. But this is, and as you can see, it's a little bit difficult to separate by eye the different categories of, so the individuals from the different categories of self-reported frailty. On the first two principal components, these are the largest, or the strongest patterns in the data. PC1 is the strongest, PC2 is the second strongest. So what that is telling us is that just looking at the data and ignoring the outcome of interest, we're not really able to strongly separate the data here for metabolomics, at least on metabolomics. And we will find that the same is true. By the way, here's a better view of how separate the different quintiles are on the first axis and on the second axis. So with proteins, we actually see a fairly similar lack of ability to distinguish them. And here, again, there is some information, but it is not really possible to separate quintile 1 from quintile 5 even. It really shouldn't be very much of a surprise that we're not able to separate the internal quintiles by any of these, because the individuals in the Airville cohort were not selected as a case control study of any kind. These people are not known to have any particular disease. So this is just, I would hesitate to call them people off the street because of how they were selected, but they were not selected for health status, really, except perhaps to be relatively healthy. Looking at the internal ones is really asking for a lot of ability to discriminate. What we should really be looking at is looking at whether we can look in these metabolomics and proteomics and clinical tests for people who are at the extremes, who are in the bottom 20% of health or in the top 20% of health as far as frailty index goes. So that's how we will be doing it. And even so, what this shows is what these plots show is that this is a hard task. Separating these quintiles of frailty is a hard task in this data. Okay, so that's ignoring the outcome. Let's actually incorporate the outcome into picking the ordering of the data, the mapping of the data. And that's what these studies do. So it's going to look for 5 different components. It's going to take 5 times as long as that to do this. So I'm just going to go ahead and tell it to do all the plots as well. I'm only going to go in detail through the metabolomics. The proteomics and clinical tests I have left as exercises for you. After we start looking at this, I'm going to start those so that they can run in the background while we're talking about metabolomics. But here, component 1, it has chosen the direction that is most informative about the outcome, the frailty index, in the data, and otherwise has rotated the data so that it is spread out along that dimension. And then we're going to look at plots of component 1 versus component 2 and component 1 again versus component 3, so that the horizontal dimension that we look at will always be component 1, and then we'll be able to evaluate. So if we look at spread of the data in the horizontal direction, we'll be talking about component 1. And after we've discussed component 1, we're going to be looking at component 2 as the vertical dimension, then component 3, then component 4, then component 5. And the next step, the not great BER step, is just printing out a description of the performance result that we're getting out of running performance measuring on this model. And in the end, that performance will be plotted as an evaluation of how many dimensions were useful. One component, the first 2, the first 3, the first 4, the first 5, for interpreting the metabolomics data as a whole as a predictor of self-reported frailty index. So I almost managed to talk long enough to finish all of this. I just waited a few seconds more. Of course, then the plots will take a little bit of time as well. Yes, the last two, sorry, I shouldn't be looking at this. All right, so here's our plot of how many components are useful in interpreting the, trying to predict the frailty index from metabolomics. And each of these lines is a different distance measure. Basically, if you look at distance as an X distance and a Y distance, these are 3 different ways of combining that. Max distance, the blue, is taking the maximum of the X distance and the Y distance. Centroid is your typical 2D distance between 2 points, where it's called centroids because we're looking at the distance between any individual point and the centroid of a distribution, for example, for quintile 1 or quintile 2, the centroid of that versus the position of a particular individual. And Mahalanobis distance is what's also called the taxicab distance. So if we have an X distance and a Y distance, and maybe a Z distance and a 4th distance and a 5th distance, that is simply the sum of all those distances. So it's the distance that a taxicab would have to do if it was going to go 5 blocks over, 10 blocks up, another 15 stories up into a building, and so on. Okay, so the main thing I want to say about this performance is we're measuring performance with a cross-validation strategy where we have threefold. So we're using 2/3 of the data to predict the other third of the frailty index from the other third. And we do that 10 times to try to minimize variation. That is a very small number of repeats. In practice, we would probably do this at least 100 repeats, maybe more. But that takes 10 times as long as doing 10 repeats. So we're doing that here. So these are the values that were plotted here for the different components and quintiles. And I'm not really going to say much more about that. But here is a PCA-like plot. Okay, so if we compared this with the the PCA plot, the corresponding PCA plot. Here's the metabolites PCA plot, right? And here's the SPLSDA plot from the same data, where it is actually using the coloring to also choose the direction. So we don't have the nice ellipses on, where'd it go? Let's see where I put it. All right. So we don't have the nice ellipses to try to show the separation between these, but I think you can see that, yeah, there is some separation of color here with the pinks and greens more up here. Well, the greens are kind of everywhere. But here we've got ellipses that kind of show the 95% confidence interval of the location of the pink diamonds, and a green one for the fourth quartile. So these are the least healthy. The Q5 is the highest frailty index. Q4 is the next highest, 20%. So these are showing the direction of bad health, and the lower ones, 1 and 2 being blue and yellow, are showing the direction of good health. So the first component of the metabolites is showing is separating out some of the people who have poor health. The second component is singling out some of the people who have better health. Okay, so and those are different combinations of metabolites that are leading to that. So if we look at so that's all I have to say about components 1 and 2. Component 1 is the same here, so we're only trying to evaluate the vertical dimension here. And here we see, again, some separation of low frailty people from high frailty people. This is actually 5, 4, and 3. So it's more of a dichotomy, whereas the other was a trichotomy. Component 4, again, separates some people with health from some people who are actually not in quintile 5, but in quintile 4 and 3. Quintile 5 is headed in the same direction as people with good health. So I'm not quite sure why that is, but that is what these metabolites have to do with. And similarly for component 5. And then these can be evaluated with area under the curve on receiver operating curves. And again, we don't really need to focus on all of the separations that it's possible to do. We're really only interested in separating out the first quintile and the fifth quintile. They're using a different color scheme here. Quintile 5 is the purple, which is best discriminated if you're used to looking at ROC curves. Getting all the way up into this corner would mean you've got 100% sensitivity. In other words, you're finding everybody in that quintile, whereas you're missing, you're finding nobody, 0% specificity. You're finding 0% of the people who are not in that quintile. All right. So let's go ahead and run the next whole set of these. You're welcome to do that same thing, and there's a summary at the end, after the end of the exercises, to show those values that come out of those ROC curves right next to each other. So we'll just run all that, and while it's running, we can switch back to the slides. All right. So we went to the notebook. We came back, here are the PCA results that I showed you. Here's the code that was, so I was supposed to go to the code after showing you this slide, but this is how simple the call is to run PLSDA. We have our predictive data, we have our outcome, and we only need to specify, in addition to that, the number of components that we want to try. Then this is our cross-validation code, and we did our plot. And then we did a few additional plots, which fortunately the Mixomics package provides. So when we came back from that, we had these, and here now you can see components 1 and 2 for all 3 types of data at once. So in each case, component 1 is separating out people with poor health, with high frailty index. And in each case, on the vertical dimension, people with better health are being separated out from the tangle. In the case of clinical tests, that happens to go the negative direction. I don't know if this has been explained to you, but like with PCA, it isn't clear for SPLSDA whether a value is going to go left or right or up or down. The main issue is that it's separating out. So multiple runs of PCA will often flip they'll give you the same exact picture, but one of the dimensions may be flipped. And that's because mathematically, there's really no difference between separating things upward and separating things downward. Although I'm not certain, but SPLSDA, since it has an outcome to look at, it may force the original direction to be higher to the right. I'm not entirely sure about that method. But as I claimed, here are the ability to the metrics that tell you how well you can separate each of the quintiles from all other quintiles. Now, we cannot separate the inside, the middle quintile from the others very well at all. A 0.5 on this would be essentially flipping a coin to say, is it or is it not in the third quintile? So this is almost no discrimination. And that's not really terribly surprising because these people are not, all very different in health. The majority of them are healthy. A few of them, perhaps less than 20%, are healthier than typical. And a few of them, perhaps 20%, perhaps less, are more unhealthy. Sorry, these people are more healthy. Those people are less healthy than the typical member of the Arivell cohort. So for those, we're getting over 75% separation. And that is at least something. But again, as I told you, this is a challenging problem. We are trying to tell how many health defects somebody has from a type of data that is very low level. This is the clinical tests are a little bit higher level because doctors have selected these tests for particular ailments that they're looking for. However, even those have an interpretation step. So it may be that you have a high value, like if I'm sure you've had blood tests recently yourself. If you have a value that's out of range, there may be a normal explanation for that. For some of the ones that I was shown on my most recent blood test, it was because I'm old. I'm now 60 years old. So my ranges have actually changed and the out of range bars are age independent. So I was told not to worry about some of those. Other ones the time before I was told, yes, do worry about, and I made some changes and they got back in line. So I'm glad I've been taking the tests. At any rate, so for block SPLSDA, which is combining these omics sets together, if you remember, last time we had to do something to match the data, the correlation data across different types of omics so that we could include them in a single analysis. Block SPLSDA does that automatically. It doesn't use the same kind of strategy, but the way it does it does explicitly assume that there is correlation in the data sets of different types, which is a good idea because this data is correlated. One person's metabolites and proteins will be correlated in a way that another person's metabolites and proteins may not. So all of that needs to be taken into account when combining these omics. And block SPLSDA, S is for sparse, we'll get back to that in a bit, needs to take that into account. Now, we have here specified that they are only have a correlation coefficient of 0.1 with each other universally, which is actually not at all true. They are much more tightly correlated than that. However, experience with block SPLSDA shows that it is better to underestimate that at this point in the design matrix. So to not throw out too much of that correlation, because some of that correlation is actually useful signal. So that is why we have essentially lied to block SPLSDA. They told us to in their vignette on how to use it. Okay, so let's go back to the notebook, and that's what our results are going to look like. So, all right, so these have all finished, and now we're at the point of running this. Now this, we can run this step and the design matrix step, and I believe this step. This shows us how much the actual correlations are. So they're not 0.1 at all. They're 0.7, 0.9, and 0.65 in these three cells respectively, and then these are mirrored. However, we've already talked about that. This is the step that will take 20 minutes to run. So if you want to, go ahead and run that. But if you do run it, you're going to want to switch to the HTML to look at the rest of this. So I'm not going to, I will go ahead and start this, and maybe we'll come back to it at the end if there's interest. As a matter of fact, I'm going to run the rest of that notebook. Let's see, which you can do with run selected cell and all below. So that'll finish off the entire notebook. But while we're letting that run, let's look at the canned results. Right. And as I said, we may not get the same results out of that run as we did out of this run, and you may not get these same results either. Okay, so here's where we left off. We just ran this. It takes about, I guess it's only about 10 to 20 minutes, for this particular step. It fits 5 components, but it only gives you 1 progress bar, and which is different from the others. And then here's the output for that. So then we can look at, this is overall. This is not for each individual omics, because we've combined the omics now. This is overall. And there's a performance estimation step that does what we did before, cross-validation. In this case, we were using 10-fold cross-validation with 10 repeats. Again, I would increase that. But here it tells us how many components turned out to be optimal for the 3 different distance measures. And in particular, the one that I like best, the centroids, it claims that this is better than that, which means that we had to have more parameters when we were fitting this model. So the amount better that using more parameters didn't justify that number of parameters. That's a model choice kind of issue. However, I disagree with, so the typical use of this table is to compute a weighted average of these, and that was coming out with just 1 as the number of components. And I know for a fact that the other methods which I have seen give much better discrimination, lower error rates, use 4 or 5. So I chose to override it and say I want 4 anyway. So then I need to make a final Diablo model. And in order to do that, I need to deal with the sparsity. Just a second. Emily, the phone's ringing. Yeah, sorry, I'm going to be a little distracted because we're getting a delivery today, and somebody's talking in my ears, but my wife will be talking about that. Okay, so I'll try to ignore that. So I need to deal with. Can you give me the phone? Sorry. Yeah, it's talking to mine. You're gonna have to. Just a moment. I'm so sorry. Okay, now you can use the phone. All right. Hearing aid problem. I told you I was getting old. All right. So yeah. So here I am trying to determine how many. I'm trying to set parameters for making this a sparse model. Right. So for each of the different types of omics, for each component, we want to limit the number of analytes that are allowed to be used to make that. And while more maybe make a more predictive model, limiting it to a small number has a couple advantages in terms of interpretation. It is much easier to interpret a measure that only includes a few analytes than one that includes 500. So for that purpose, lasso regression is used for the same kind of reason. You want to have a simple explanation rather than a very complex explanation. You can show that for the most part, it's possible to get good results with a much smaller number of analytes for this kind of prediction. So we're going to try to do that. In this case, this test.keepx sets up a grid of 3, 6, 9, 12 as limits for the metabolites, same for proteins, and 3, 6, or 9 for the clinical measures. That how many is a matter of choice, but the point is it's going to make a cube out of all 48 possibilities there for the sparsity parameters, and then we pick some. So that process takes another 12 minutes here, and it gives these outputs. Frankly, I don't know what these numbers actually mean, but that doesn't matter. Diablo does. So I use the ones that it decided were optimal to try to restrict the sparsity of the model, and then I can get our output. So these three plots are essentially what is shown in the slides, although I took the slides from different. Actually, I don't remember whether I think I took the ones in the slides from a different run from this, so they won't quite match up. But let's go back to the slides to take a look at that. Okay, yeah, definitely a different run, but we interpret this the same way we did before. We look here, in all cases, the horizontal is component 1, the vertical is component 2. So one thing you may notice is that the component 2 is not adding a whole lot to component 1, but the roles are essentially the same. So in each of the omics, we're looking at separating out some of the people with the worst health, whereas in component 2, we are separating out people with better health. And in this case, all three of them are headed the same direction. So I don't know whether that was by chance or whether it's a feature of the optimization process in SPLSTA. However, Diablo gives you a variety of additional visualization plots, and I encourage you, if you take a look at their vignettes, to go through those in some detail. There are a couple runs at the bottom of the notebook that I basically commented out by turning them into raw cells. Because one of them didn't work, and both of them take too much time. So I decided I would let you explore that on your own, but I wanted to give you some examples. So instead of the plots we were just looking at, which showed individuals against components 1 and 2. These are plots of individual analytes against component 1 and component 2. So this is essentially a representation of the most important parts of the eigenvector for component 1 and the eigenvector for component 2. The red red triangles are individual proteins that contribute strongly to component 1, and same with metabolites in clinical tests. So that tells you roughly how many there are that are strong. I don't understand the scale on these. It is not identical to the weight that's actually given to each. But there's a handful for component 1 of all across all types, and a handful for component 2. Here's component 3 and component 4, which we have not looked at in any of the visualizations before in the notebook. And what you can see here is that there's a large number of proteins that I think one of the limits, I think, was 12 for proteins. So there's some set of these, some 12 of these that are important for component 3, and they're pointing toward the negative side, and another set of them that are even more important for component 4. And so the 12 for these and the 12 for those add up to some of the ones in there. Presumably some of these were not included in the actual model. These are just showing their effect if they had been chosen. All right. Let's take a look at the hits for component 1. And I think what you'll see here are some results that are very similar to what we saw in the PCA analysis and in particular in the clustering and overrepresentation. We see FABP4 and leptin, which are both proteins involved in energy production and in fatty acid oxidation and the Krebs cycle or the TCA cycle. The laboratory tests are all 3 about insulin and insulin reactivity in insulin resistance. And the metabolites are somewhat different. There are some sphinganines and sphingosines, which are components of the cell wall. They are made out of diacylglycerols and triacylglycerols, so they're not entirely separate. However, cortisol is a sex hormone, well, a steroid hormone anyway, and the glucuronide is something that gets added to it to make it soluble so it can be discarded from the body, and that's why we're finding it in concentration in the blood. And someone who knows a lot better about these metabolites than I do might be able to interpret it more, but one thing I would like to point out is that all of these have weights pointing in the same direction. So all of these, an increase in any one of these in the values in the data will push you toward the right of component 1. So all of these are markers of poor health. For component 2, we see markers that push you both directions. So if you remember, down on component 2 was in the direction of good health. Quantile 1 and quantile 2 quintile 1 and 2 on the frailty index. And we have some things that push you in that direction. And we have others that have positive effects, which presumably would be pushing you in the opposite direction. And again, trying to explain exactly what that means about health is beyond the scope here, but also that is why we do these, so that we can look at these and try to figure that out. So this kind of model is good for making hypotheses that can be tested afterwards. As I said about epidemiology, the health science space is very complicated, and there are rigorous testing requirements for anything that's going to be used in it for a medical practice. So you should definitely consider these analyses as aimed at generating hypotheses, not at making firm decisions about what to do with these values. Okay, so that ends the first, our use of the first notebook. And because of the way that this works on SageMaker, I'm going to go ahead and stop that. Well, actually, I'm going to get rid of that notebook entirely. I'm going to stop it and close it so that I can run the other one without any trouble. The HTML notebooks don't actually run, so they don't use nearly as many resources as the notebooks, the live notebooks. Okay, so for session 2.2, let me get some of the data loading started. We're going to get to our first ggplot call, so that, I don't know, well, actually, it's the second ggplot call. So I'm going to start those while we're talking about the slides. Okay. Right. So here, we're going to be working with the same data, except I'd like to remind you that we're going to be looking at yet the last of the three definitions of frailty index. And there's a good reason for that, which I'll get to in a minute. So longitudinal data analysis requires special methods for a couple of reasons. One is that we're looking at the same measurement over time. And if you think about it, if you have an individual person and you measure, say, the potassium in their blood, and 6 months later, the person has not changed their diet, and you measure their potassium in their blood again, chances are very good that it's going to be at roughly the same level as it was before, because it's the same person. Whereas if you chose another person and measured their potassium level, maybe this person is a vegan and the first one was a carnivore, you're going to have somewhat different values for that person, but those differences are going to be consistent over time between one person and another. Okay, so the point is that when you measure over time, the measurements at one time point are typically correlated to the measurements for the same person at a different time point, right, over different time points. Now, that's not entirely true. There are some things that vary over the course of each day. So if you measure once in the morning and 6 months later you measure in the afternoon, those two values may be totally uncorrelated. It depends very much on which protein or metabolite or clinical test you're looking at. For clinical tests, they try to do that in a standardized way, and that's why for some of them they ask you to have fasted the night before, so that they know that what you just ate can make a big difference in those, and they want to standardize that from one test to the next so that they do end up correlated from one test to the next. Measuring so many proteins, and some of them are tied to circadian rhythm, that is not going to be possible. You could not possibly specify all the things an individual would have to do in order to not affect any of those tests that way. So we are going to have to treat each individual analyte as independent, having a different story as far as how sensitive it is to when during the day or whether you fasted or whatnot. So for these, I think they may have given the advice to fast because they wanted to measure things that are affected by fasting, but people weren't restricted in when they made their appointments and things like that. So at any rate, the point is that we need to use some kind of predictive method that deals with this structure, this time correlation structure in the data. And we're going to have to use methods that are appropriate for the type of omics data that we have. So for instance, if we were going to collect EEGs, you get electroencephalograms, you get a lot of data points in a few minutes. thousands of data points maybe in a few minutes or 10 minutes for an EEG. Also, you can kind of synchronize one person's EEG with another person's EEG, or an EEG from one point on the head to another point, or measuring electrical impulses from the heart. So all of those can be dealt with in a particular way, and you're going to need some method that can handle thousands, or in some cases, if you're using Fitbit data, maybe even millions of data points over a long period of time. For us, we have three. So we have three measurements. We have one at baseline, in other words, whenever the person joined the program, and that's a different day than necessarily anybody else did, but then roughly 6 months later and roughly another 6 months later. There are going to be data issues with collecting data from humans. So part of the reason I'm going into this is that a large part of the reason for this seminar is that this kind of data, this multi-omic kind of data, is starting to become more available. It's starting to become available in hospital stays, in clinical lab studies. People are starting to be able to measure themselves for a lot of these things, or asked to be measured for these. So all of those are going to become data sources for people like us in the future. And so I'm trying to describe some of the data issues that are going to come up anytime you try to deal with this kind of data, because they're very different from what you might expect from having taken courses and where textbooks tend to show you very clean looking data. Also, many more systematized trials, like clinical trials to date, have collected large numbers of people that are cases or controls for a particular disease. And they've very carefully set up their data collection so that they avoid issues of the types I'm talking about here. However, even in those, you are going to have people who missed their appointment, or who had 5 blood vials collected, and one of them got dropped on the floor. They had to come back in a few days and get that one vial retaken. Well, when that kind of thing happens, 4 of the vials have data from one point in that person's life, and the 5th vial is from a different time of day on a different day. So that data is decorrelated in a way that someone who got all their 5 vials at the same time are not decorrelated. Those are all from the same time point. So I've gone over a number of these because these are going to be ubiquitous issues in systems biology. So we're going to look a little bit at how we handle them in this case, but mostly I wanted to let you know that they're there and they're always going to happen to me. I'm sorry. Okay, so what do we want to do with this data? That's another good question. We may want to predict future values. For example, with potassium, take potassium measurement, we might want to have an idea what an individual's potassium measurement will be next time he comes in, so that we can say, well, if it didn't change, he clearly didn't follow our protocol, because the protocol should have changed it. Things like that. So we can try to predict future values of the measurements themselves. We can look for structure in the whole set of measurements. This is what I think of as systems analysis. So you're trying to look for the mechanisms underlying the system, where, if you change potassium content, you might have to also, that might trigger a change in chlorine ion content, because one of them is positively charged and the other one's negatively charged. And if you don't have a balance of positive and negative charges, your body has static electricity, things like that. That's a silly example, but there are much better examples. But those are things that we can look for. And generally, when we do PCA, those are the kinds of correlations or clustering analyses we are trying to do. We can also try, as in clinical trials, to determine differences between groups. So in our example, we didn't start out with a treatment group and an untreated group, or a control group and a case group, people with and without a disease, we had to define groups. So we measured frailty index, and we can look at quintile 1 versus quintile 5. Those are two groups. They don't span the entire set of data, but they are groups that we can compare. Or we can try to reach the Holy Grail, which is to try to identify things that are actually causing reactions we see in the system. And one of the methods, most of the methods that are statistical methods that are intended to try to find causal relationships exploit the idea that causes have to come before effects in time. So there are a variety of approaches now that try to use that fact to determine that something is actually causing an effect. Whether that's successful is a totally different question and beyond the aims of this talk. Okay, so what methods are we going to use for this? What goals do we have? Well, in our case, we are going to be trying to characterize how successful the Aravel program was. Aravel collected all this information about clients and offered lifestyle coaching or health decision coaching to the clients in an effort to improve their health. We now have a measure, the frailty index, of the health of these individuals. So did that improve over time? That is the question, basic question we are trying to answer. And we are going to answer that by trying to make a model of the average frailty index over time for these individuals. The only frailty index we have that we've measured, we were able to compute over time, is the longitudinal one taken from the laboratory tests. Okay, so we're going to be trying to fit a model to the data so that it fits the longitudinal frailty index. Okay, that's our goal. How are we going to do that? Well, that is essentially a regression problem. We need to use a regression method that, so fitting a model to data, and the model shows you what the typical value in the data is. In the notebook, there is a plot of how that laboratory-based frailty index looks over time. It is a spaghetti diagram. I find it completely uninterpretable. I would shudder to try to say what is the typical trajectory on there. So making this model will actually give us a much better visualization than that actual look at the data does of how things changed on average for the Aravel data. Okay, so what we're going to use is called a generalized linear mixed effects model, or a GLMM. So LM is a linear model, which is typical regression. LMMs are linear mixed effects models. Generalized linear models are another variant, and we're going to add those two ideas together to get generalized linear mixed effects models. We didn't do this first. Other people have done this before, but I'm going to go through why each of these parts is important. So first, a generalized linear model is intended to expand the types of data that you can use linear models for. And it does that through two innovations. One is, instead of trying to predict a particular value y, so for us that would be the quintile of, it would be the lab test-based frailty index value. We can try to predict a function of that. And secondly, the only requirement about this function is that it needs to be a one-to-one mapping. So for instance, you could use the logarithm of something. That's a one-to-one mapping. If you know A is the log of B, you can invert that and say B is e to the A. And all of these other examples that are given are invertible in that way. They're all monotonically increasing functions, essentially, or monotonically decreasing in the case of the inverse. But, all right, so that's one thing. And I actually think we're using the identity map in this case, so I don't think that that one's particularly important for us. But we also have each of these values here is what we're going to be calling an effect. So we want to find a parameter that shows how much of effect A and how much of effect B and so on is required to fit the model, fit the data to a linear model. So linear means these are summed up, they're weights and sums and no multiplications or any other funny business. But the other big innovation in generalized linear models is that you don't have to use a Gaussian distribution as the distribution for the error function. So when we were measuring things like metabolites and proteins and laboratory tests are usually either those or ratios of things and things like that. There's a wide variety of types of things here. We want to have a more general distribution family that we can use to model the error in all of these different things, and including frailty index, so that we can fit a model that is more realistic to the data than one provided by the Gaussian model. To take an example, protein measurements, the amount of a protein is determined by a process that is fundamentally not additive. It is like stepping on a gas pedal. When a cell is making a protein, it has a mRNA that is using to translate into proteins, and it does that at a certain rate, like the rate at which you're letting gasoline into your engine. That does not add, one dollop of protein every time. It ramps up the production of that protein. So you might go from producing essentially none of that protein to producing 10 units per minute, right? That is a rate, not I made 10 molecules of protein. So it is not an additive thing, which is what Gaussian models are good for. Gaussian model is a sum of many little effects, adds up to a bell-shaped curve. Instead, what you have is really more of a log Gaussian, right, or a log normal variable for protein production. And the Poisson is a similar kind of thing. A gamma model is a similar kind of thing. Negative binomials are also similar. These are all variants in the exponential family for measuring things that have one or another type of process for how they are produced that is different from the Gaussian model. So those are the reasons why general linear models are used, and we're going to explain especially the error distribution for that. I believe that we use a gamma model, which is a fairly general version of all of these. The other thing we're going to do, the mixed effect part, is that in addition to these individual effects where we really estimate one parameter, one coefficient for that effect that is the same across all individuals, we have another type of effect called a random effect. So those ones are called fixed effects. It's a fixed value all the way across the set of data, all the individuals. What linear mixed effects models allow you to do is add random effects, which are random variables that have one value per group for some set of groups. Okay, so the way I'll just describe the way we're going to use that. Each group, in our case, is an individual. And what we're using this to model is the starting point for the value that we're measuring over time. That's the frailty index, the lab-based frailty index. So we know that they're correlated from one time to the next time. What we're doing is we're inventing we're assuming there was a frailty index, an unknown frailty index before the 3 that we were able to measure every 6 months. We don't know what it is, but we provide a distribution, or at least we allow it to form a distribution that shows the distribution of starting points for frailty for all the individuals in our study, all right? And so we're going to fit parameters for each group, i.e. each individual, that represent that starting point, all right? But we're not going to make them specific values, all right? We're not going to try to guess what the actual, value would have been had we measured it and calculated it. What we're going to do is we're going to allow it to be anything in that distribution for this individual, all right? And average the quality of the, try to fit the model in a way that uses a value from that distribution that is appropriate for this data by averaging averaging over all possible values of that in the data fitting process. Okay, so I'm sorry, that's a fairly complicated idea, but that is the idea of a random effect. So there are 2 points about it. One is that there's one value per group. The values for each group are different from each other. They have nothing to do with each other. But within that group, all individuals use that one same value. And we don't actually estimate that one value. We marginalize out of it. We average out over all possible values given a distribution. All right. So if we had groups of 10 rather than individuals as groups, we would be using the same starting point for 10 different people. That's not very realistic, so we're doing it one per. Okay, so that I hope that explains why we're using the framework that we are using. I've given you a website down at the bottom, a link to a website that you can follow to learn more about GLMMs from somebody who knows them better than I do. So I highly recommend that. Okay, so since we're talking about a new type of frailty index, let's talk about the actual model we're going to use in that framework and look at some statistics about it. So all of the frailty indices cover roughly the same range. Here we're not actually looking at well, actually, I think we are looking at the lab only frailty index, but we're only looking at the first time point. So the baseline one. And what we've done is split this data into females and males and 2 age groups, under 55 and over 55, roughly. Well, 55 goes in the under category. So the point here is twofold. First of all, we see that on the right, we see that the distribution of frailty, which I've plotted kind of strangely so that I can match the scale on that and this. This is a histogram showing how many individuals had a frailty index in which value for females versus males. And the primary difference you see here is that there are more males as a fraction of males. There are more males with a high frailty index and more females with a low frailty index. And the reason for that, we can actually explain looking on the left, which is that the distribution of frailty indices for males and females under 55 are pretty much the same. They have the same median, but those are both different from the frailty distribution for males and females older than 55. They have essentially the same distribution. There's some difference in variance, but again, it's got the same median. So really the difference that we're seeing here is due to a difference in how many females are young in this data set versus old, versus what fraction of the men are young versus old. There are more older men in this data set than younger men relative to how many older females and younger females. That's the only way to describe, to explain these two plots. All right, so in order to make our model with GLMM technology, we're going to use the following things to estimate this frailty index. We're going to use the shape of a spline fit to the frailty index over time. We're going to, not the actual frailty, because that's what we're modeling, but this spline fit is a curve that passes through all the frailty index, the 3 frailty indexes we have for each individual. And these basically capture the shape of that curve, how much it's changing and how fast it's changing, without actually capturing the value there, all right? But we're also going to use the individual's age, 4 principal components of an analysis of genetic markers that tells us roughly what continent these people are from. So the first principal component is separates people based on whether they're from Africa or not. I mean, their parents are living in Africa or not. And then PC2 is, I think it's European versus Asian. PC3 is Asian versus East Asian versus South Asian, and PC4 is from the Americas versus from the Old World, roughly speaking. I happen to know that because I've done a lot of that kind of analysis, but the point is that is the big rough scale distribution of people around the world. It can be represented in these 4 statistics. So basically, we're taking age and ethnic background into account, as well as the shape of the frailty index. And we're using a starting point. So we tried 3 different models for each client. So the ones in parentheses are the random effects in a GLMM model. And here are the effects, and here is the grouping. So per client, we have a constant term plus a slope term multiplied by the T1 spline element. All right. So that's just to give us a way of modeling. Pardon me. We tried 3 different versions of the random effect model, and this is the one that won out according to a model selection criterion called Akaika's information criterion. We tried just a constant, we tried just a slope element, and we tried the combination. And the combination, even though it takes 2 parameters per client rather than just 1 for either of the other 2, it was able to provide enough improvement in the quality of the model to overcome a penalty for those extra parameters, which is how the Akaika's information criterion helps you choose models. Okay, so I hope that was all clear. If not, you're welcome to ask me again later. But let's take a look at the results. So if we take a quick look at the notebook here. All right, so, oh, I haven't done all the plots, I'm sorry. Let's just run all the rest of it. Run selected cell and all the, okay. Yeah, so if we catch up, there's that horrible spaghetti plot of what the data actually looks like. So we're trying to say, what is the average trajectory through that for females, and what is the average trajectory for males? If you can do that from this data, more power to you. I can't. Okay, so let's see. Right, so this is the trying out the 3 different versions of the random effect. I don't know time on running short. Okay. Yeah. So what I wanted to get down to was, where do we run the model? This is where we split it into above and below. Oh, that's after we've run it once already. Okay, so we run the model here. Yeah, so right? Okay, so here's where we get to run the model. Again, we have to define the formula, but then running it is right here. So this is the code that actually runs it, right, as a run of Glimmer. Okay, so once again, the formula is getting the data set up and defining the formula is really the nuts and bolts of how to use that. And please look at the documentation for how to do that. Okay. So here's the end result. Okay, modeling females and males separately. We had 674 females and 425 males for the longitudinal analysis. And what we see for both is that, we see that older has higher values. So generally speaking, people with higher frailty are less well. In both cases, as a result of being in the Aravel program, the males and females both dropped a small amount on average of their frailty index. So the lifestyle coaching works is the overall message there. Now, I want to point out that this is a fairly small change in average. All right, so here's the whole distribution of the frailty values across all the individuals. This little section corresponds to just this dotted section. So we're talking about a change of, okay, so this is 22% of the health defects, 20%. We're talking about a change of maybe 2%, roughly, and then it changes less than 1% over the next part. And then it seems to diverge. Now, I caution you for putting too much interpretation on these ending parts, because the data was supposed to be collected at each of these dashed lines. The only reason we have any data at all after that point is that people shifted their appointments to some extent. So if it were me, I would not try to interpret these different shifts. I would be looking mostly at where these lines cross the intended time points, which are also the knots at which the spline shape was measured when we were adding that to the model. Okay. I should have about 5 minutes left, 4 or 5 minutes left before our 10 minutes for questions. So I want to show some additional, briefly touch on some additional types of things we can do to look at how this performance happened. One is to look at polygenic risk score. Now, we had a polygenic risk score for longevity. In other words, if you have a high value for this, your genetics say you may live to a very high age. I think it's 95%, 95th percentile or 98th percentile of all human ages. Or maybe it's the ages in the data set we're looking at. At any rate, that's a detail I'm not real clear on at the moment. But the point is that if you have a higher score for this, you're more likely, according to your genetics, to live a long time. If you have a lower score, it's more likely that it is more likely than for other people that you might die early due to some predisposition to disease. Okay, and you can see that effect actually over where I've separated out these two box plots into different age, so ages of different individuals in the study. For the most part, there's no correlation for the people between polygenic risk score and people's actual age when they're young. All right. Now there might be a little bit of a tiny trend as you increase in age with females, but with both of them at the last time point, the people who are fairly old when they start the program, they tend to have a higher longevity predisposition to longevity. Okay, so this is very anecdotal. These are very small numbers of people, but this is actually what one would expect for this kind of a risk score. Okay, that's the entire reason I'm showing it to you. We don't see much difference between men and women in our study cohort, but we do see a rise over age, a very small rise over age. So how does that actually go with our frailty? Well, here what we've done is we've separated out individuals for 4 different quartiles on PRS and shown how these 4 different groups fare on this model. And what it shows is that people in the first quartile, in other words, people with the most likelihood of dying young, have a higher frailty index, as we might expect. The people in the middle have a lower frailty index, but the people at the high end of the frailty index, so the ones who are most likely to live long, have a higher frailty index among women. That is different from the pattern shown among men. For men, people who have a high risk of early life disease have a poorer frailty index, and everybody else tends to be down in the lower section. And that is because of the effect we noted earlier, which is that, which I think has been noted earlier, which is that women have a much larger difference in frailty based on whether they are over 55 or under 55 than men do. So something happens for women that at around age 55 that leads to a dramatic increase in frailty, or at least separating at that age, you see a dramatic difference in frailty, whereas for men, you don't. And again, I wouldn't try to interpret this wide variation way at the top end, because we have even fewer older age men than women, I believe, in this. All right, so there's really not much data to be looking at that far. And that takes me to the end. So I believe I have 10 minutes for questions.
Okay. So can everyone see me and hear me and see the slides? Perfect. Okay, everyone. Welcome to session 3, machine learning analysis. I'm excited to be your guide for the afternoon. And so today I'm going to be talking a little bit about machine learning, how it works, the process and steps that data scientists and machine learning researchers are going to take on a typical data science project. I'm going to walk you through those steps, and then we're going to actually get to dive into some code and do some machine learning. And before I begin, I sort of want to just talk a little bit about why we might do machine learning. And sessions 1 and 2, Max and Lance did a great job talking about the frailty index, why it's important, why we might want to learn more about it. And with machine learning, we're going to continue on that journey. We're going to continue to learn more about frailty index, and we're going to try and see if we can predict which patients will actually have a high frailty index. And not only that, but we're going to try and learn about which features and variables are important in predicting high frailty. So I'll talk about all of that today. I also want to give a bit of background about myself. My name is Alexandra Ralevski. I'm a data scientist here at the Institute for Systems Biology in Jennifer Hadlock's lab. I work with machine learning, data science, and large language models to build predictive models of disease for patients using electronic health record data. So before we begin, I just want to let people know, if this is the first session you're coming to, that there is a Discord that's available if you have questions about the code, just general questions about any of the sessions, where you can talk to and connect with other people in the community. We'll be using Amazon SageMaker. That's where all the code is hosted. I'll do a little demo of how to get into SageMaker quickly, but there should have also been slides sent out in a welcome email to you that should guide you through the steps of how to get to SageMaker and get to that code. And all of the code will be posted, or is already posted on GitHub, along with all the notebooks and the PDFs of all the slides. So for those people who got an invite to Amazon SageMaker, should be everyone, if you signed up for this workshop, it should ask you to sign in. Once you sign in, you'll click on Amazon SageMaker Studio, and that will take you to a window that looks like this. And you might have to click on Jupyter Lab in the corner here. And then it should take you to your own personal workspace, Jupyter Lab space that's been set up for you. If it's not running, you can hit run and you can start it. If it should be open and running, and if it is, you can click open, and then it'll take you to something that looks like this. So you'll have the Aging Workshop 24 folder, you can click on it, and then here are the 3 sessions along with all the slides. We're going to be working in session 3 today, so you double click session 3, and you'll have 2 notebooks, the data analysis and the ML logistic regression notebook. Those are the notebooks that have the code that we'll be working in today. And if you want PDFs of the code, you can also follow along in that way as well. The code has, I've rerun the code, so it should have all the outputs there already, but you can feel free to follow along, and you can always hit run and rerun the cell. It should work, as you follow along with me. Okay, so before we dive into any code, I want to start by giving an overview of sort of what machine learning is, because I think everybody has, maybe they have a different definition of machine learning. LLMs are very popular right now. People are hearing a lot of buzzwords. And so I just want to make sure everyone's kind of on the same page. And I want to talk through the steps and walk people through the steps of a typical machine learning pipeline, right? Of what are all the questions we might ask ourselves? What are the different decisions that we might make at different points? And what are our different options at each decision point? So first, again, what we want to do today and what a lot of machine learning is concerned with is predicting, right? Predicting the future. And so an example to think about is, let's say you have a soccer team, and you're a fan of this team, and your friend might ask you, hey, well, do you think the team's going to win tomorrow? Let's say it's the Seattle Reign, right? And what are you going to do? Well, you're going to think, okay, well, how many games have they won so far this season? Is anyone injured? How many goals have they scored? There's different features or variables that you already have in your mind that are associated with the outcome of winning, right? And so what you do is you take all those features into account, you sort of come up with a model in your mind, and then you make a prediction. You say, yes, okay, I think this team is going to win tomorrow. And then you watch the game and you see whether or not they've won. Hopefully, if they win, you bet some money on it. I'm just kidding. And then, you can maybe update. If they didn't win, you might update your model and so on and so forth. So that's basically what we're going to be doing today. Instead of a model in our minds, we're going to use machine learning models that are based on math to help us make these predictions. But I also want to just point out that machine learning can be used for a lot of different things. And it's really important to understand why you're doing something, what you're doing it for, and who the intended user is. We'll talk a little bit more about that. So I just included this cartoon because I thought it was kind of funny. This coach is talking to his teammates or to his players, and he's saying, okay, the other team's using machine learning to predict our play, right? So we're going to kick the ball with our other foot. Even if the model that the other team built is not very good, looks like they're probably still going to win, right? Because they've already gotten this team to to play terribly, right? And so it's just an example of, always think about your intended user, how your model is going to be used, and who it's for. Another thing to think about is, if we knew exactly what features we should include in a model, we probably wouldn't need to build that model, right? Because we already know what's useful. And so a lot of times we don't know exactly what to include. We don't know what data to use. We don't know which features to include. And so a lot of it's trial and error, right? Sometimes you just put some stuff in. Again, you're making educated guesses at each point. But sometimes you put some stuff in, you see how it looks. And then if it doesn't look good, well, you might start over, right? Or you might try different things. And so I'll talk you through the different steps and decisions points that you might make at each step. But keep in mind, it is a lot of trial and error, and that is a lot of the fun. You spend the majority of your time, like Lance said, cleaning data, figuring out which data to include before you even get to the machine learning model part. Okay, so first I want to give just an overview of the landscape of machine learning. And this is actually, if you would believe it, the least ugly version of this photo that I saw online. It does seem a little bit overwhelming, but I'll walk you through it. So basically, there's sort of 3 main categories of machine learning. And this picture is actually already out of date because it doesn't even include large language models. And large language models actually include, most of them include some combination of supervised learning, unsupervised learning, and reinforced learning, actually. But we're going to be concerning ourselves with supervised learning on the right-hand side here. And again, supervised learning is when you already know the outcome. You're trying to predict a particular outcome. Your outcome can take 2 forms. Your outcome can either be a quantitative variable, or basically a number, right? Are you predicting age? Are you predicting population growth, right? Some examples here. Or are you predicting a class, right? Are you predicting whether or not something is fraudulent, yes or no? Are you predicting whether or not this email is spam, yes or no? You can also do multiple classes of categories. So what Lance and Max showed you before, they're focusing on regression, and that's because our outcome variable, which is frailty index, is a number, right? So we could make it into a regression problem. But I actually turned it into a classification problem for us because I thought I'd show you how that's done. It's fairly straightforward. And also that I think it's some of the performance metrics for understanding classification problems are a little bit easier to understand. So we're going to be dealing with the classification problem today, but I would encourage you to also run it as a regression problem. So that's supervised learning. Unsupervised learning is when you don't have a particular outcome, you don't know what the outcome is. So an example might be if you have a group of breast cancer patients and they have different genes, but at different levels of transcription, and you maybe want to cluster those patients together based on the level of activity of those genes, but you don't know what those clusters should be ahead of time. You can do unsupervised learning and clustering to make those groups. Reinforcement learning is very popular with game AI like DeepMind for chess or AlphaGo, where you're basically given a reward, the model is given a reward for doing the task well, which is winning, or given a penalty for losing. And so the game will play itself, the model will play itself over and over and over again, trying to maximize that reward. So these are some of the different types of machine learning. It's a wild and beautiful landscape, and I would encourage people to learn more about it if they're interested. But again, we're going to be sticking to a very particular type of problem here today. Okay, so again, just an overview of when I say we're going to build a model to make predictions. Well, what does that really mean? So again, we're going to start with some data, right? And again, this could be, you know, the history of all the games that your team has played this season, let's say, for example. And I'll also use medical examples as well, but I'm starting with sports because I think it's a little bit easier for people to follow and understand. So let's say you have the data of all the stats for your team that year. And then let's say you have some features, and some people use the word features, variables, covariates, predictors. In this context, they all mean the same thing, which is basically, what are the things that are important for helping you predict your outcome, right? And here, this label that we have is our outcome, which is, did the team win, yes or no? And in our case, in the model we're building today, it's going to be high frailty index, yes or no. And I'll talk about how we're going to make that outcome. So you take all the features, right? All the ones that you think are important, and you have their labels. So for each game, right? Or in our case, for each patient, you'll have all the different features that you think are relevant with all that data. And then at the end, you'll say, okay, did this team win? Yes or no? Did this patient have a high frailty index? Yes or no? And you're going to feed that information to the model, and the model is going to learn the relationship between the features and the label. And more importantly, it's going to learn which features are important for predicting this label. So it's not just that we want to know whether or not the team won. We want to know what is important in helping the team win, right? What is important for having a high frailty index or a low frailty index? That's what we want to learn. So that's what we're going to do today. So here, all this is saying is we have our outcome Y. This is our dependent variable, and we're going to use our independent variables, our features, to help us predict that Y. And so this top half here, this is usually called, or this is called training. You're training the model, you're having it learn the relationship between the features and the outcome. And then you want to test the model, right? You want to see, well, did the model do a good job? Did we build a good model? So you'll take new data that the model's never seen before, has the same set of features. And then you're going to say, okay, model, from everything you've learned here, tell me whether or not these patients are going to have high frailty index, or tell me whether or not my team is going to win. And it's going to give you an output, it's going to give you a label, and you're going to compare it to the actual label. And you're going to see how well that model did, right? And this is called, again, this is called training, and this is called the testing. And so one thing before we move on, I think, especially when people are starting out in data science and machine learning, there's this idea that we have to build a perfect model, right? We have to build something that's right 100% of the time. That's not possible, right? It's in very rare circumstances, if you have an enormous amount of data and you're predicting something that's fairly straightforward, like, you know, fraudulent document, yes or no, maybe, or spam or ham email, ham being good email. You might be able to build a model that's really good, like extremely accurate. But for the most part, the world is complex. People are complex, right? We don't know whether or not our team is going to win the game. If we did, betting companies would be out of business, right? And the reason we don't know is because we might not, there's so many variables that you might include. And oftentimes you can't include every possible variable in your model. And a lot of times things can just happen. People can be outliers. There can be things that we might not have predicted. And so there's this very famous quote from the statistician George Box, who says that basically all models are wrong, right? You're never going to get a model that's going to perfectly understand the relationship between your features and your outcome. But that's not really what we're trying to do. We're trying to build models that are useful, right? And what do we mean by useful? We mean a model that has relatively high accuracy and can consistently, between data sets, every time we feed it new data, it can consistently give us accurate results. So that's really what we're trying to do. And anytime you see a model that is perfectly accurate, you should be suspicious of that model, again, because it's pretty much impossible to do for all the reasons I just mentioned. Okay, so that's just, again, some overview. Now let's talk through the main steps that you're going to go through when you want to build a machine learning model. First one is, I would say, the most important one, right? You want to define your problem and you want to choose your outcome, okay? And defining the problem is not necessarily as simple as, okay, well, I want to build a model because I want to learn more about this thing. That's an amiable goal. That's wonderful, right? It's in the pursuit of research and science. We love this. But you always want to think about, well, who is my model going to benefit, right? Who is going to use it, and is it worth building? So in the work that I do, again, I'm building risk prediction models of disease for patients using electronic health record data. So the first question in my mind, every time we think about whether or not we want to build something, is, how is this going to help the patient, right? How is this going to help the doctor? How is the doctor going to use this model? And oftentimes, depending on your use case, that might determine what kind of model you end up building and what kind of outcome you end up trying to predict. So I'll give you an example. Let's say you have a model with 10,000 features, right? And it's highly accurate. And then you give it to a doctor and you're like, okay, at every meeting you have with your patient, every visit, ask them these 10,000 questions and then you'll get an answer. And the doctor's like, that's ridiculous. Of course I'm not going to do that, right? It's completely impractical. So what we might actually opt for is actually choosing a model that has slightly less accuracy, but it only has 5 features or 10 features, because that's something that's actually going to get used in the clinic. And so again, it's always really important to consider who your target audience is, how they're going to use your model. And in relationship to that, you're going to choose your outcome based on how you're defining your problem. So again, today, we're interested in better understanding the mechanisms that drive frailty and drive aging. And so we're going to include variables and features related to aging, and our outcome is going to be, again, predicting frailty index. But you could choose a different outcome, right? It really depends on your problem and what you're trying to do. And I'll talk you through the different points in the code and throughout the presentation in which you could choose a slightly different outcome, or how you could define your outcome differently. And I would encourage you to try it out and see how your results differ. Next, you're going to choose your data set. Sometimes you might start with a data set. So here we started with Aravel, and we're basically prodding this amazing data set for different questions and different answers, right? So sometimes 1 and 2 might happen congruently, 2 might happen first. But again, it's really important that you are defining your problem and choosing the data that's related to your problem. You have to choose between 2 data sets, one that is very high quality and one that is very low quality. You want to choose the one that's high quality. What do I mean by high quality? It means that there isn't a lot of missingness. Lance talked about that. I'll touch a little bit more about missingness later on. But it has low missingness. You trust the data. You understand where it came from. You know that you're very confident that the results and the numbers that you see are accurate and true. And the reason this is important is because there's a famous saying in data science, which is garbage in, garbage out, which is if you don't have high quality data, a machine learning model is not some magical thing that just takes crappy data and turns it into an amazing model, right? If you have bad data, you're going to get a bad model. And so that's why we spend most of our time as data scientists cleaning the data, making sure we understand it, making sure it's high quality, to make sure that we can get a model that's as accurate as it can be. Again, deciding which variables to include. And I think in my next slide, I'm going to talk a little bit about the different steps and ways that you can choose your variables. Again, you're not going to know. If you knew exactly which variables to include, you probably wouldn't need to build a model. So there's going to be some trial and error in choosing different types of variables, and maybe going through different rounds of modeling to decide which ones you end up wanting to include. Then you're going to pick a model, right? Again, that comes very, very far down. A lot of people will start, yeah, absolutely, I want to use this model, and then I want to try different data. These three steps are just as important, if not more important, right? We'll talk a little bit about the different models that you might choose. Again, it's going to depend on your problem and your outcome. We're going to use logistic regression and tree-based models today, but there's many different options that you might choose, and I'll talk a little bit more about that. Then you want to train your model and test your model like I talked about earlier. And then you want to measure your performance, and I'll talk about all of that. Okay, so again, just to sort of hammer home this point, there's a common term in data science called overfitting, right? And again, this is where we go back to this picture right here. What are the chances that the new data that you give your model is going to be exactly like the data it was trained on, right? Probably not very high, right? If you're dealing with patients or you're dealing with sports or anything, the chances is that the new data is going to have some variability. It's not going to be perfectly like the original data the model was trained on. And so if you build a model that just understands the relationship in this data, it's probably not going to do a good job of understanding the relationship in the new data. So what do I mean by that? So take a look at this picture here. Let's say we're trying to discriminate between our X's and O's here, right? If we have a model that basically creates this pattern, let's think on a high level here. The model is basically taking this pattern, right? And let's say you get new data and the X's and O's are in slightly different places. They're still divided, but maybe have some X's up here, some O's down here. And this model just applies this exact same pattern over these data. Well, it's probably not going to do a very good job predicting, right? Because it's really just understanding the relationship of this data. And it's not flexible enough to understand slightly different nuances and relationships for new data. And so what you actually want is a model that generally approximates the relationship. So here it's doing a fairly good job of discriminating between X's and O's. It's missing some examples, but that's okay, right? Maybe there's going to be some O's down here next time or some X's up here. That's fine. This is a fairly accurate model, but it's robust enough to handle new data. So this is really what we're looking for. This is a case where the model that we're choosing, let's say there's a linear model, is not complex enough to understand that this is a non-linear relationship between these data. So you really want to make sure, you often want to start with a simpler model. I'll talk a little bit more about that, but you want to start with a simpler model, but if you're not getting good results, that's when you start using more and more complex models. But if you start with an overly complex model, not only are you in danger of sometimes overfitting the data, more complex models are prone to overfitting, but you might actually lose some interpretability in your data. And again, it depends on what your outcome is and what your problem is. But usually you want to start with simpler models and increase the complexity. And you ultimately want to, again, end up with a model that has a good understanding of the relationship, but can be applied to new data fairly well. This is oftentimes called the bias variance trade-off. I won't talk too much about it, but that's the general gist. Okay. So let me just take a second here. So again, the first step here is we want to define our problem, right? And Lance and Max have talked about this a lot. Our problem, what we're interested in trying to solve is better understanding the mechanisms that drive frailty, right? So we're going to predict patients that have a high frailty index. So how are we going to do that? So I'm just going to switch briefly over to the data analysis notebook here. And here we're creating a box plot of our frailty index, right? And this is the merged frailty index of, I believe, all of those 69 categories. And here this is giving us a frailty score. And from this box plot, we can see that the lowest frailty score that someone has is just above 0.05. And the highest is just above 0.4. And this box plot is showing us, this is the median, is this orange line here. It's just under, I think, 0.22. And this box plot is showing us about, so 50% of the patients have a score between just under 0.24 and 0.15. And so what we want to do is we want to say, okay, well, this upper line here represents the 75th percentile. So 75% of all patients have a frailty index score of below 0.24. So similar to what David Furman did with his inflammasome module of looking at high versus low patients, we're actually going to take the patients that have that are above this quantile, right? And we're going to define these 25% of patients as having a high frailty index, right? So we're looking at this upper quantile in this column, and we're saying that the cutoff is 0.24. We can see that here. And then what we're going to do is we're going to create a new column in our data frame. And this is also happening in this machine learning notebook as well, and I'll review it again when we get to it. But we're basically saying is create me a new column that for everyone who has a value greater than this cutoff, right, which we're defining as 0.24, I want you to give that patient a 1, and we're going to define this as the positive class. And everyone who has a value below that cutoff is going to be assigned a 0, and this is the negative class. And so what we're going to be doing with our models is we're going to try and predict which patients are going to have a high frailty index, right, as well as a low frailty index. And again, I'll talk a little bit more about that. But that's how we're taking this regression problem, right? So we're taking a quantitative variable and we're turning it into a binary classification problem where we're just predicting 0 and 1. And so that's, again, that's the problem we're trying to solve. That's the outcome we've chosen. But again, you can choose a different outcome, right? You can maybe do everyone below the 25th percentile is in the negative class and everyone above the 25th percentile is in the positive class. Or you can divide it up into different quartiles or quantiles, depending on your problem of interest. And again, I would also encourage you to talk with experts in the field to get a better sense of what a reasonable outcome is. Okay, so then we're going to pick our data set. This has already been done for us, right? We already have the Arabelle data set. We're very lucky. And this is just an example. We'll show you the actual data set, and you should have already interacted with the data set a little bit in the notebooks. But just to let you know, we have 169 patients that we're going to be using for our machine learning predictions today. And we have 76 features. So 76 things, variables that we think are important and useful in predicting this outcome, which we've now converted to a 0 and a 1. So here we can see for this patient, these are some of their features and some of their values. And this patient has a frailty index below 0.24. We're classifying it as a 0. And conversely, these patients have a high frailty index. And so all the features we're going to be using are demographics like sex and age. The WGCNA values the metabolomics and proteomics values that Lance created, and different labs like glucose, triglycerides. Okay. Then, what normally we would do before we even, once we defined our problem and our outcome, or maybe even while we're trying to define our problem and our outcome, we might do some data analysis and some exploration. So if we go back to this notebook, you can do things like, okay, let's say I have my 2 classes that I've defined, and I want to better understand the relationship between different features, right? So I really want to understand how HOMO-IR, insulin resistance varies for patients of different ages, right? And I would encourage you can take, here's our data frame, you can take it and you can look at all the different features that we're using, and you can choose different ones, right? So here, these are all the columns. These are all the features that we're going to be using. So maybe you want to look at calcium. Maybe you want to look at glucose. And you can swap some of those values in, and you can see how this plot might change, right? So I would encourage you to play around and interact with the data and build some of these plots yourself, right? Then we can also look at the spread, the distribution of our frailty index by the number of patients. You can see the majority of patients have a frailty index of around 0.15. You can also look at LDL, HDL, whatever you're interested in. And so a lot of times data scientists will spend a lot of time really understanding the data, the relationships of the data, to help better define their outcome or help them choose which variables they do or do not want to include. So this process can take a long time. Here, this is a nice way to sort of quickly visualize a lot of data together. So again, we have our frailty categories of 0 and 1, and maybe we want to look at the relationship between age and insulin, or HOMO-IR and alkaline phosphatase. And again, I would encourage you to swap out some of these features. You have to put them in both of these lists. We don't have to get into why. and run this code and see what you get, and just explore the data a little bit more and get more familiar with it. It's a lot of fun, actually, I find. Okay, so that's the data analysis part. So again, we would normally be spending a lot more time on that section, but in the interest of time and moving forward, we won't be able to today. But again, I would encourage you to look into that. Okay, the next step, and this is a question, again, that people ask all the time, is which features to include. So sort of two main approaches that you can take, and a lot of times people will do both, which is where you can ask an expert and say, hey, I don't know anything about aging, right? And if that's the case, you could always read more, better educate yourself and understand the problem in the field. But you can also talk to experts, and you can ask them, hey, what do you think I should include? And then you can also do domain agnostic approaches, right? Where you use other techniques to reduce the number of features down. Let's say you have 10,000 features that you think are interesting, but you don't want to model with 10,000 features. So there's different approaches you can take, mainly where you're selecting a subset of those features that you think are relevant. So one approach people might take is you could say, okay, well, let's say I have 10 features and I just want to build models with all the different combinations of those 10 possible features. It's not a bad idea. And you want to just pick the model that has the highest performance. It's not a bad idea. The problem is that when you get more than 40 predictors or 40 variables, you're trying to fit over 1 trillion models. And so it's not computationally feasible when you have a high number of predictors. And 40 isn't really high. We have 76 that we're using right now, and that's considered pretty low. A lot of times, again, when you're dealing with genomic data and biological data, you can have thousands and thousands of features. So another option is what's called forwards or backwards stepwise selection, and where you can basically start with a model with no features, and then you can add more and more features as the performance, add those features that improves the performance of the model. Or you can do the reverse, where you start with a model with all your features and then just reduce the number of features reduce the feature has the least effect on performance, and so on and so forth. Well, you might ask yourself, well, Alex, if I do that, I'm always going to end up with the model that has the most features, right? Because I'm either adding more and more features as the model gets better, or I'm removing features as the model gets worse. And so you might think, okay, well, just always choose the model that has the most features. Not always true. What we're doing here is we're trying to choose features that's based on our results from the training data, right? Just because a model does well on the training data does not necessarily mean it's going to do well on the test data. And so when you have models, different types of models, and models with different numbers of predictors, you always want to test on the test set, okay? You always want to test on new unseen data. Now think about why that is. Let's say I have a model with 3 features, and then let's say the performance is, 80% accurate. We'll choose accuracy. So it's 80% accurate. And then I add in a 4th feature. So I have a model with 4 features. Well, the performance of that model of the 4-feature model is never going to be worse than the 3-feature model, right? Even if that new feature that I added provided absolutely no, it didn't increase the performance at all. At minimum, this model is going to have the same accuracy, 80% accuracy as this 3-feature model, okay? Usually adding in another feature will increase the performance somewhat. And so the idea is that you can't compare these 2 models on the training data, because this model will always perform better, or at least will never perform worse than this model. So you always want to make sure that when you're taking multiple models, you're testing them on the test set, on new unseen data, okay? Another option, and one that we're going to explore a little bit today, is called shrinkage. And this is where you're doing regularization. And there's 2 methods called lasso and ridge, and I believe Max and Lance talked a little bit about this. And it's basically where when you have a coefficient in your model, a variable in your model, it is a higher value. Usually the model attributes that feature to being more important, but that might not always be the case. And so you can use these regularization methods to basically shrink the value of these coefficients. And lasso specifically can reduce the value of these coefficients to essentially 0 if the feature does not have is not correlated with the outcome. So what's an example? Let's say, again, let's go back to our sports example, and you want to include, number of shots on target and penalty kicks and astrological sign of the goalie, right? Astrological sign of the goalie probably is not going to be important in determining whether or not your team might win, right? Unless they're a Scorpio. I'm just kidding. I love my Scorpios. And so what lasso will do is it'll reduce the value of that feature, of that astrological sign feature, to essentially 0, and it'll basically remove it from the model. And so that's one way to remove features from your model. Ridge will not do that for differences in mathematical reasons we don't have to get into, but it will reduce the value of those coefficients close to 0, of those coefficients that aren't useful, those variables that aren't useful in your model. And so we'll talk a little bit about lasso and ridge today. That's what we're going to use. And then there's other dimension reduction techniques. Again, every time you're adding a new feature into the model, you can consider it as increasing the dimensionality of your model. And so again, if let's say you have 10,000 features, you might want to reduce it down to a few principal components. That's another way, again, to decide which features you're going to include and how to reduce the number of features down, because it's usually what people want. If you do have a model on your test set and it has 5 features, and you have a model that has 1,000 features, and they basically have the same performance, people will almost always choose the model with fewer features because it's easier to interpret, it's easier to understand, and it's computationally more efficient. Okay, feature engineering. So again, there's so much that you can do with your features and your variables. I'll touch a little bit upon all the different options, but again, you're already seeing how many different choices you might make when it comes to building a machine learning model. And so, of course, you can always add in more features. Let's say you're not getting very good performance. You're thinking, well, maybe I'm not capturing certain parts of the data, and there's certain things that are important to this outcome that I haven't included in my model. So, for example, some things we could have included, maybe BMI, education, income. Sometimes it's important, sometimes it's not. Medications. You might want to remove highly correlated variables. So if you have 2 variables that are basically doing the same job in explaining the outcome, if you remove one, your model performance is essentially going to stay the same. So you might want to remove, if you have 2 highly correlated variables, you might want to remove one of them, or maybe you want to combine them into a different variable. We won't deal with that today, but just something to think about. And especially when you're talking about biology and genetics, you can have a lot of things that are very, very highly correlated. Lance touched upon missingness. I'll touch a little bit more upon that when we get into the notebook. Again, you might want to choose a different outcome, right? Maybe you want to predict age as an example. We'll talk a little bit about class imbalance and how you might deal with that. And there's many, many more things that you can do. But again, it's important to really not just make a decision at each point, but understand why you're making that decision and the implications for your outcome and the people who are going to be using this model. Okay, step 4, well, choose a model, right? So again, there's many different options. I would encourage you to start with the simplest option. Today, we're going to be working with logistic regression and XGBoost, which is a tree-based method. And I'll explain a little bit more about tree-based methods when we get to that section. All of the models, so tree-based models, support vector machines, neural networks, can work with both types of outcomes, classification and regression outcomes. Logistic regression, despite the name, is a bit confusing, so I'll just spend a moment talking about it. Logistic regression is based on linear regression, and that's used for regression outcomes. However, logistic regression is confusingly almost always used with classification outcomes, even though it has regression in the name. So logistic regression is basically a classification model, whereas the rest of these models can be used with either outcome. So it is a bit confusing, but again, we're using logistic regression and XGBoost today to look at our classification, our binary classification problem of, does this patient have a high frailty index, yes or no? And again, logistic regression is the simplest of these models, and these models get increasingly more complex. Don't start with a neural network unless you absolutely know that you need to, right? They're can be computationally expensive, difficult to set up, a little bit finicky, and they don't have very good interpretability, right? Oftentimes you don't really know why the model is giving you the output it's giving you. Whereas logistic regression and XGBoost it's much easier to understand how the model got to that conclusion. But again, it depends on your outcome and your problem. So always consider that at every step. Okay, the next thing you want to do once you've chosen your model is, well, train it on the data and test it and see how well it does. So again, you want to split your data into, well, actually, let me back up a second. Let's say you have, we have our 800 patients. Let's say we trained our model on all 800 patients. well, how are we going to test to see if our model did well, right? Do we have to go back out and get more patients? Well, we can't, right? Because the Aravel study is done. There aren't more patients that we can choose from. So oftentimes what people are going to do is they'll take the whole amount of patients that they have, all the data, and they'll split it up. And they'll take 80% of that data and use that for training, and 20% of that data, they're holding it out and they want to test at the end to see how well their model does. You can do different splits. 80-20 is the most popular. Another thing that's very common and highly recommended, and I would recommend, is cross-validation. I don't have too much time to get into it. I'll touch upon it very briefly, but it is extremely important. And there's a couple different reasons you might want to use it. So I'll just talk quickly about what it is. So basically, you're taking your training data and you're dividing it into an equal number of parts. This test block here is not this test block here. This test set, you've completely separated out. It's living in la la land. You don't touch it. You don't look at it until you absolutely have your final model and you're ready to test. You are deciding on different models. Certain models might have what's called hyperparameters. So there's different ways to tweak the model. And you might not know which hyperparameter is going to be best, right, on your test set. You don't want to do all your training and then just cross your fingers and hope that the model and the hyperparameters you've chosen are the right ones for your test set. So what you can do is kind of a little bit of cheating, right? It's not actually cheating, but what you're basically saying is I'm going to take all of my training data and I'm going to divide it into, this is 10-fold. So this is 10-fold cross-validation, and each fold is referred to as a K. So K-fold depends on the number of folds. So this is, again, 10-fold. The most common is 5 or 10-fold. Divide it into 10 equal parts, and the first 9 parts you're going to train on, and you're going to use the 10th part as your test set, right? So that's supposed to mimic what you might see in your actual test data. And then you want to you go through the rest of the training data and use this part as your test set, and then the rest is your train, and then this part is your test set, and you go all the way down. And then you're basically going to take the average of the results from the test set, and you're going to look, depending on the performance metrics you're looking at, you want to choose the hyperparameters or the model that had the lowest error, right, or had the highest performance on that test set. And that's what you're going to use when you want to go and test on your actual testing data. And so we're going to use, I won't have time to go into it today, but it is in the code. You can use, again, cross-validation for hyperparameter tuning. So in this, when I was talking about lasso and ridge, it works by assigning a penalty to larger coefficients. But you don't, depending on the problem in the model, you're not sure what the value of the shrinkage penalty could be. It ranges anywhere from, I think, 0.1 to, it could go infinitely high, but usually it's like 0.1 to maybe 1, 10, 100. So you might want to try different values of this shrinkage penalty, of this lambda, and whichever value gives you the best performance, that's the value of this shrinkage penalty that you're going to use on your test set. And so we're going to do cross-validation. You'll see that in the code. We won't have time to, again, talk through it today, but the code is there if you want to run it. But that's the general idea. Okay. The next thing we want to do is we want to measure our performance of our model. How do we know whether or not it's doing a good job, right? There's many different metrics that we can use. The metrics we're going to be looking at today are called precision and recall and area under the receiver operating characteristic curve. That sounds like a lot of words. I will walk you through exactly what it means. Okay, so let's think back to our examples. We have our patients. We've labeled them. We know which patients have a high frailty index and a low frailty index, right? So we have our actual values for each patient, whether or not it's a 0 and there's a 1. So this is going to be on our test set. So we're going to take our test set. We're going to say, okay, model, you've trained, you understand the relationship, we hope, between the variables and the outcome. Tell us, we're going to give you new data. Tell us if whether or not you think this patient should have a high frailty index or a low frailty index. So if, let's say we have patient 1 and the patient actually does have a high frailty index, and the model also predicts that the patient has a high frailty index, well, that's a true positive, right? It was actually true, and the model predicted to be true. Similar, that's what the TP stands for. TN is true negative. The patient actually had a low frailty index, and the model also predicted that the patient would have a low frailty index. So really, and I should explain that this whole thing right here is called a confusion matrix. it is very aptly named. When I first saw this many moons ago, I was like, what is going on here? But I'm going to walk you through it. It is pretty easy to understand once you understand the different components of what's happening. And we're going to output confusion matrices for all of our models, so we're going to understand how well it's doing. But again, you want most of your patients to either be classified as true negative or true positive. The problems start to come up is when the model is, the patient is actually labeled as having a high frailty index, but the model predicts them as a low frailty index patient. That's a false negative. The opposite is true for false positive patients. So, okay, so what does that kind of look like? What does that mean? Well, let me just, I'm going to hide this column for a second right here. So I just want to let's look at some examples of what we might get out from a confusion matrix. So again, let's say that the model predicted 1, but the patient was actually, I changed this originally because I had changed the probability. Let me just do this for a second. It's actually a true negative, right? The model predicted a 0. It was actually a 0, it's true negative. And then we can go down the line and say, okay, this is true positive, true negative. This is actually supposed to be 0 as well, okay? And then we can tally all of these up. And here we'll see that we have 3 true negatives, 3 true positives, and 1 each of false positives and false negatives, okay? So this is what our confusion matrix is gonna look like. So there's one thing that I forgot to mention, well, not forgot to mention, but I'm gonna explain to you is that the model actually isn't giving us a 1 or a 0 as its output. What the logistic regression model is doing, and this is slightly different from how the tree-based methods do it, and I'll explain that later. What the model is actually doing is it's actually just giving us a probability for the positive class. So it's outputting a probability between 0 and 1 as to whether or not that patient has a high frailty index. So if it outputs a probability of 0.9, saying there's 90% probability that this patient should be classified as having a high frailty index. Okay, well, what do we do with that data, right? So let's look at some examples. And again, I've just made this up as a sort of toy example here. But let's say this model is saying the probability is, this is what is actually being outputted. Probability of this patient is 0.25, 0.89, 0.65. So we have to ask ourselves, okay, well, what if a patient has a probability of 0.5, right? Does that mean that the patient should have a high frailty index or a low frailty index? And so this is what's called threshold tuning or threshold moving, where the default threshold is 0.5. So when you run the model out of the box, you set the threshold at 0.5. So anything 0.5 and above, the model will classify as a 1. Anything 0.5 and below, the model will classify as a 0. And it's going from those raw probabilities. And I want to just pause here. Is really important to understand. This threshold that we are setting here is not, I repeat, is not the same threshold that we have set when we originally classified our classes in 0 or 1. It's not this same cutoff, right? The model has no concept of what this cutoff is. It has really no concept or caring about the 0 or 1 or what they mean, essentially, right? We have classified the patients this way. The model is basically just saying, I understand that these patients are in the positive class, and I want to predict their probability of ending up in the positive class. And this threshold is a threshold for the probability. It's not the threshold for high or low frailty. It's an important distinction to understand. And so what you can do is you can move this threshold up or down. And let's say, so we go back here and let's say the threshold was 0.5, these are outcomes, but let's say we change the threshold to 0.3, right? So anything 0.3 and above, we're actually saying is going to be classified as a 1. So now this is going to stay a 0, right? But now this is actually going to be changed to a 1. So this actually, from going from false negative, has actually now become a true positive, right? But similarly, this 0.3 is actually going from a 0, it's not going to be a 1. So this is now what? A false positive. So that's actually going to change the numbers in our confusion matrix. And that's going to change the performance metrics of our model. How is it going to change the performance metrics? Let me show you. The way that we calculate performance is based on, again, this confusion matrix. So the most popular and common metrics are something called precision and recall. And recall is also known as sensitivity. And specificity is also extremely common. And here what I'm showing you is literally how we calculate precision and recall. So precision, it's looking at of all the things that were labeled as 1, of all the possible values that were labeled as 1 by the model, right? All the true positives and all the false positives. How many did it actually get correct? So how many true positives out of all the possible things that it labeled positive, right? Recall, on the other hand, is looking at of all the patients that were actually truly positive, how many did the model actually label as positive, right? So precision is more understanding your false positive rate, and recall is more really understanding your false negative rate. So let's look at an example here. Here we have 12 true negatives and 9 true positives, 11 false positives, and 1 false negative. Well, we can calculate our precision. We can say true positives plus false positives, 9 plus 11, and then divide 9 divided by 9 plus 11, right? So 9 out of 20. It's a precision of 0.45. That's not great, right? We want our precision and our recall numbers to be high, right? We want our false positives and false negative numbers to be low. And if these numbers are low, as you can see, because they're in the numerator, then you're going to get a resulting higher numbers for precision and recall. Similarly, we can see here, we don't have a lot of false negatives, so our recall is actually quite high. It's 0.9. F1 is, it's called the harmonic mean. You don't really have to worry about what that means, but it's basically somewhere in between precision and recall. So if you have a value of your recall that's very high and your precision that's very low, you're going to get a number that's in between, somewhere in between these two numbers. Again, you can also calculate specificity. It's again about 0.52 because we have a high number of false positives. So these are the metrics that we're going to be looking at, mostly precision and recall, to understand, again, how well our model is doing. Because again, it's going to give us an understanding of, is this model classifying more false positives or false negatives, et cetera, et cetera. And again, another common metric to look at is OROC, which is area under the receiver operating characteristic curve. Again, this looks at our true positive rate, and I explained this in one of the Discord questions, and our false positive rate, which is just 1 minus specificity. But basically, if you have a perfect classifier, it's going to be perfectly classifying all your samples as true positives, and you're going to have no false positives. And as you start to move away from this area, you're going to have a worse model. Basically, this model is calculated by it's looking at every single threshold value, and it's plotting the confusion matrix for each threshold value, and it's calculating your true positive and false positive rate at each threshold. So if you have a model that does better at the majority of thresholds, then you're going to have a higher area under this whole curve. Okay, so an OROC of 1 means that your model is perfect. Again, be suspicious of that. And if you have an OROC of 0.9, again, it's operating about 0.9% of this space. It's doing very, very well, actually. Precision recall curve is basically, instead of using true positive and false positive rate, it looks at precision recall. And I made a note here that it's a little bit better when you have imbalanced data sets. We have an imbalanced data set, although we're going to rebalance it. I'll talk a little bit about that. But because it's looking more at false positives, again, it's looking more at the false positive rate. It's not really affected by the true negative rate. Okay, that was a lot of talking. We are now going to get to the exciting part, which is actually running the code and looking at the outcome and output from our model. Okay, so I want everyone to move over to the Machine Learning Logistic Regression tab or notebook. And again, you can just either running by hitting this play button here or hitting shift, holding down shift and hitting enter. This is just to load all the libraries for everything we need. This is a function to visually output our confusion matrix. There is a way to output a confusion matrix using scikit-learn, but it wasn't working with AWS, so we just made this function instead. Okay, here's our data. We've already loaded it. These are all the columns. We've talked about that. Again, this is just a nice way to visualize the data. Here we can see we have our merge-fi class as it originally was. This is our frailty index. We have our value for each one of our patients. And then here you can see we are again converting that into a binary outcome of 0 or 1. And then the next thing we're going to do is we're going to, let me just show this real quick. So here now we can see that we've created this new column, and our outcomes are now classified to 0 and 1. And then we've dropped this column because we don't want to include it, right? Because it's directly derived from this column. We only want to use this column to predict, and we don't want to use the column that we use to create this one, right? Because it would be highly correlated and wouldn't actually tell us anything. We already know that they're related. So that's why we're dropping this column here. Then we want to define our categorical and our numerical columns. Well, what does that mean? Well, you're going to treat numerical data differently than you're going to treat categorical data, right? So categorical data might be something like sex, right? And here we've already reclassified. We only had male and female patients. We've reclassified that as, I believe, 0 for male and 1 for female. The reason we're doing that is because machine learning models have no concept of, in this case, M and F. They have no idea what that means. They can only deal with numbers. They can't deal with letters or strings, right? So we have to convert this into a language that the model is going to understand. And then let's say you have a variable like race, and Lance talked a little bit about that briefly, where you have multiple categories of patients. And here what we've done is, and again, this was code that was done previously, is we've done what's called one hot encoded. And so we actually created a column for each one of the races. And if a patient, for a given patient, they're only going to have a 0 or 1. The true is 1 and the false is 0. So this patient is going to have a 1 in this column and a 0 in the rest of the columns, right? Alternatively, let's say if this patient identified as East Asian, there would be a 1 here and the rest of these columns would be 0. Okay. And so the reason that we separate out our categorical and numerical features is because if we're missing values in these columns, we're going to treat that missingness differently. I'll talk a little bit about that in a second. And so what I would actually encourage people to do is you can choose whichever features you want to include from the list that we have here. We're including all of the features, but maybe you want to run a model, run a model with like 5 features. So you can do that. Determine whether or not the feature is numerical or categorical, and then you can put it in a list here. Because we're using all the features, I basically just defined the categorical ones and dropped all of the categorical ones, and the rest that were remaining are numerical. But you don't have to do that. You can just create a list right here of the features that you're interested in and run that model. Okay. So then what we're doing here is we're defining our outcome, right? This is our merge FI class. This is our 0 or 1 features. And then that's our outcome. And then the rest of these we're going to use as the variables that we want to include in our model. And we're going to drop this column from obviously the variables that we want to include. This is just to print out to make sure it's a little bit of sanity check to make sure we're using the right feature. So here we know there's only 0s and 1s. And next, we're going to do the train test split that I talked to you guys about. And if people want to read more about it, there's a website here from the scikit-learn module that explains the train test split. Basically, we're choosing 80-20. So all we have to do is say our test set, we want it to be 20%. It'll automatically understand that the training set will then be 80%. And again, we're using our X and our Y that we defined up here. And we're just splitting it into our train, X train, X test, Y train, and Y test. I'll explain what those mean in a second. One thing I want to point out is that we're also stratifying the data. This does not equal yes. This is Y. This is our outcome. What does stratifying mean? Well, basically, if you look and you can see that our classes are imbalanced, right? So if we're randomly dividing the data into 80 and 20%, you might inadvertently choose an entire training set that only has just patients in the negative class, and you have no outcomes that you're trying to predict. So stratifying by your outcome means that this proportion of 3 to 1 is going to be maintained across your training and testing data. And that's really important. Oops, sorry, I'm getting ahead of myself here. Okay, so that's what's happening up here. And now this is, in order to deal with the fact that we have imbalanced data, you can run the model with that imbalance. That's perfectly fine. You can see what results you get. But a lot of times people like to balance out the classes so they have an equal number of positive patients in the positive class and the negative class. Well, how do we do that if we don't have more patients? Well, what we can do is you can randomly oversample the minority class, which in our case is the positive class. So what we're going to basically do is we're going to take we're going to randomly select with replacement. So we can go back and take the same patient over and over again. And we're going to choose random selection of these patients until we have an equal number of patients in our positive class and our negative class, okay? Now, again, we're only doing this on the training data. We don't want to do it on the testing data because we don't want any of that data to leak into our training. So we're only doing it on the training data, okay? So these are our new overly sampled X's and Y's. So this is the data we're going to train on. This is our 80% of our data that has our features. This Y is basically just all the answers. I'll show you what that looks like in a second. And then this is the set that we're going to predict on. This is our X test, okay? And now once we've oversampled the data, we can see that the ratio to our positive and negative class is now 1, right? So it's gone from 3 to 1 to 1. So there's an equal number of patients in each class. Now we want to impute, right? If we have any missing values in our data, we want to impute that missing value. And Lance talked a little bit about this. He did a great job. Again, you want to have different strategies for if your data is categorical, if it's either a 0 or a 1. Usually if that data is missing, you want to replace it usually with a 0. You want to assume that, okay, if the patient, we don't have medication data for this patient, it's probably because they weren't on that medication, we can assign it with a 0. Versus assigning it as a 1 has a lot more implications. But again, always make sure you understand which decision you're making at each step and why. When we have numerical data, there's many different strategies that you can take to replace that data. The mean is a very popular one. You can do random forest imputation. That takes usually a lot more accurate, but it takes a long time. You can impute with the median. You can impute with the most common number. There's many different strategies. And you can change this, and you can look at the documentation here for what different strategies that you could use, and I would encourage you to try different ones out. For simplicity, we're just imputing with the mean, but again, try different things. The last thing we're going to do is we're going to scale our variables. What does that mean? Well, let's say you have a value of triglycerides that go from 176 to, I don't know, 40, 50, and then you have potassium, which is at 4. The model is going to treat these bigger numbers as more important, right? But it's because they're on different scales. They have different units. And so what you want to do is you want to make sure that all of the units, all of the features are treated equally to start, right? So they're being put on the same scale. How do we do that? Well, you want all of the values for that column to be between a 0 and a 1. So the model understands that up front, all of the columns should be treated equally, right? To start, and then the model's going to understand which ones are more important. But basically, there's a couple different ways you can do this. We're going to use min-max scaling, which basically for each column, the imputer that we're using is going to look at the minimum value and the maximum value for that column. So let's say the minimum value for triglycerides was 63 and the max was 176. It's going to assign this a 0 and this a 1. And then it's going to scale the rest of the values to be between that 0 and 1 for each column. So the proportion and relationship of that column is still maintained, right? But again, it's still between that 0 and 1. Okay. So the next thing we're going to do is we're going to transfit the data. So by fitting, we mean we're going to replace replace missing values and scale it. And then we're going to transform the data and get a new version of that data with these transformed values. Important thing to keep in mind is that we're going to actually only only fit the data using the information from the training set. Well, what does that mean? It means I only want to use the mean of the columns and the min-max of the columns of the data from the training set. I don't want to use it from the testing set, or I don't want to take all the data initially and do my scaling. Why? Because I don't want to have any information from the testing set. If I have information from the testing set, then that's helping the model sort of cheat and understand a little bit more about the relationships in that data. And I don't want to do that. I only want the model to learn from the training data. And so I'm only going to fit on the training data and I'm going to transform both, right? I still need to convert the values from my patients to, again, be scaled and to have that mean value, but I'm going to use the values from my training data. And so these are our new these are our new data frames, our new training set and our new testing set. And here I've just converted, these are NumPy arrays, and so you can't really visualize them, so I've just converted them to pandas data frames. So here now we can see our new training set, right? All of the values are between 0 and 1, and there should be no missing values. That's what it's going to look like, right? And then this is our Y training set. What does that mean? These are just, you can consider them the answers to the test. So we're going to feed the model this training data. We're going to say, these are all the features, figure out what's important, and these are all the answers, and try to understand the relationship between these two. Then once it's done, we're going to give it testing data, and we're going to say, okay, here are our new patients, give us your predictions. And then we're going to compare it to the Y test predictions, and we're going to use those to get our performance metrics. Okay, so exciting. Now we can actually get to the modeling part. So here what we're doing, what's kind of amazing is that after all of this, the actual running of the logistic regression model code is just basically 4 lines, where it's really, well, actually just 2 lines. You call the model, and then you fit the model to your training data. And then these are going to give you your, these are the probabilities that we talked about, and these are the actual predictions that we've converted to 0 or 1. And then we're going to use these values to get, again, our accuracy, our precision, our recall. And when I say we're using the model out of the box, again, there are other hyperparameters that you can tune for logistic regression. It's more so concerned with regularization with lasso and ridge. And so there's a lot more that you can do, but just for simplicity's sake, we're just running things sort of out of the box and seeing what we can get. Okay, so what do we get? Well, first we can see the precision of our model it's not amazing, right? It's 0.53. The recall is pretty good. The F1 score is in between both of them, 0.64. But our precision recall AUC in our OROC or ROC AUCs is pretty good, right? 0.88 is nothing to spit at. That's actually not bad for just out of the box, first time, not changing, not adjusting anything. It's not bad. So let's take a look at our precision recall curve. And this is actually all in the presentation as well for people who are interested. I'll be switching back and forth between the presentation and this right here. This is our precision recall curve. So again, the area under that curve is pretty high, it's 0.81. And then here's our confusion matrix. And now we can actually interpret and understand what this means, right? So we have a high number of true negatives. The model's doing pretty well with 31 true positives. And again, this is 154 patients in our test set. So this is actual, these numbers refer to actual patients, 89 patients that were correctly classified, plus 31 patients that were also correctly classified. But here we can see we have a high number of false positives, right? And that's why we have a pretty low precision score of 0.53. So now we understand what this means, right? So now, well, let's do lasso, right? And so again, lasso can be used to reduce the number of features in our model, but it can, and as a result, it usually almost always results in an increase in performance. And so what people will do is they'll run lasso and ridge, and even if it doesn't remove features from your model, it still just helps again to increase performance. And so people will usually run lasso and ridge as well. And again, the code is almost identical. It's the same logistic regression model, but it's called an L1 penalty, and ridge is the L2 penalty. Don't worry about what the solver is. You can read more about that if you're interested. But let's look at the results from lasso and ridge. We can go into our slides here. And here we can see is lasso on the left and ridge on the right. That ridge actually is doing a slightly better job. Our ROC is 0.88, precision slightly higher, recall slightly higher. Again, it's not doing like amazingly better, but it's still doing slightly better, right? And what we can actually also do is we can look at the features in these models that have the highest coefficients, the highest values, and that's indicative of which features this model thinks is the most important. And we'll talk a little bit about feature importance shortly. But here you can see that lasso and ridge actually both think that insulin is extremely important in predicting the outcome. Calcium, vitamin D, HOMO-IR, and we'll compare this to the tree-based model that we're going to run shortly. And it's interesting to run different models and see how the feature importance changes between them. Okay, and now here we can look at our confusion matrices. Again, the differences in performance are slight because we're really only classifying one more patient as true positive. So we're not like blowing anyone's mind here. But again, it's important to understand what,
how we come to these conclusions. Okay, so now let's look at a tree-based method. So now we're going to do a different type of method from logistic regression. And even though decision trees seem complicated, what underlies them is simple for people to understand. And it's a lot of ways that people make decisions, and especially in medicine, the way that doctors make a decision. So everyone has probably done some sort of like quiz, in People magazine in your life, where you say, okay, which celebrity makeup style is yours, and it'll ask you like, okay, what's your age and what's your favorite color and what's your favorite makeup? And then you'll go down the tree, and then it'll say, okay, you're like a Maybelline girl or whatever, right? That's essentially what's happening in decision trees. Is you start with a root node, right? You consider a question that you're asking, and it's going to classify based on, and it can do either numbers or categories, right? It can handle either type of data. But say, okay, let's say the patient is, and I'm not entirely sure. I don't think this is super accurate. I just pulled this from online. It's just a visual to help you understand. But let's say you have a patient that's over the age of 30 and they're a smoker. Well, they're probably maybe at higher risk of heart attack, right? And so we're going to have something similar in the models that we're creating. Is for each one of our features, the model is going to create these different nodes, and it's going to divide it up, and it's going to say, okay, patients under this age, we're putting on the left-hand side, patients under this age, we're going to the right-hand side, and then it might choose sex, and then it might choose vitamin D value, and it's going to choose each one of the variables and understand how choosing that specific combination of features is going to determine whether or not a patient is going to be highest or lowest risk. And so it wants to categorize as many patients as possible correctly at the end. And so it's going to choose many different combinations of these nodes to basically maximize correctly categorizing the number of patients. And so this is again, a tree-based method. There's different types of methods. Random forest, which uses bagging. We're going to look at a boosting model today. It's called extreme gradient boosting. We don't have to worry about the details of boosting, but essentially the more advanced tree-based methods like random forest and boosting, they use ensembles of trees. So they're not just using one tree, they're using many, many, many different combinations of trees. The ways that those trees are built is different from random forest and XGBoost. Don't have to worry about that. But just understand that we're going to take the information from many different trees that use different combinations of these features to then predict. It's going to come up with sort of, it's either going to have an ensemble of trees, and it's going to run the patient down all of those trees, and whichever, let's say 50 of the trees say it should be categorized as high frailty and 49 of the trees say low frailty, then it's going to categorize that patient as high frailty. Or you can end up with sort of one tree that has been built over many different iterations, and you can run that patient down the tree, and the end result of high or low risk will be what the tree outputs. So that's essentially what we're doing here. Okay, so again, the code to run the XGBoost model. Oh, and I want to point out, actually, when I talked earlier about cross-fold validation and picking the right parameter for something like lasso, this is the code here, where we're basically finding the optimal value of lambda. And then we can actually see the values of those coefficients that the optimal model has chosen. So I won't have time to go through that right now, but again, you can do cross-fold validation. It's built into the scikit-learn models. And it's something I would encourage you to run and try out. Okay, so then we can look at, this is the lasso code again, and ridge code again, very similar. It's confusion matrices. Again, so to run the XGBoost code, again, it's really simple. You call the XGBoost library, and we're doing a classifier. You could do XGBoost regressor if you have a regressor problem. And again, we're fitting to our training data, and then we're telling the model, hey, this is our test set. Give us the predictions for your test set. And we're going to use the same performance metrics. Okay, so what do we get? Well, the model does a little bit better, right? So even though, we're getting a precision recall AUC of 0.82, and the precision is actually higher, it's 0.63. Our recall is a little bit lower, right? But overall, if we look at the confusion matrix, it actually looks like the model is doing better, right? We're having a lot higher number of true negatives. And even though we're losing some true positives, we're having a significant drop in our number of false positives, right? And that's why our precision score is a little bit higher. But again, we have a higher number of false negatives. And one thing I should mention actually is I really want people to understand which threshold you choose, which model you choose, again, is really going to depend on the problem that you're trying to solve, right? So you may have a problem that you're trying to solve that you could absolutely need to have as few numbers of false negatives as possible, right? So you might want to move that threshold to be such that it really excludes a lot of, it doesn't correctly, excuse me, it doesn't misclassify a lot of patients as false negatives. You might get a higher number of false positives, but that's okay. What's an example? Let's say you are trying to predict something we're all familiar with, like patients who might be contagious for a highly infectious disease, right? And you want to develop a model or a test to predict whether or not this patient is contagious. It's really important that your model, if we look back here, that your model is correctly classifying patients as, obviously, of course, a 1 or a 0. But if it misses, if your patient actually is highly contagious and the model is predicting a 0 and you just let that patient walk off, it has extreme consequences, right? Whereas if the patient isn't actually contagious, but the model thinks the patient is, of course, that's not great for the patient. Maybe they have to be quarantined, et cetera. But the implications of that are much less severe than missing this patient. So again, you might want to move that threshold such that you are not missing, you're missing very, very few false negative patients. So you're not incorrectly classifying these patients. Of course, there's always going to be a trade-off, but you're willing to accept that trade-off and you're willing to have a much lower precision at the expense of making sure that your recall is sufficiently high so you can capture all those patients. So again, always think about the problem at hand. Okay, so I have a little bit of time left. I'm going to talk again, I'm going to talk about, now that we have all these models, let's say we've picked our model. We're going to pick our XGBoost model. We think it's performing pretty well. And we don't just want to understand why, we just want to understand which patient is going to have a higher low frailty index, right? That's great. But we also want to understand why, right? What are the features that are contributing to a patient having a high or low frailty index, right? And in our case, we really want to understand the mechanistic pathways that might be driving frailty. And again, the machine learning model is not going to tell us exactly if you have this, that means this outcome, right? Machine learning model is just a representation of some part of the relationship between that feature and that outcome. Especially when you're, again, dealing with complex systems, you're not going to be able to include every single feature. And so again, the results that we're going to see are a launching point for us to go back into the lab, to go back into the literature to see if we can better understand and tease out the true nature of that relationship. But machine learning is a great way to start to dig into some of those answers and those relationships and truly get a better understanding of what might be happening. So if you have different models, so again, we can look at our logistic regression model or XGBoost model. The way that these models calculate whether or not a feature is important is different depending on that model. We won't get into how each model calculates feature importance, but you want to kind of say, well, okay, maybe my lasso model and my XGBoost model are kind of identical. Do I go with the features from the lasso model? Do I go with features from the XGBoost model? Do I go with features from the Ridge model? They're calculated in different ways. How do I compare them? And so what you can do is you can have a method that we're going to use today that can actually use the same approach regardless of the model to tell you which features are the most important. And today we're just going to look at the features from our XGBoost model. And the method we're going to use today is called SHAP. And I have the slides to explain SHAP. I'm not going to go deep into the details of it, but for those people that are interested, you can read about it. And I put a little bit of a blurb in the speaker notes if you want to read more about it. But basically, SHAP is based on Shapley values from game theory. And if those words mean absolutely nothing to you, don't worry. You can think of it in this way, right? Let's say you have 5 people who are going to the airport and they're all taking a cab together. And when they get to the airport, the cab costs $100. how much should each person pay towards that cab ride, right? Let's say Joe lives 100 miles away and Sue lives 2 miles away. Well, Joe should probably pay more, right? Because he's responsible for more of the ride than Sue was, right? Joe wants to be a jerk. He can say everyone should be divided equally, but like, don't be that guy, Joe. You know what I mean? Pay your share of the cab fare. And that's basically what SHAP is doing. It wants to understand how much each feature contributes to the overall outcome. So those features that contribute more are going to be more important. Okay. And I will show you the output of the plots that we have. And I will explain to you how SHAP is understanding feature importance. Now, before I show you the results, I'm going to just show you how to interpret these plots, because when you look at them first, they're a little bit visually overwhelming. And we can also just go through the code real quick for people that want to see. It's really simple. You should import SHAP at the beginning if you're doing this somewhere else. AWS seems to already have this capability, which is amazing. But basically what you're going to do is you're going to call the SHAP, and we're explaining our XGBoost model. So we're going to use a tree explainer. And then the SHAP values that we're going to get are going to be run from this explainer model. Okay. And then we're going to use a summary plot to get the output. And so before I show you the summary plot, I want to just show you how to interpret the plot. And again, we're going to use the example just to help people understand of which features contribute most to helping your team win a game. So let's say we're talking about a soccer game. And again, these are the features that we've included in our model: total shots on target, home away. I actually don't know what half of this stuff is. I took this from online, but you understand the basic concept, right? So these are all the features. And then we're going to get something that looks like this. Okay. And each dot is actually going to represent a specific data point. So in our case, each dot is actually going to represent an actual patient. And so here, first, you want to look at this SHAP value. That's where extracting is the value, how much this feature is contributing to the outcome for each feature. And so you can see this gray line down the middle here, and everything to the right of that gray line is going to have more of an impact on the outcome, right? So you can consider it that all of the features, the feature values, and I'll explain what that means in a second, to the right of the line contribute more. Like positive SHAP values contribute more to the team winning. Well, negative SHAP values are indicative of losing. Okay, I'll explain that again in a second if it's not clear to people. The next thing that you want to look at is the actual value of that feature itself. So here you can see this color bar. Blue is low feature value and red is high. So let's take this feature, total shots on target, from attacking penalty box, which I assume just means the total shots that are taken right in front of the goal, essentially. And so let's say this feature itself ranged from 0 shots to like 100 shots. So blue is going to be 0, red is going to be 100, and the values in between that are going to be, 30, 40, 50, 60. Okay. So the way to interpret this is that having a higher number of total shots on target is more indicative of your team winning the game. And that makes sense, right? So that's how you're going to interpret it. Each feature, you want to understand the direction. Like, blue is always going to mean low and red is always going to mean high. What does that mean for that particular feature? And all those values that are on the right-hand side are contribute more to the team winning or to your outcome, and the left-hand side is not towards the outcome. And the last thing to keep in mind is that the order of features are indicative of importance. So what this plot is saying is that this feature is the number one most important feature in determining whether or not your team is going to win. And then this is the second most, third most, fourth most. One thing I want to keep in mind is that we are talking here about a very simple, not necessarily simple, but it's easy to understand how total shots on target is going to contribute to your team winning. When we're talking about, again, systems biology, biological mechanisms, just because you have a feature that's extremely important, it doesn't mean that that is the reason why someone has a high frailty index, okay? Again, our machine learning model, we're just using it to better help us understand the features that might be important, but again, we might be missing other things that aren't included in there. So we always have to take it with a little bit of a grain of salt. Okay, let's look at the actual results because that's all I ever want to see, right? Okay, so this is the SHAP plot that we see, okay? And here, well, what do we see? Looks like homo-IR, insulin resistance. The model is saying that having a high insulin resistance, higher value for insulin resistance, is more likely to contribute to someone having a high frailty index. And here you can see that having a low, all these patients have a low value for the homo-IR. You can kind of see a little bit, it's kind of difficult, that there are some patients that do have a blue dots, which means they do have a low value for the homo-IR, but they actually ended up also having a high frailty index. So this alone is not, you're never gonna have one feature that's gonna explain it. The next most important feature is the proteomics, the brown cluster from the proteomics, which is also what Lance saw. And so when you have different results that are being replicated in different ways, by different methods, by different people, and you're getting similar answers, that's also reassuring, right? It's tends to point towards something that's important. And so if I remember correctly, a lot of the markers in this brown cluster were for inflammatory response, right? Response to interleukin-1, T cell leukocyte differentiation. So it's saying having higher values for those features is more likely to contribute to a higher frailty index, right? And again, that makes sense. And so you can just go down the plot and continue to interpret the plot. And so I asked some of the aging experts, I'm not an aging expert myself, asked some of the aging experts from our group if they had some insight into some of these results. And they said, well, this makes a little bit of sense, right? There's metabolic features that are associated with frailty, so homo-IR, HbA1c, triglycerides, which is also very important, that are known to be associated with frailty. And there's a historical view of frailty as someone having a low body weight. You think of like a very frail old person, but is growing evidence of a population of obese frail patients, so patients that have a high frailty index, but they also have some sort of metabolic function or disease. And that might be what we're seeing here. Right? We also have patients shown that low levels of vitamin D are also associated with frailty. Here you can see this is a low value for vitamin D is associated with the higher frailty index. But however, you can say, okay, we'll just give people vitamin D supplements that'll lower their risk of frailty. There's mixed results in these studies, right? So again, it's never that simple, right? Also, low levels of calcium, right, is linked to vitamin D insufficiency, and low levels of potassium. They have a loss of muscle mass and frailty. And for people who are interested, I have dropped the links to these papers in the slide notes for people who want to read more about this. And of course, you can ask some of the aging experts for further insight into this. So again, this is what the machine learning model is telling us. So again, in the last few minutes, I just want to touch upon different ways that we could improve our models. And again, I've talked a lot about this, but just to reiterate, again, you can change, we reduce the class imbalance, but you can go back and you can take out that class imbalance and you can run it with the original 3-to-1 imbalance and see what results you get, see how it changes the model, and ask yourself, why does reducing the class imbalance help improve the model? Some homework for you. Hyperparameter tuning, that's what I talked about previously. Actually, if you'll see in, actually, if you go beyond the SHAP plot code, yeah, so sometimes these SHAP plots don't run very well in different environments. So these SHAP plots actually shouldn't look like this. So if you're getting these results, don't worry about it. We can just skip over those. That's why I didn't go into them. Again, just to touch briefly upon hyperparameters. Again, like for tree-based methods, you can choose different numbers of trees, how deep the tree grows, many different things. And you might want to run through different parameters and pick the one with the best parameters that you want to use on your test set. And so here I just did a very simple set of hyperparameters along with cross-validation. And here we can look, oh, this didn't actually end up running. Well, you run both of these cells. This is going to take a little bit of time, again, because it has to run through many different iterations of many different values. And you can output, you can look at the confusion matrix from that model. And I believe it actually did the best so far. And there's another, you can also manually adjust the thresholds. So you can do, again, threshold tuning. And here I just made a little bit of code for you if you want to run it with the lasso model, and you can choose a different value for your threshold. Let's say you want to choose 0.9, and you can see how that's going to change these different metrics. What machine learning researchers, data scientists will do is they'll usually just take all the different thresholds from like 0.1 to 0.9. They'll loop through them, and then they'll pick that threshold that has the highest value for the particular metric they're interested in. But I thought it'd be cool if people wanted to just play around and see how changing the threshold will affect your metrics. Again, we can always change our outcome to have a different outcome. And I encourage people to change that cutoff and see how it's going to change the model outcome. You could always gather more data. You can, my God, you can never have enough data. if you can always add more data and gather more data, please do so. Again, feature engineering I talked about before, and again, trying many other models, more complex models, if you're interested. So that's it. I think I'm pretty much at time. Thank you so much. If there's any questions, I will address them in the Discord and in Zoom. I hope this has been useful to you, and I wish you best of luck on your data science and machine learning journeys in the future. Thank you.
Welcome to the seminar part of the symposium, which will feature talks from Dr. Jesse Poganik from Harvard Medical School and Dr. Cara Green from University of Wisconsin-Madison. Time for Q&A will be right after the two talks, so please make sure to deposit your questions in Zoom using the Q&A button at the bottom of the screen, and please note which of the speakers the question is intended for. You can also upvote other questions. Each talk will be 20 minutes, and after both talks, we will have about 20 minutes for questions to both speakers. It's my pleasure to introduce our first speaker, Dr. Jesse Poganik. Dr. Poganik is an instructor in medicine at Brigham and Women's Hospital and Harvard Medical School. His research focuses on understanding the most fundamental aspects of aging and leveraging this knowledge to identify the most efficient ways to extend health lifespan. He leads several projects aimed at understanding the temporal dynamics of biological age, the intercellular and intertissue dynamics of aging, and how aging may best be quantitatively measured. Dr. Poganik also serves on the Executive Committee of Biomarkers of Aging Consortium, and he will talk to us today about biomarkers of aging and the dynamic nature of biological age. Dr. Poganik, it's all yours.
Okay, thank you very much. Hopefully everyone sees the slides. Thank you for the invitation. Thank you for putting on this super useful workshop, and greetings from Lisbon, Portugal, where it's currently 11 p.m. Thank you for the introduction. I'm Jesse Poganik. I won't repeat all of that, but I want to tell you today about biomarkers of aging and insights that we've gained from these biomarkers into the nature of biological age. And everything that we do, or everything that I do, centers around this central question, which is, what is aging? And I think this is really the most interesting scientific question. But it's also not something that we're going to solve in a 20-minute talk. So what we'll do now, and what we do in our research, is to take a step back and we ask another question, which is, what is the nature of biological age? And the distinction here is important. So it's worth spending a minute on. Aging is the process that happens over the life course. Biological age is a parameter that we use to understand something about aging. And so we study features of this parameter, biological age, and I'll tell you about two stories today, one about the temporal dynamics of biological age, and one about how biological age dynamics work between tissues. And the fundamental idea is that if we understand these features better, we may be able to find better ways to target aging and ultimately improve healthy lifespan, which I think is the kind of universal goal of this field. This temporal story starts with this hypothesis that biological age is not static. Every time I give this talk, I think I need to convince people less and less, because we know now that biological age can be accelerated by things like stress and disease. We know from experimental models and even some clinical trials now that it can be reduced by various drugs and interventions and experimental systems. But we wanted to flip the script a bit and ask, could there be situations where biological age can be increased transiently and then relax back to the baseline after a period of recovery? And to ask this type of question, you need to have tools to measure aging, and that's where the biomarkers of aging come in. So these are really revolutionary tools because they allow you for the first time to quantify aging in a way that's really informed by systems biology. So I think they're very relevant to what we're talking about today. And I have here listed just a small selection of DNA methylation-based biomarkers of aging. And this is a selection. There are many more out there. And there are many more beyond DNA methylation. More and more come out. So more on the biomarkers in detail later, but the most important thing to understand is that the use of these biomarkers is that they allow you to measure something that you could consider normal aging if the prediction is in line with what you would expect, or accelerated or decelerated aging, and to extract a parameter that we call age deviation, which is the chronological age adjusted biomarker prediction of biological age. And so with these tools in mind, we turn to one of the most classical models in aging biology, which is called heterochronic parabiosis, which is a system where you have two mice, one old and one young, and you surgically attach them so that the circulation is shared between the two mice. Uniquely in our study, which we did in collaboration with Jim White's lab, we separated the mice after 3 months of parabiosis and we let them recover for 2 months. As a control, you can use 2 age-matched young mice and repeat the same experiment. And then we applied our biomarkers of aging to the liver tissue from these mice. So somewhat unsurprisingly, because this model has been around for a while, when we compared heterochronic mice, young mice that were exposed to old circulation, we saw an increase in the biological age read out at the level here of DNA methylation. But what was surprising to us was after detachment and recovery, the biological age recovered back to baseline. And this was true if we looked at it from the perspective of a clock that was trained specifically for the liver. It was true if we looked at it with a clock that was based on multiple tissues called a pan-tissue clock. It was also true if we used an orthogonal method of DNA methylation profiling using sequencing instead of microarrays. Consistent results across the board. And even beyond DNA methylation, we observed consistent results. For instance, at the level of gene expression, using signatures of aging and longevity developed by Dr. Alex Tyszkowski in our lab, you can see that if you compare the signatures of aging in the liver or the whole mouse organism, they align very nicely with the signatures of heterochronic parabiosis, or I should say the effects of heterochronic parabiosis, and they're almost perfectly reversed after the recovery. So again, showing that you have this transient acceleration that can be reversed. It's also true at the level of metabolites. So in collaboration with Clary Klish's lab at the Broad Institute, we looked at a set of aging-associated metabolites, and we compared their behavior during aging with their behavior during parabiosis in our experiment. And you can see that during the parabiosis, there was a positive correlation with the behavior during aging and an inverse association with the recovery process. So all of this tells a consistent story that you can have, at least in this experimental system, a transient increase in biological age that can be reversed after a period of recovery. And this is all well and good in a very artificial system like heterochronic parabiosis, but the question is, does this happen in humans? And so we hypothesized that exposure to circulation is not the only thing that goes on with heterochronic parabiosis. There's also a very severe amount of physiological stress because there's a major surgery that goes on, and it turns out that the mice don't like being attached physically for 3 months. So we started looking for data sets taken from people who are undergoing very severe physiological stress. And we started with this publicly available data set from the individuals who had emergency surgery and again before they were discharged from the hospital. And across multiple biomarkers that we examined, we saw something that looked very similar to what we found in parabiosis. Namely that from before surgery to after surgery, there was an increase in the biological age, but after discharge, this was partially or fully reversed. Not equally for all the patients, as you can see, and that's something we need to understand better. But what's very interesting is that in this system, the separation between the first 2 time points is less than 24 hours, and we have this relatively large effect. And then even in the last time point, that's only about 7 days. So this means that the biomarkers and by extension biological age seems to be rather fluctuational. We moved on to examine pregnancy, also very physiologically stressful, and we started this in mice. We got them pregnant, we took blood over time, and we saw, again, a kind of U-shaped curve where before to during pregnancy, there was an increase. Afterwards, after they gave birth, there was a recovery. And in humans, when we wrote this paper in 2013, we were a little frustrated because we couldn't find any human data sets that span the whole period from before pregnancy to postpartum. But nevertheless, looking across multiple data sets, we found that as a function of trimester, biological age increases. I'm just showing representative biomarker, but this was true across multiple ones that we analyzed. And in this single data set, we could find at the time that analyzes from when the people are already pregnant to postpartum, we did observe a significant recovery effect. But what's really exciting is that this year, this was independently replicated, again in cell metabolism by Kieran O'Donnell's group. And using a much more robust data set, they almost perfectly replicated the results that we observed. So that's very exciting. And I saw also Kaylin Ryan in the audience. Shouldn't forget to mention that he has a study that also confirms the age accelerating part of this in a population that hasn't been traditionally studied. So it's very nice to see the results replicated. And finally, we hypothesized that a very severe infectious disease could have a similar effect. So we were doing this in 2021. We got patients at our hospital who had very severe COVID-19, and we profiled them as well. By the time that we could get samples from these patients, their biological age was already increased. You can tell that because for this biomarker, Dunedin POAM, and we got consistent results with the second generation of this biomarker, Dunedin PACE. It was already accelerated. The baseline value for this biomarker is 1, but nevertheless, by the time these people were released, there was a partial reduction in their biological age. This was stronger in females, also present in males, but we found much more heterogeneity in the trajectories of the males. So this is also interesting, this effect of these reversible changes, because it turns out that it may be a good way to rapidly screen potential drugs that target aging pathways. So our COVID-19 cohort was given various experimental protocols because of the time it was collected. These were hydroxychloroquine, remdesivir, and tocilizumab, which is a monoclonal antibody. And you can see that what we did was we used the difference between time points 3 and 4 that we had profiled as a proxy for their biological age recovery rate. And interestingly enough, tocilizumab across the biomarkers that we examined enhanced the rate of biological age recovery. And we've replicated this now in different systems. So it seems to be a way to potentially screen drugs that target aging, although of course more work always needs to be done. But anyway, I'd like to switch gears now and tell you another story, a newer story about intertissue dynamics of biological age. And ironically enough, this also starts from parabiosis. You've seen the data from the liver tissue, but we didn't only examine the liver tissue, we examined many tissues in this paper, and very remarkably, the results are perfectly consistent across every tissue that we analyze. Surprisingly, the most surprising for me was that the brain effect. And I'm not showing the data, but what we also found were that the effects were not only consistent, they were reciprocal between the two animals. So you see here the results from the young animals, which got older. The old animals became biologically younger. And after separation, they lived longer. My colleague Bohan Zhang wrote another paper in Nature Aging about the old animals because the effect was actually quite different. But this got us thinking, what if you don't take two whole organisms, but you take a single tissue and you put it into an organism of a different age? And so in collaboration with Stefan Tullius's lab at Brigham and Women's Hospital, we studied this model of heterochronic transplantation of hearts. So in this mouse model, it's not a transplant in the sense that a human gets a transplant, but rather a second heart is placed into the major vessels of the neck. This turns out to be very useful experimentally because you can monitor the heart function because it does beat. You can palpate this through the neck, and the original heart turns out to be a very useful control in this case. So we did a series of age mismatch transplantation experiments, and very remarkably, what you can see is if you put an old heart into a young or a middle-aged animal, it gets biologically younger, again, showing you representative results, but we analyzed multiple biomarkers. If you put a young heart into an old or a middle-aged animal, you get this very pronounced acceleration of aging and no significant effects across the isochronic controls. And now the question about reciprocity comes, does this tissue of different age feed back on the organism? And the answer, surprisingly enough, in contrast to parabiosis, is no, not really. So we looked at the native heart, the original heart that the mouse had, and there were not really significant or drastic effects across the board. And this is also true in the blood and the liver tissue. So in this system, the graft is very strongly affected. The rest of the organism seems not to care so much. And moving beyond the biomarkers, we analyzed a bit deeper the DNA methylation data. So what you're seeing here is a group of genes that harbor CpG sites that change significantly in the heart as a function of aging. And we compared, as with the metabolites before, the behavior during aging with the behavior during the transplant. And you can see that if you put a young heart into an old environment, you get almost this perfect correlation with the behavior during aging, showing that the young heart becomes biologically older. And the inverse is true if you put an old heart into a young organism. So the heart becomes biologically younger. For the sake of time, I'm not going to show you any more data, but we found consistent results at the level of gene expression, and interestingly enough, consistent results in humans, which received age-mismatched heart transplants. So to just sum up what I've shown you so far, I've shown you that reversible changes can happen in biological age as read out by these biomarkers. We believe that these are true effects on biological age because we observe across biomarkers, across data modalities, and across these very diverse models. I think the implication of this is that biology has harnessed some kind of age reversal mechanisms to be able to deal with these stress-induced increases, and studying those further, I think, would be very interesting. And, you know, fundamentally, I think the fact that biological age, this parameter, is dynamic suggests that there is a potential for biological age reversal. But of course, there's much more work to be done to prove that. However, you know, the lesson of the transplant, I think, is that intertissue age dynamics may present a roadblock. So for people who are proposing to target specific organs, you know, if you could magically rejuvenate the heart in an elderly individual, the question that arises from our data is, does it matter if the systemic environment causes the age to rebound to old, you know, in the space of just a few months? But leaving all of that aside just for a second, all of these studies are enabled by biomarkers of aging, which are, like I said, these revolutionary research tools that are rapidly moving from our preclinical world in the lab into the clinic. And, you know, in spite of this, there are a lot of roadblocks, a lot of open questions, a lot of basic questions that don't have good answers, like which biomarkers should we be using? What are they actually measuring? How will we ever get regulatory approval for these biomarkers? And these are big questions. These are questions that no one person or one research group is going to be able to solve. So to take action for this, we formed this consortium, Biomarkers of Aging Consortium, in late 2022. We've been working for about a year and a half now, and it's really taken off and been incredibly rewarding. And the impetus for this work is that, you know, the current status quo is that these biomarkers were able to quantitatively assess aging, but in the clinical context, that number that we believe represents something about biological age doesn't have any actionable insight for a clinician. So this really limits the translational utility. And the vision is that in the future, these biomarkers will be able to have some kind of actionable insight. So they might tell a clinician, you need to more frequently screen this patient or change their therapy or something like that. And this would ultimately increase the translational utility. But there are all these roadblocks that prevent this from happening. And even very basic things like data sharing. In the preclinical realm, which we work on, turns out to be a very big issue. If anyone here has worked with human data, you probably know what I'm talking about. But ultimately, this hampers systematic validation, and that's a big problem because that ultimately prevents us from getting these tools into the clinic. So we started by laying out this roadmap for validation and evaluation of biomarkers of aging, trying to lay the groundwork to get these things in the clinic. We published the first paper in 2013 and the second paper this year. We have several more in the pipeline right now. And in addition to the conceptual work, we've also been trying to build practical tools to meet some of these challenges that I've told you about already, such as unbiased benchmarking and validation, data harmonization, which turns out to be a non-trivial task. And we also try to foster some innovation in aging biomarkers so that we get the next generation of aging biomarkers, whatever number generation you personally believe that we're up to. But we have a set of initiatives. One is a tool we built called BioLearn, and the other is this Biomarkers of Aging Challenge. And BioLearn is an open source library for biomarkers of aging built by these incredibly talented people. It's on GitHub, so I would encourage you to use some of the skills you've learned from this workshop today. Go download BioLearn and play around with biomarkers of aging. What we have is a number of harmonized human biomarkers of aging. Seth and the team worked with the developers to make sure the implementation was correct, and also harmonized data sets. So you can either validate a new biomarker of aging or do unbiased validation of existing biomarkers of aging. And in parallel, we have this Biomarkers of Aging Challenge. We started it in December. It's going in phases. There are cash prizes. This is a snapshot of the leaderboard. You can see when we started this, we had some of the existing biomarkers of aging, like Horvath's on this leaderboard, and the competitors even now have just blown these out of the water. They're no longer on the leaderboards. You can see the mean absolute errors are down to 2.6 years compared to the previous maximum, which was on the order of 3.5. So that's all very exciting. We also have an annual conference series. We had the first one in December 2013. It was very successful. We have another one coming up at Harvard Medical School this November. If you're interested, please join us. Everything that we've accomplished with the consortium, I think we've done because we have this incredible group of people that make it up, to whom we're very grateful. If you'd like to learn more, agingconsortium.org. And finally, these are my personal acknowledgements. Thank you again for the invitation. If you'd like to learn more, you can scan that QR code. And after Cara's talk, I will be very happy to take questions. So thank you.
Thank you so much for your amazing talk, Dr. Poganik. Then let's move on next talk. It's my pleasure to introduce our next speaker, Dr. Cara Green. Dr. Green earned her PhD from the University of Aberdeen in Scotland with Dr. John Speakman by studying calorie restriction and aging. Then she studied a postdoc with Dr. Dudley Laming at the University of Wisconsin-Madison. At Madison, she continued to focus on aging and investigates the role of genetic background, sex, and proteins on metabolic health and longevity. Today, she will talk about heterogeneity and the response to protein intake. Please continue to post your questions using the Q&A button at the bottom of the Zoom screen. At that time, please specify which of the speakers you intended for. Okay, then Dr. Green, please start your talk.
Hi, thank you so much for that introduction. And I'd like to thank the ISB and Alice and Noah and everybody for making this such a fab day. And so today, this is going to be a little bit different. So I did molecular biology and biochemistry at uni, and then I did my PhD more in physiology and my postdoc more in physiology. But alongside that, I've started to get more into big data analysis and systems biology, but I use it more as a way to supplement the physiological work that I do on a day-to-day basis. So I'm just going to give you a few examples of how I've been using these bigger, more data-intensive techniques to try and dig into some of the work that I do and that I'm interested in. And so today I'm going to try and convince you that you aren't what you eat, but you are probably a mixture of your sex and genetics and your diet as well. And so if I can get this working, that would be fantastic. There we go. And so as an aging researcher, what we are usually interested in is how do we make people healthier? How do we increase the amount of time we live longer? And as a nutritionist, I'm interested in what I can eat to reduce the burden of age-associated disease. And as I'm sure you're aware, age has a huge societal cost. So it's a risk factor for lots of chronic diseases that we'd rather not get. It causes productivity loss, earnings loss, medical costs. They're in the hundreds of billions already in the States, and that's only going to get worse because by 2050, the American 85-year-old population will triple. And it also has, and probably most importantly, an unquantifiable social and mental health cost. You don't want to be spending your later years when you've maybe finally retired just feeling awful all the time. And so in my research, what I'm trying to do is extend this period of time before we start to gain these comorbidities. And I do that usually through diet. And one of the ways we do this is through low protein diets. So low protein diets can improve health span and a lot of these parameters that we associate with aging in both mice and in humans. And so this is a study done by Dr. Richardson at the University of Madison. So you can see, it's been shown many times by many groups. This is a glucose tolerance test. You inject the mice with glucose, it gets into the bloodstream. You see how long it takes for the glucose to disappear. If you cut the mouse's protein by 2/3, you can see you get this improvement in their ability to get rid of glucose from the bloodstream, reduce blood glucose at the fasting, and reduced insulin levels. And then this is a study we did at UW. The other UW, you can see this was over about 40 days, males on a low protein diet, and they had reduced body weight, reduced fat mass, and improved fasting blood glucose. Lower is better. And it also increases lifespan in the mice. So this is a complex graph. This is from Solon-Bio 2014. It's an amazing paper. Would highly recommend it to anybody. But what you can see is just focus on this red line, which is the lowest protein intake, and the mice are living longer on this low protein, high carbohydrate diet. And the best part about these low protein diets, to me, and I came from a calorie restriction lab, is that they occur without calorie restriction, which actually, if you look at most diets, the basis of most diets is just that they restrict calories, whether that's time restricted feeding, whether that's the Atkins, like you can only eat so much butter in a day. And the mice and humans, and I just said that, they eat more calories on these low protein diets and they see all these benefits, win-win. However, a lot of the recent dietary advice we get in the media is actually that we should be eating more protein and less carbohydrates. If you actually look at long-term prospective analysis studies in humans and in some clinical trials, you actually see that low protein diets in humans are associated with reduced cancer and mortality, and high protein diets in humans tend to be associated with a doubling of diabetes risk and an increase in cardiovascular mortality. So that's kind of the basis of why I got into studying low protein. The next step I took was I was writing a review with my boss, and I was looking at some studies where they looked at sex and genetic background and how that impacted the response to calorie restricted diets in mice. And I said, where's the study on protein restriction? And he said, nobody's done it. And I was like, bingo, okay, my first big project. And so I had two awesome undergrads on this study, Kat and Jesse. They're amazing. And we basically set out to look at how sex and strain affect the response to a low protein diet intake. So we've got these 3 strains. Those of you who aren't big mouse people, C57 Black 6, everyone's favorite strain, super responsive, always works. TBAs, not as responsive, tend to ignore whatever you feed them. And then the Het3s are genetically variable, and I'll talk a bit more about that later. And we had 3 levels of protein in the diet, 21%, 14%, and 7%. And we know that 7% extends lifespan. We didn't know what 14% would do. We put it in to see if there was a threshold effect. And so these are what our diets are made of. That's what I just told you about. Proteins from whey, the carbs are from cornstarch. And I didn't talk about the Het3s. I took that slide out. And the Het3s are genetically variable. They're bred from 4 different strains of mice. You breed them together, they come out all different colors. Kind of like if you mix like a black lab and a yellow lab, all the puppies are different colors. And that's what it's like with these mice. So they're a bit more heterogeneous. So we thought that this would give us a bit more of a robust response to these diets. If they had responded, that response would be more robust because they're not genetically exactly the same. Okay, and so what we saw in our C57 Black 6 male mice was that on these low protein diets in blue here, they didn't really gain weight over the 3 months that they were on diet. However, the control and the 14% protein, the medium protein mice did gain weight. So that's great. The DBAs, as expected, gave us the equivalent of showing us the middle finger. They weren't interested. Their body weights didn't change at all between the diet groups. And then the Het3s, like I said, were a bit more variable, hence these big error bars. But in general, the low protein diet mice also didn't gain as much weight over time. In the females, the female black 6 mice were totally non-responsive to these low protein diets, maybe very, very slightly, but it wasn't significant. The DBA females, again, not responsive. Maybe towards the end of the study, they did start to lose a bit of weight. And then the Het3s, again, were non-responsive. So at this point, we know that the low protein diet, at least 7%, worked in the males and not in the females, except for the DBAs who don't care. And we measured a lot of other things. We obviously measured food consumption. So as I kind of said earlier, the mice, the C57, most of the male mice on the low protein diets ate more, despite the fact they were losing weight. The Het3 females also lost more on the low protein diet. They didn't gain weight, but they didn't lose weight, but they were eating more calories. So that's pretty ace. I would go for that. We measured a lot of other things. I'm just going to go through this very quickly because it's not really relevant. But this would be the equivalent of all those clinical measures we were looking at in the patients earlier. We measured lots of different glucose tolerance attributes. The key takeaway here is that on L, which is the low protein diet, the blue triangles from before, the C57 males tend to have significant improvements across all these different measures of glycemic control. And the females also do, though not as many as the males. And we did something called metabolic chambers, where we put the mice in these little boxes for 2 days, and we see how active they are, how much energy they expend, that kind of thing. And again, we see that the males have increases in things like energy expenditure, but the rest of the mice groups didn't really. And that kind of accounts probably for some of that weight loss on the low protein diet, despite the fact they're eating more calories. But to really dig into this, what I did was move this figure so you can't read what's written, is I correlated all these different phenotypic changes with protein intake, calories from protein intake in the diet, and then just did some hierarchical clustering. And what we see is that generally all the sexes and strains tended towards the same response, despite the fact that they weren't always significant. So it's really just a magnitude issue. And so at the top here, this is traits that increased with increased protein intake. So lean mass was higher, which is pretty expected. But also things that we consider to be unhealthy were also higher. So fasting blood glucose, body weight, pancreas weight, insulin. So these AUCs, if they're high, it's bad. And then these are the traits that increased with decreased dietary protein. So here we have energy expenditure, activity levels, fasting insulin levels. These are all improved on a low protein diet. But what's really interesting is these middle traits, because they didn't really cluster high or low. And so these are the traits that are sex and strain variable. And so here we actually have adiposity, so fat relative to lean, and we have brown adipose tissue weight, we have insulin sensitivity, and we have circulating FGF21. And FGF21 is a hormone that is thought to increase energy expenditure. Previous studies of protein restriction in our favorite C57 black 6 mice, males only, if you knock out FGF21, you don't see any benefits of a low protein diet. So that was really interesting and exciting. I also did a PCA, which we've all learned about today, which is great. And we can see that there is some separation. So orange was our non-responders, our DBAs, and in black here we have our C57, so our positive responders, and then we have our Het3s in the middle here, so they share a bit of the traits from the DBAs and the C57s. And that makes sense because the Het3s are made up from C57s and DBAs, plus 2 other strains. And then if we look at sex, we actually see that males and females also separate out. What's interesting here is across the 3 strains, the female mice were actually less variable than the males, which if you're a mouse researcher is often the reason why people often use males is because everyone says they're less variable, but that is in fact a lie. I'm gonna get off my soapbox now. And then what I did was I looked at the variables. So again, we sort of learned about this today already. So these are all the variables that went into those PCA plots. And what's cool about this is showing basically how the data pulls in certain directions, and things that are correlated. If things are close together, it means they act quite similarly. And if things are pointing in opposite directions, then they're probably acting in the opposite. So we found that energy expenditure was pretty important for pulling apart these groups. And on the opposite side of things, how heavy the mice were and their lean mass was pulling them in the other direction. So these things were oppositely correlated. So this is just kind of showing us what might be important in terms of how the mice are responding to protein restriction and why they respond differently. I did a lot of omics analysis on this data. So from the liver, we did metabolomics, lipidomics, and transcriptomics. I took the significantly differentially expressed variables from each of those groups and concatenated it together with all of the phenotypic data I just showed you. Then we did something called a silhouette score to choose the number of groups we were going to cut these into. And then we did a big hierarchical clustering plot. And this is what we ended up with. So I know this is a bit complex. Well, probably not after what we've seen today, but for me, this is complex. And so we have our 6 different clusters, and within those clusters, we have usually a phenotypic change, a transcriptomic change, a metabolomic change, and a lipidomic change. And the reason this is really cool is because FGF21 and energy expenditure, which I previously told you were related, and they are still related, clustered in different sections. So FGF21 actually clustered more with changes in insulin, with thermogenesis, which you would expect, but also with mTOR signaling, which is related to lifespan, and energy expenditure clustered in its own separate cluster. Which was also interesting is that adiposity clustered with this longevity regulating pathway. And I'm an aging researcher, so I was like, oh, bingo. I looked at some of the genes in this pathway, and what we found was that a lot of these genes are actually associated with mTOR, so mammalian target of rapamycin inhibition, these genes here. And actually, if you inhibit mTOR, what you do is you extend lifespan in mice. There have been lots and lots of studies about that. It's pretty impressive. Now, so this is just a really exciting thing that we found just by concatenating all our data together and doing some clustering. And so broadly, what we found was that a short-term low-protein diet actually benefited everybody, but that magnitude was different depending on your sex and your genetic background. And we also did it in old mice and found that we saw improvements in old mice as well. But what I was really interested in was this genetic background effect, because sure, this is great. My 3 strains all responded differently, but there are much more than 3 strains of mice. There are much more than 3 flavors of humans. And so I really wanted to know, how far did this variation from genetic background go? And so my next study was born. And I also wanted to figure out if there were any key genes that might be driving whether you're a positive or a negative or a non-responder to a low protein diet. Because I think in terms of precision nutrition, that's where we're going next. So someone wants to go and age healthily, that is really going to be dependent on who they are. And so this was the next step in my study. I had another two amazing undergrads for this study, Annalisa and Grace, who are fab. They have recently graduated and they're on to bigger and better things. And what we did was we took 40 strains of BXD mice, which I'll go through in a sec, and we put them on control and low protein diets. We nixed the medium protein diet because it didn't do anything. And we put them on these diets for 8 weeks and we did a bunch of phenotypic testing. And so the BXD mice, for those of you who don't know, are a panel of mice. There's about 200. They're all genotyped. They started from a C57 black 6, so our non-responder, and a DBA, sorry, our good responder, and the DBA, our non-responder, and they bred them together, and they bred them together, and they bred them together, and they went through all these different recombination events until they ended up with this recombinant inbred set of mice. But within each group, they're genetically identical. And so what's cool, is they inherit different sections of their DNA from the different 2 parents. And what's cool is that you can see across these 40 strains of mice, the response to a low protein diet is massive. Some mice gain 6 grams, which doesn't seem like a lot, but if you're a 20-gram mouse, that's a lot. And some of them lost 5 grams. You can see our parent strains here. So our C57 black 6 on the left and our DBAs on the right. And what's really cool is that their recombinant inbred offspring, if you like, from the different strains actually ended up with more powerful effects than their parents, which is pretty cool. And then if you keep your eye on BXD 113 at the bottom there, and then you look for it in the new graph, you can see that this is the difference. This is the female mice, and you can see that between strains, there was huge sex differences. It was very uncommon to see a male and a female from the same strain responding in the same way. So this sexual dimorphism is also incredibly different between the different strains, as I'm sure it is between different people. And then again, our parent strains. And so the next step is to try and look at, oh, okay, no, it's not. If we look across the board at all our different phenotypes that I measured in this study, and then again correlated that with protein intake, like I showed you before, we actually see if something's in orange, it means that high protein resulted in a high value. So if you ate a lot of protein, you had a high fasting blood glucose. If you ate a lot of protein here, in the adiposity, you had a high adiposity in yellow and a low adiposity in pink. And so I think just to highlight here, what's interesting is that for most of the phenotypes I measured, there was a mixture. Some did well, some did badly. But actually, in terms of fasting blood glucose and glycemic control, actually a high protein diet was in general always negative. A low protein diet was always positive. And that's just kind of shown here. You see low protein versus control. They nearly always decrease their fasting blood glucose on the low protein diets in males and females. And the next step really was to try and identify what genes might be important for this. And this is kind of where we are in the study. This is all unpublished, and I'm still kind of following up with this study. This guy, so I use something called QTL mapping, which is quantitative trait loci mapping. There's a guy called Carl Bremen, I don't know where on the screen, but he's in the bottom left corner. And he's at UW, the other UW, and he actually wrote the code for quantitative trait loci mapping in R. And so you end up with this kind of thing, which I'll go through in a sec. And so this is lean mass change in males. So for the highest QTL for lean mass change, if you inherited that section of where that loci is from a C57 parent, you probably don't have lean mass change on a low protein diet. If you inherited that section from a DBA parent, you probably lose around 1.5 grams on a low protein diet. And this is interesting because we don't really want to be losing lean mass on these diets, especially if you think about healthy aging, sarcopenia, osteoporosis. So this is for lean mass change in males. So you do a bunch of coding that gives you a significance threshold. And so we found this lovely peak on chromosome 19, which is zoomed in here. And so each section is about 0.2 centimorgans. So it's a decent area of the DNA. And so for this section, there's actually quite a lot of genes in this position. So I can't really go into that right now because we're still sort of narrowing down our list. And then when we looked in females this was looking at fat mass change in the females, we found this lovely peak on chromosome 4. There was actually only one gene in this section called EPHA7, which has growth factor binding activity. And I looked at EPHA7 in some of our transcriptomic data from previous, and actually there are no changes, but it is sexually dimorphic, which I thought was really interesting. So it's significant for sex, but not for low protein diet, unfortunately. But what is interesting is I found this paper which showed if you knock out EPHA7 in mice, they actually become obese as they age. And so it's potential that this gene might have some interesting job for low protein diets, whether you gain fat mass or not. And so I'm going to zoom through this because I'm nearly out of time. But the plan for this study is basically to identify gene candidates, to cross-reference them with human GWAS data, and then to start validating some genes of interest. So in conclusion, everything affects how you respond to diet. So you are a bit what you eat, but you're mostly not. You're mostly who you are. There might be a role for lots of other things, sex hormones, epigenetics, exercise. That's all still to be determined. Generating context around this is key, I think, looking at how your genes interact with your phenotype, interact with your metabolites, etc. We've gone through that today, and I think that's going to be so important for understanding these changes. And what do we target research, right? There's only so much money. How do we choose what we start to look at? And I'm using this QTL mapping, but there's many other ways. So I'd like to thank the Lambing Lab. This kind of massive project takes a village, and the villagers are great. And all our collaborators and funding as well, and the ISB for having this thing. So thank you so much for listening, and I'm ready for questions.
Hi, thank you, Cara, and thank you, Jesse. I'd like to invite you and Professor John Newman to join us for the Q&A session. Jesse, can you please join us? There you go. Okay, so thank you so much for both of your talks. Both has been great. We've got several questions. I think I can open it up with a question of my own, a bit basic, Jesse, is to you. You talked about biomarkers of aging, but what were the criteria of, for a factor to be considered a biomarker of aging, and how many of them, or do you consider the question of targetability in their prioritization?
Mm-hmm. Yeah, so a very good question. Strictly speaking, a biomarker of aging is anything that predictably changes as a function of age and you can model. What we typically study are these integrative omic-based biomarkers of aging that use things like methylation profiling, RNA sequencing, metabolomic profiling, with many features. And then, machine learning approaches and more recently AI approaches are used to model these factors and to select the factors. To try to summarize this as quickly as I can, because this question goes to the heart of the reason that we wrote that 2013 Cell paper with the consortium, because you can categorize what's happened in the last 10 years, which is when these biomarkers have really exploded, with the evolution of what's gone on. So to your question of targetability, in the most general sense, no, that's not considered during the design of these biomarkers. These machine learning approaches, most commonly at the early part of that field, elastic net regression was used, and all that cares about is fitting the model using a minimum number of features, right? So, but this question of causality, targetability, all these things, this has become much more on the minds of researchers in more recent years. So we had a paper that was published by a really talented PhD student in our lab, Kijun Ying, last year, I don't remember, in Nature Aging, where he used Mendelian randomization to try to categorize CPG sites as either being causal to the aging process or adaptive as a consequence of the aging process. And he built biomarkers based on those subsets. So that's one way that you can introduce that kind of biological informed perspective to these biomarkers of aging. But that's kind of the cutting edge of where the field is right now.
I can start with the first question for Cara. Nice to see you again. By way of introduction for everyone else, I'm John Newman. I'm a geriatrician and basic translational scientist at the Buck Institute. Cara, really cool data about the BXD strains and the divergence of response to protein restriction. I remember there was a similar experiment around 10 years ago now, right, with showing a diversity of responses to calorie restriction. And you've obviously thought about this. There's similar but not entirely overlapping mechanisms. How much overlap is there in the strain responses between protein restriction and calorie restriction?
Yeah, that's a great question. Unfortunately, they're not really directly comparable because that calorie restriction study, which is excellent, they actually looked at lifespan. So they took all those strains and let them live for years and years, which good for them because that's a lot of work. I would say the range, if you looked at sort of magnitude over a number of strains, it would be pretty similar, I think. What you do find is it was also a different set of recombinant inbred mice. They use the ILSX recombinant inbred background, which is slightly different. But yeah, I'd say the number of highs and lows was pretty similar looking across our different phenotypes, which is pretty cool. We do restrict, the protein is restricted much more than calories. So because we restrict by 66% and the calorie, and that study was maybe 30 or 40%, maybe even 20%.
And one question from the chat, and please everyone continue to put questions in Q&A. Does the source of protein matter, plant versus meat matter?
Yes, it does. So we use whey protein for our mouse study, which is actually a bit difficult to categorize because some people categorize it as animal protein and some people categorize it as non-animal protein. But in the human work they've done, so a lot of the prospective cohort analyses that they've done in humans, they did pull out animal versus plant. And in general, what you see is if the source of protein was plant protein, you don't see the same impact on mortality, on cardiovascular health, and things like that. So there's probably been 3 or 4 studies out where they have actually looked at protein type. And yeah, it seems to be animal protein that is having the negative effect. There's also a couple
Oh, go ahead. No,
I was going to say there's a few questions in the Discord as well.
Okay. I just wanted to ask, in this context, have you had any experience performing mediation analysis, trying to see what's the role of the microbiome in mediating these effects?
No, I haven't, but I have the poop in the freezer. If anybody would want to do the microbiome analysis, I would love that, because there is a lot of interaction with the microbiome, and especially amino acids, which is obviously going to be a big part of this, right? Is there are some microbiota that can make small amounts of isoleucine, leucine, valine, and things like some of those essential amino acids. So yeah, I think that would be really interesting. I would love to do that. I'm currently trying to get enough money to do the RNA-seq, so it's on my list.
Jesse, there's a question in the chat which I shared from Magdalena Olson. Great presentation. Is it biological age reversal or biological aging pace reversal? In other words, could you just become younger or reduce the aging velocity? And I want to expand that a sec. Aging changes slowly as you measure it clinically, right? And for the traditional thinking about DNA methylation clocks was that they were relatively stable. Now we know differently. And that may depend, of course, on which DNA methylation clocks, what features, how is it trained. But if you're seeing such rapid changes in a short time, are you measuring underlying state of that slowly changing biological aging, or are you measuring a derivative, a pace of change? If so, we need both, right? We need to be able to both measure pace of change, but also know where we are. So how are you thinking about that with all the interesting data that you showed?
Yeah, so two things I would bring up. So first of all, we do need the pace of aging, and luckily we have that now from Dan Belsky's group. And to bring up Kaylin Ryan again, he's doing a lot of work with those biomarkers now. So we have biomarkers that are directly predicting the rate of aging. They're trained on longitudinal clinical traits from this really great study, the Dunedin study in Dunedin, New Zealand. And they built methylation predictors and made a composite score, which they call Dunedin pace. And it's unique among these biomarkers of aging because, unlike the traditional method of predicting, the age as a number that would correspond to chronological age, they predict a rate based on a baseline rate of 1. So, but anyway, the more philosophical part of that question, I guess, I think comes to this very important distinction between aging and biological age. We're actually writing a paper on this right now, because you're right, aging is a process that's slow. It happens over time. We believe, it's the essence of the process is accumulation of molecular damage. And so, reversal of that process versus reversal of biological age, which is not necessarily the same thing as biological aging, right? I think there's no reason why biological age could not be reversible or fluctual over short time periods. The question, of course, is how does that play into the trajectory over the life course? And that's something that we need to do more work to understand, but it's something we're actively working on.
In this regard, and with the context of this workshop, I had a question to Jesse of whether you have done any predictive analysis trying to predict those that would have rapid restoration of biological age versus slow restoration of. Could you find any would age play a part, or other components that you could identify that predict the trajectory of restoration?
Yeah, so that's a dream of mine, and we don't have the statistical power to be able to do it yet. But yeah, I mean, it's the obvious. It's an obvious next step of that research, because people ask me all the time. Uniquely in our study, we have these individual trajectories, right? There are very few of them. That's the problem, to try to do something like predictive analysis or deeper modeling. But it is clear from the individual trajectories that not the same thing happens in different individuals, right? And why would you expect it to? Different people age differently. Different people have different responses to within their biology. So my hope is that one day we expand these, and that's why it's so nice to see some of these results replicated in larger data sets. I think once we have enough of these compiled, we will be able to do that, and that'll be very useful. And it actually goes to go back to the consortium work. One of the big open questions in the field right now is the individual utility of biomarkers of aging. There are companies that are selling these tests to people and claiming that they can be accurate at the individual level. But the fact is we have no idea whether that's true or not, because all of the work has been done at the cohort level. And so as research tools in cohorts, in experimental settings, they're super useful, very revolutionary. At the individual level, we don't know how useful an individual biomarker value can be in terms of overall health, aging trajectory, et cetera.
Cara, as I'm sure you know well, so many mouse experiments in the past used male mice only or single strains. Obviously, we're missing so much by not looking at different strains and, of course, both sexes. And you showed a fascinating example of using a heterogeneous mix of mouse strains to better understand the biology at work. And that's awesome. We also use mouse experiments preclinically to try to predict responses. So every time I see a single cell atlas that's meant to represent mammals and people, every time you see an experiment in like an APP model, an Alzheimer's model, it's meant to predict human responses. What have you learned about how we should be thinking about preclinical mouse experiments? Should we, what is representative of human biology?
Yeah, I think that is the $1 million question, I guess. I think something we should definitely be doing is looking at males and females at the very least, which is something that I'm always trying to fight for. But yeah, I definitely think looking at something like the heterogeneous mouse model, because the nice thing about it is that multi-strain studies put people off, and rightly so, because you're doubling your N every time if you want to look at 1 strain or 2 strain or 3 strains. The nice thing about the heterogeneous mice, actually the National Institute on Aging use the Het3 mice to for their intervention testing program, which is their drug testing program where they test all their potential anti-aging drugs. And I think the nice thing about the Het3s is actually, or any mixed mouse model, is that you can individually genotype each one. And then actually what you can do is something like the QTL mapping, using each individual mouse as a strain. And so I definitely think trying to think about, a lot of it I think also depends on what you're trying to measure. But if you're trying to measure something as vague as weight loss or fat loss, I definitely think trying to increase your variability at the top level in terms of the genetic variability is going to just help you produce slightly more reproducible results when you get to the clinic. Because the C57 Black 6 strains, or any of those inbred strains, they have been bred for so long that they have now developed their own phenotypes and funny little things that humans just would never have. So yeah, I think more variability.
In this context, there's a question from the chat about that you mentioned that there was less variation in the female mice, and
Yes.
do you have to do any corrections for female hormonal cycle?
I didn't do any corrections on the female mice. I don't really know. I know it's got something to do with mucus plugs, but I don't really know how to test the female mouse hormonal cycle. That just came out of the PCA when I looked at sex. And I was so surprised to see that the females were less variable in their phenotypic response than males. I mean, these are young mice, which they tend to get more variable as they age anyway. But yeah, I didn't do any corrections. That's just the raw data, really.
And one from the chat, imagine for Jesse from Anonymous. Thanks for the great talks. How predictive are aging markers across different species, particularly in long-lived vertebrates and/or invertebrates like planaria, which seem not to age at all?
Yeah, so a very interesting question. And, you know, Steve Horvath has done a lot of work with his mammalian methylation consortium, different consortiums of the biomarkers of aging. But they generated many methylation profiles across ages for many, many different species. I think the final number was like 240 or something like that. And so they have a set of amazing papers, one in Nature Aging, with their universal mammalian clocks, one in Science about genetic traits underlying the epigenetic data. And it's quite interesting. I mean, I would encourage you to take a look if you want a deep dive in that sense. But I mean, one interesting tidbit I can give you is that you can build biomarkers of aging for species that are considered quote unquote non-aging. And the one that's most relevant to our lab is the naked mole rat. which some people may know is it's a rodent that's about the same size as a mouse, but unlike a mouse, which lives about 3 years, naked mole rat lives about 30 years. We think, we don't actually know the, the maximum lifespan. So people have suggested that the naked mole rat is non-aging, or at least demographically non-aging. But it turns out you can build a model of age using methylation data from the naked mole rat. We've done that in our lab. My colleague, Dr. Chaba and Steve Horvath did that in collaboration with some other labs.
And then a question from chat, I think for Dr. Green. At the risk of complicating things further, do we know the role of exercise or wheel running in the protein restriction story? Many of us non-nutritionists are familiar with the recommendation to eat more protein, especially when undertaking a rigorous exercise regimen, and curious how the protein restriction aging story meshes with exercise, eat more protein recommendations.
Such a good question, thank you. Yeah, so I will say those prospective cohort analyses in humans that I mentioned it's a whole population. And on average, our population is sedentary. So I will say those recommendations, and they're not recommendations because I'm not a registered dietitian, are in a sedentary population. However, what we have done is when we feed mice high protein diets, in the same way we feed them low protein diets, they gain weight and they have worse glycemic control. However, if we combine those high protein diets with weight pulling training, so we attach a little cart to the mouse, like a tiny little horse, and we fill that cart with weight, and we basically make them pull the weight every day. And so that's the equivalent of weight, maybe not every day, every other day. And when you combine the high protein diet with the exercise, you don't see those negative effects. So I would say, on average, there's nothing wrong with the high protein diet if you're using that protein for something. But if you're just eating it and not doing anything with it, then it seems like it can have some negative health effects. And I think we kind of think of nutrition very linearly, like, okay, I use up the carbs, then I use up the fat, then I use up the protein. It's like, it's not actually biological. We do mostly use carbs for energy, but we're using all three macronutrients all the time for different things. And so, yeah, that's what I would say. So yeah, don't give up your high protein diet if you're running a marathon.
Hey, we are at the top of the hour. I'm sorry we couldn't get to all the questions, and I personally have like 10 more questions. But I would like to thank Professor John Newman from the Buck Institute for joining me today, as well as our amazing speakers, Dr. Jesse Poganik and Dr. Cara Green. Thank you very much. And that concludes the seminar part of our workshop. You're welcome to leave. And we are now turning into our closing remarks. So we've had an incredible day of sessions and talk. We got great questions, great participation from you, the audience. Thank you again to our event sponsors, the UW Nathan Schock Center, OptiSpan, Olink Proteomics, and Phenome Health. And a special thank you to our outstanding speakers, our attendees, and our support staff. Specifically, I'd like to thank our instructors, Drs. Lance Pflieger, Max Robinson, and Alex Ralevski, our keynote speaker, Professor David Furman, our seminar speakers, Dr. Jesse Poganik and Dr. Cara Green, as well as Professor John Newman, our teaching assistants, Drs. Kengo Watanabe and Dantong Zhu, as well as Gwenlyn Glusman and myself, our administrative support staff, Dominic Lewis, Allison Kudla, Joe Meister, Audrey Hubbard, Connor Kelly, Victoria Ull, and Shana Brega, and our head of data science, Dr. Andrew Mages. We could not have brought this event to you without such a talented and dedicated team. As a nonprofit research organization, we rely on generous support of our donors. If you would like to support this workshop and other events at ISB, we would greatly appreciate it. You can make it an online donation at isbscience.org/donate. Please learn more about ISB at our website, isbscience.org, and continue to share your thoughts about the series on social media using hashtag ISB Systems of Longevity. You can also tag ISB via ISB SCI. Thank you all for joining us and have a great rest of your day.